Dear participant, a very good morning to you all. Namaskar. I heartily welcome you all to this international e-conference. In this present COVID era, it is really difficult to host a physical session, but it equips us to go virtually and disseminate knowledge. Of course, it also helps us to find out the best fraternity in the scientific world, and without expending a single rupee, we can host a very good conference, and the participant, they are reaping a very good knowledge. I heartily welcome you all again. Very shortly, we'll start the inaugural session. Please be patient. So now I welcome the organizing secretary, Dr. Pratibha Rani Deep, to speak out.
Good morning to all. It's my warm welcome to you all to the opening of the International E-Conference on Recent Advances on Biological Sciences. am delighted to welcome all the participants and members who are attending this conferences. In this pandemic situation, hope you all are safe. We are here again at the end of this year 2020 together in this scientific webinar organized by Department of Botany, School of Applied Sciences, Centurion University of Technology and Management, Odisha. It's my pleasure to welcome you all on behalf of Department of Botany to this December gathering. Welcome you all to this international e-conference, having a speaker from all around the world in this wonderful session. It's a glorious moment to extend my warm wishes to organizing secretary on behalf Department of Botany, Centurion University, Technology and Management, Odisha. I want to convey my heartfelt gratitude to our Honorable Vice Chancellor, Professor Supriya Patnaik, ma'am. And a special thanks goes to Supriya Patnaik, ma'am, Vice Chancellor, ma'am, accepting our invitation and presiding over the session in spite of her busy schedule. I must also thankful to Dean Madam Dr. Yashasvi Nayak, Technical Director and Mentor of School of Applied Sciences, Dr. Dipankar Bhattacharya, Professor Ashwini Kumar Rathore, HOD, ex-HOD, Department of Botany, Mr. Somnath Sarangi, Regional Director, sir, as the key advisor member, helping us in organizing this seminar or e-conference, without whose support it was not possible to organize such a seminar. I also thankful all the faculty members of all the campuses of the Centurion University of Technology and Management, and the students and the enthusiastic participants who have encouraged us and is actively to organize this program. I request you, all the participants, to start up and stand up and start the session by, we're starting the session by lighting the lamp. Hello. Welcome. Second session is followed by the welcome song.
या कुन्देन्दु तुषारहार धवला या शुभ्रवस्त्रान्विता या वीणा वरदण्डमण्डितकरा या पद्मासना या ब्रह्माच्युत शङ्कर प्रभृतिभिः देवैः सदा पूजिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापः।
Hello. Now, I request Dr. Rukmini Mishra, Department HOD, Department of Botany, Bhubaneswar campus. Welcome our ma'am, guest of honor, to address. Welcome and address.
Thank you so much, ma'am. A very good morning to all of you present over here. It's my honor and privilege to welcome our Honorable Vice Chancellor, ma'am, Professor Supriya Patnaik. Professor Supriya Patnaik, she is a ma'am. She has over 25 years of experience in the development sector and academia. She has earned a reputation of being a highly competent professional working with bilateral, multilateral, government and non-government organizations. She has demonstrated excellent relationship management skills with multiple stakeholders for policy advocacy and good program management skills. She has specifically provided advisory support in all programs on social development and governance aspects. She has maintained oversight of all the DFID programs in the state of Odisha over the past 15 years. She has also taken increasingly challenging roles in planning, implementing, monitoring, and evaluating the large programs with a focus on results. In addition to this, Madam has always maintained her interest in academics, being involved in teaching, training, supervision, research, and conducting study tours across several continents. She is presently holding a position of an adjunct professor at the RMIT University, Melbourne, Australia, and she is also a research fellow at the St. Petersburg State University, Russia. She has a number of publications to her credit. She is a very supportive, very positive, and very friendly person with the students, and we are blessed to have you, ma'am. Kindly grace the occasion with your valuable advice. Please, ma'am.
Good morning, everybody. Thank you so much for inviting me to this on this occasion. First of all, I'd like to again add my warm welcome to all the guest speakers, all my colleagues, and students who may be participating in this event. Thank you so much. It's a Sunday, and I know it's really asking everybody a lot. I just want to speak a little bit about the university um, and why it is that we are focusing on some of these areas, so that it sets the kind of tone for the entire conference. So we are just a 10-year-old university, and we have not had a lot of focus on research previously. And I might add that even though we are a 10-year-old university, our School of Applied Sciences even started later than that. So it's even newer, I might add. But I must say that, we have, for a 10-year-old university, we have moved a long way. And we have tried to, the team has worked so hard to bring it where it is today. I so we've so we started off initially only as a organization which is meant for training, education, and that kind, just like a traditional conventional university. And along the way, of course, you can see the what are the trends today. But I think we have always tried to kind of eke out or make our own path. So somewhere along the way, we decided that we also need to focus on research, and so that's how we have entered into research. And as my colleagues and everybody is aware, only for the benefit of the guest speakers, that we now have several research centers, and which every one of our faculty is attached to, aligned to, and based on their areas of interest, and they're all working towards that. I think there is a long way to go. But what I might add is that while we, and in a way, I'm glad that this is called recent advances in biological sciences, so that we don't kind of end up working in our own silos. I think the world is increasingly interconnected and increasingly boundaryless on one hand, and very boundaried on the other hand. But I think we should look at how we can look, even though these research centers have been created, it is how do we work across centers, how do we collaborate across centers, how is it that we can do research that is more meaningful and relevant to the current society in which we live and for the future. And I think that's what we are really aiming at. And we that's why I mean, it's been wonderful that we've been having these series of webinars and conferences on recent advances in various fields. And I think that's where our faculty and students will benefit the most. I look forward to the deliberations that come out of this, and what it is that will help us in our agenda that we have set ourselves, and how is it that we can move forward in some of these areas to uh, to address some. Again, I would like to say, we keep on focusing on what is relevant to society. Especially, I keep mentioning this because I'm a social scientist, and I believe that research should not be done for research sake alone. It should be done for uh, for its relevance to society. And therefore, I think that it's very important that some of these areas are actually addressed. I think also there's a wonderful and great opportunity. I think that we have so many things, so many areas that are unexplored. In fact, I think, at least in the field of social sciences, sometimes I think we are under researched. I am not in a position to speak about science, but in the social sciences itself, I think in India, we are under researched. Every time I think of something and then I start looking for what kind of material there is a lot of material. But when you actually start looking at the more critical analytical work, there isn't much. So therefore, I think there is a great opportunity to move into those areas. And I look forward to all of you discussing some of these areas, and charting out a new agenda, or a new path, that this is the area that we as a university will focus on, and these are the kinds of things we'll do. And I hope that some of it emerges from these various deliberations you're having as a school, and you can then look at what it is that you would want to explore. So I think with these few words, I'd just like to welcome all of you again to this event. And I wish this webinar conference big success. Thank you all.
Thank you so much, ma'am. It's always a pleasure to hear you. And truly, ma'am, whatever you said, it's very true. There are many areas are still need to be explored. And these kind of conferences and webinars are the platform where we can just know what are the areas, what research is going on around. And it's a platform for the students, also the faculties, to know those kind of field where we can work on. Thank you so much, ma'am. Now I welcome our Dean of School of Applied Sciences, Honorable Dr. Yashaswi Naik, ma'am. She has a PhD in environmental science with specialization in poultry management and vermicomposting. She has 16 years of teaching experience and also possesses specialization in poultry management, vermicomposting. She has published research articles in national and international repute. She has also undergone few training programs. She is a very active leader. Thank you so much, ma'am, for coming. Kindly grace the occasion.
Thank you so much, madam. Good morning to all of you here. Honorable Vice Chancellor, madam, Dr. Supriya Patnaik, madam, respected mentor director, sir, Dr. Dipankar Bhattacharya, respected regional director, sir, Dr. Pradeep Sadangi, sir, respected principal, sir, Dr. Somnath Sadangi, head of the department of Bhubaneswar campus, Dr. Rukmini Mishra, madam, head of the department, Bolangir campus, Dr. Shantanu Bhattacharya. I also extend my warm welcome to the eminent guest speakers, scientists, dear faculty members, and my dear students. It's my privilege and great pleasure to be a part of this international conference, that is Recent Advances in Biological Sciences. Biology is fast moving from being a descriptive field to an exact science, a transition that will increasingly require input from all the branches of natural sciences. The cynosure of the conference will be on biological sciences and innovative research, tie-up, enhancing frontline studies in almost all aspects of modern biology. The core vision of the conference is to provide an excellent academic forum and interactive platforms for enhance of ideas, views with participating members on the emerging trends and challenges in research in the field of basic and life sciences. Its objective is to abreast the emerging scientists with latest developments of this field by providing them an opportunity to enrich their vision in research by undertaking the stock of present studies of research and to gather future research work. In my view, this conference will definitely groom the minds of young scientists to face the challenges of future for effective and efficient research and also the professional responsibilities. I welcome you all in this international conference. Thank you so much. Thank you, madam. Thank you so much.
Thank you so much, ma'am. That is really very much motivating. Thank you so much, ma'am. So now I hand it over to Pratima ma'am for this.
Thank you, ma'am. Thank you, ma'am, Yashasvi ma'am, and VC ma'am also for taking out your time and participating. It's really very encouraging for us as well as the young enthusiastic participants who are looking forward and are part of this department as well as it is university. Okay, thank you all of you. Now I request Dipankar sir, Dipankar Bhattacharya, Technical Director and Mentor, School of Applied Sciences, Century University of Technology and Management, to place his speech. This brief description of sir. This is Professor Dipankar Bhattacharya, did his PhD on biosensor from Jadavpur University. He did his postdoc at University of Quebec at Canada and later joined there as research professor. His research area in Canada was machine learning and chemical engineering. He has worked with many renowned aluminum companies such as Alumineri, Elote of Canada, Hydro Aluminium of Norway, Rio Tinto of France, etc. Coming back to India, he joined Centurion University of Technology and Management. He was the Dean of School of Applied Sciences and now working as mentor and technical director of Gram Tarang Food. He has around 200 publications in peer-reviewed journals, 5 patents, including 1 US patent. He has students from around 20 countries. I welcome Dipankar Bhattacharya, sir, to place his speech. Over to you.
Thank you, madam. Thank you, madam. Is it audible?
Yes, sir.
First of all, I thank our Honorable Vice Chancellor, Madam, for her kind support. I also thank Professor Pradeep Sarengiji for promoting Bolangir in all aspects in research. I also thank Professor Somnath Sarengiji and Professor J.K. Meher for promoting a lot of research in Bolangir and this kind of conferences. Dr. Jyoti Prakash Roth and Dr. Shantanu Bhattacharya to connect renowned researchers all over the world. They are doing it in a series like this. I thank the organizing committee, the Department of Botany, all my fellow faculty members, my colleagues, and dear students. The topic of today's conference is recent advances on biological sciences. Now, in today's life, biology has really encompassed all our lives. So different aspects of life we cover through biology. And the major areas where biology has a lot of influence now in recent times, it's cellular and molecular life sciences, plant sciences, pharmacology, ecology, agriculture, etc. So it covers practically all the different areas where we can think of. To make our life really smooth, it is working like anything. Now, today, the guest speakers, I welcome them. Actually, I was going through their work, and what I found that they are going to help us enhance our research activities in Centurion through their speeches. So the different areas where we are going to focus today are the omics, which aims at collective characterization and quantification of biological molecules. And it covers genomics, proteomics, and metabolomics. Also, I found there will be some discussion on next-gen sequencing for rapidly sequencing of whole genomes, then microbial biotechnology and enzymology, which has some contribution in pharmacology also. Then protein detection, and I found Dr. Narendra Pradhan. He has worked on this proximity ligation array on protein detection also. So I was checking these kind of things. And finally, there is bioinformatics also. So it is actually covering all the different research areas possible in biological sciences. And there is one talk on intellectual property law, which is really important in the context of any research in biological sciences. So these are the areas that we are going to cover today, and we will learn a lot. And these researches are aligned with some research centers of Centurion University. For example, the Genetics Research Center, and then the Phytopharma Research Center, Pharmacy, that research center. So, and also even agriculture, they might have a lot of information in that. So these researches, these presentations will be aligned to our research centers also. So there is a lot of opportunity for us to learn. So finally, I thank our invited speakers, Dr. Narendra Pradhan, Dr. Shukanto Bhattacharya, Dr. Niharika Bhattacharya, Dr. Ranjan Nando, Dr. Sandeep Dhanda, Dr. Sonalisa Pandey for their illuminating presentations. So I welcome all of you and thank you.
Thank you, sir. Thank you for such a wonderful speech and also introducing other members who are present other than our university about the prospects and future of our university and opportunities present in our university. Thank you, sir. Now, I welcome our regional director, Sri Pradeep Kumar Sarangi of Palangir campus to present his speech. Sir Pradeep Kumar Sarangi, Regional Director, Century University of Technology Management, Balangir Campus, Odisha, is an industrious person. Because of his industrious effort, the regional campus took shape in this backward district of India, that is Balangir. A man of words, Mr. Sarangi gives 100% to this institution, and in just 10 years of its inception, Balangir got a second higher education center after Rajendra College. Going with the mandate of Centurion University, he started the mushroom cultivation, phenol production, poultry farming to give employment, integrated education to the students to make student industry ready, which is duly recognized in the new education policy. Welcome you, sir, and thankful to him for whose support it was not possible for us to arrange such a conference in such a short span of time. Now I welcome Pradeep sir to present speech.
Thank you, madam.
Over to you, sir. Thank you, sir.
Thank you, madam. A very good morning to all of you. Esteemed Bhishi madam, Dr. Yashaswi Naik, Dean School of Applied Sciences, Professor Dr. Dipankar Bhattacharya, Director Gram Tarang, all dignitaries of this virtual conference, and my dear students, I heartily welcome you all of the International Conference of Recent Advance in Biological Science. From the inception, Century University's agenda has been shaping lives and empowering communities. Going further with the agenda, Century University Odisha has incepted a regional campus in educationally and economically backward district, Balangir, to empower and employ the community. The entire focus of the campus is the integrate education with employment. We inculcate the skills in the students from formative age and make them market ready to face all the situation like in this pandemic. Our campus successfully implemented mushroom culture, vermicomposting, etc. Aligned with our mandate, we have a pool of renowned and experienced faculties. We offer bachelor's, master's, and PhD courses in various disciplines. Many of our students are successfully placed and working in reputed organization. I'm happy and confident that such a noble initiative taken by the Department of Botany will not only help us in spreading the utility of the subject for the scientific community, but will also inspire our students for understanding the depth and importance of recent advances in biological sciences. I congratulate the convener, organizing committee members, and coordinators for hosting the international webinar within a short span of time and wish the conferences as a grand success. I am sure you all enjoy the session. Thank you all once again. Thank you all.
Thank you, sir. Thank you for your wonderful speech. Now we are moving towards our technical session. Keeping in mind the young generation, so we have designed the technical session which covers all the aspects of biological sciences and the recent research taking place in biological sciences. So now I hand over the mic to Dr. Jyoti Prakash Ratho for the technical conduction of the technical session. Thank you all.
A warm welcome again. May I the technical.
Thank you, Dr. Rathod. So, well, and very good morning to all of you, and I welcome to this international conference on recent advances in biological sciences. And now you already almost uh, complete that introductory session. Now we'll move to the technical session. So our technical session has been divided into three different sections. In the first section, we'll have a wonderful talk by two eminent speakers from the USA, Dr. Sandeep K. Dhanda and Sonalisa Pandey. And this session will be chaired by Dr. Ranjan.
Good morning to all of you. Hello.
Nobody is audible. Is somebody speaking?
No, ma'am, maybe some problem. Just check.
Good morning to all of you. Is it audible?
Yeah, it's audible.
Yes.
Yeah,
Now,
I think now Dr. Jyoti has to introduce Dr. Narendra Pradhan, the
Yeah,
first invited talk in this morning session. Dr. Jyoti, please.
Yes, Dr. Nanda, I'm quite available. Well, as the agenda goes, in that first technical session, we'll start with Dr. Sanjeev Dhanda sir. then followed by Dr. Sonali Pandey, and you will chair the session. Dr. Ranjananda will chair the session. And finally, Dr. Narendra Pradhan will speak out in the third technical session because it is still midnight in Sweden. So that's what the time zone forced us to schedule likewise. So little bit change was there in the agenda, and as I discussed the chronology of the speaker, the session will goes like that. We'll start the session shortly.
या कुन्देन्दु
Yes, we'll hopefully, because this is a winter morning, so that's what we should start early. That so that we can finish up and don't bother the participant to wait till late evening. Hopefully, I am audible there.
Yes.
So, a hearty welcome to all participants. Very good morning. Of course, it is really hard to wake up in the this chilling winter and attending, and it is not a music session, rather an academic session. So we always acknowledge the participant enthusiasm to wake up early and get ready for the session. A sincere acknowledgement from the organizing committee goes towards them. So this session, the first technical session, will have two speakers, Dr. Sanjeev Dhanda from US and Sonali Pandey from San Diego, US. And the session will be chaired by Dr. Ranjananda, who is the group leader and heading the Translational Health Group at International Center for Genetic Engineering and Biotechnology, New Delhi. So it is really, I feel myself proud to introduce Dr. Nanda, an eminent biologist, a handful of biologists to be counted in India who has got the Bill and Melinda Gates Foundation research grant to conduct the research. I believe when he got that award, only 4 people could able to make it. And of course, as the image suggests, that is the present picture of Rajananda, Dr. Nanda. So he is just 42. So you can imagine how much he has made effort to lead the scientific community of India. Dr. Nanda, a son of Odisha, he had his bachelor's from Utkal University. Then he marched on to School of Life Sciences, done his MSc way back in 2001. And he was the first JRF Junior Research Fellow awardee of that batch. And he was called upon by the Professor Rintu Banerjee in IIT Kharagpur to join his group, where he spent quite good of his training time to build up his career. He finished his PhD, then he went to the capital, New Delhi. So after PhD, every Indian student thinks how to get out of India. But Dr. Nanda has a flip idea. He rather tried to save India, rather tried to build his career in all the hurdles in India. Believe me, when he joined ICGB as a research scientist, the mass spectrometry was not at an working stage. And it was Ranjan Nanda's hand that makes the mass spec work and produce paper like printers. So, and there is no ancestry of lab to put Ranjan in ICGB group. It was his own effort. He made his career, he saved his career on his own. And within a short span of time, he could able to get the Bill and Melinda Gates Foundation support to develop the electronic nose for early detection of a quite age-old disease that is prevalent in this tropical region of India, the TB. So he garnered the support of Bill and Melinda Gates to develop this electronic nose. Then, and he keep on working and duly recognized by the scientific fraternity, leading to the scaled his leader, and he leading to the group leader of Translational Health Group at International Center for Genetic Engineering and Biotechnology, New Delhi. I welcome Dr. Nanda to chair the session. Then our next speaker, it's time to move to the next speaker. Let me introduce our next speaker, Dr. Sanjeev Kumar Dhanda. Of course, a bioinformatician. He completed his early education from government school and college in Hisar district of Haryana. Then he earned his master's degree in science at MD University, Rohtak. Then after finishing his early education, he joined his MSc, of course, in my center, Bioinformatics Center in Jawaharlal Nehru University, New Delhi. Then he finished his PhD from IMTech, Chandigarh. Then he got a postdoc from Division of Vaccine and Discovery at La Jolla Institute for Allergy and Immunology, after which He joined as a research scientist in Department of Oncology, St. Jude Children's Research Hospital, United States. He's enthusiastic to develop bioinformatics tool to address current biological problems. Currently, he is working as a research scientist at St. Jude Children's Research Hospital at Memphis, US. Welcome you, sir. Now the plot to Dr. Nanda. So now we can start the session.
Thank you, Dr. Raj, for
So, Nanda, please. Yes, sir. Please go on.
So thank you, Dr. Raj, for your kind words. And now we'll welcome Dr. Sandeep for his kind presentation. Dr. Sandeep, please.
All right, thank you very much. And I think my screen is visible and I'm audible to everyone, right?
Yeah, it's clear.
All right, thank you very much. And I would like to acknowledge the Centurion University of Technology and Management University in Odisha and the School of Applied Sciences Department of Botany for organizing this wonderful conference and inviting me as one of the speakers. I would like to thank all the leadership there for giving me this opportunity. Today, I'll be talking about future and career prospectus in bioinformatics. And I'm currently working at St. Jude Children's Research Hospital as introduced by our leadership in Centurion University. And with that, let's move on to my presentation without wasting much of the time in this stuff. And this is going to be the outline of my presentation. I will start introducing the bioinformatics because I know there is a huge different diversity in the background of the people. People are coming from the bachelor background in the master's degree, and some of them are also faculties. So I'll briefly introduce my understanding of bioinformatics. Then I'll go through some of the example from my personal experience, like what did I learn from there being in bioinformatics. Then, as I mentioned, it is going to be a career session as well. Then I will mention what are the skills that you are supposed to have if you are willing to continue your career in bioinformatics. Then I will touch a little bit of my understanding about the current scenario, how bioinformatics is growing and how you can make your career there. And then obviously I will touch some fundamental about the bioinformatics and India, how it is growing well in India and how we can see a bright future there as well. And obviously we are not going to talk the bright spot of bioinformatics. I will also go through the practical issues being in bioinformatics. Obviously there is some dark side that I will briefly mention. And at the end, I will go with some take-home message, what is the take-home message of my understanding being in bioinformatics for around 10 years. So let's start with the introduction part. So introduction to bioinformatics. In a simplistic term, bioinformatics is a marriage between biology and computer science. Obviously, we are using all the computational technology, but we are trying to address the biological problem. And the end product is the bioinformatics. So it's a lens of computer science to see the data in the biological or biomedical sciences. Obviously, I'll be going through most of the applications that are there in the medicine because of my experience aligned in that direction. But if you people are from the botanical background, you'll also see the bioinformatics is contributing in a huge amount in your field as well. I do not have much experience in application of bioinformatics in plant sciences, but if you go through the details of it, I will show some of the people who are working there. And if you read their research paper, you will get to know how you can contribute even within the botanical research and how you can still opt for the bioinformatics part. And these are some of the applications that I could list here. There is a huge list, but I could not list everything in just one slide. And this small list include the medicine, vaccine, pharmaceutical drug design, toxicology, molecular evolution, biosensor, biometry, biological computer modeling, or the DNA computing. These are the small set of the applications that are there in bioinformatics, but the complete or the comprehensive list will be way bigger than to be included or could be completed in one slide or even one presentation. Let's go with a brief history of the bioinformatics so that we understand from where we are starting. So the biometrics was there even before 2001, but there was a big boost in 2001 because of this Human Genome Project. The Human Genome Project was actually published in parallel in two different well-renowned journals, the Nature and Science, on the same day, and they both published the Human Genome Draft. And I will touch upon how it was started. Obviously, I'm saying biometrics was there even before 2001, but there is a big boost and that came out after these publications, because these publications had opened a new era. And this was a man in one of the publication right there. His name is Craig Venter, and he was leading a private industry, Celera Genomics, that time, and it was single-handedly trying to sequence the complete Human Genome Project. At the same time, the public efforts were mediated through different countries and NIH funded in US, and there was a different set of the cloning that people were doing for the Human Genome Draft. And ultimately these effort bring fruits, and they published in Nature and Science at the same time in that 2001. And if you look at now, so obviously Genome Project was started in 1990, and it took around 11 years to complete the first Human Genome Draft, which was published in 2001. Then if you see later on, it started a boom of biometrics, and every country is now putting effort to sequence their population, and which was initiated in 2008 by calling it 1000 Genome Project, where the goal was to sequence not only just one individual, but thousands of individuals. And in 2010, UK started to sequence their population, and they started with 10,000 people. And then every country start jumping into this race when UK announced in 2012 that we are going to sequence 100,000 individuals, like 1 lakh people in our country. Then the different country followed the same suit, like Saudi Arabia started in 2013. Similarly, Australia started in 2016. Dubai started in 2018 with 100,000 individual. USA also jumped in the same race with 1 million target in 2015. And similarly, China took same kind of target of 1 million sequencing of their population in 2017. India has also started initially with 20,000 people, and now we are marching forward in that direction. And we publish this kind of understanding in Bioinformation Journal as an editorial, and you can find out the different perspective of genome cataloging in different countries. Moving on further with biometrics in pandemic, obviously biometrics has played a huge role here as well, because we are talking about the recent advances, and pandemic science is the most recent advance that you see. And biometrics has played role significantly in urgently needed diagnosis, how you can have diagnostic in primer designing, and how you can come up with a PCR reaction so that you can find out this virus using some PCR reaction. There is also effort from the biometrics using the drug repurposing or monitoring the viral genome, how it is evolving. Like you have heard of this news, there is a new strain coming up, and all this is being discovered because there is underneath biometrics playing the role in sequencing the genome. And once they sequence it, compare the genome and find out these are the mutations which are contributing to this new strain. And that's why we are able to discover this in a faster manner. And obviously, I'm going to touch upon the help designing in the better vaccine. All of this understanding was published by another colleague here, Sebastian from National University of Singapore, in this Health Informatics journal. And now I will touch upon the personal experience coming from the help designing better vaccine. When we talk about the vaccine, we need to understand what's the base of the vaccine. And if you describe the very fundamental in the vaccine part, and before that, I would like to touch upon how I moved in different career perspective. So I started my bachelor's degree in biology from Kurukshetra University, and then I did my master degree from MDU in biotech. Then I moved from the biotech to the biometrics in my PhD degree in a joint PhD program at IMTech GNU. And I did every research in Chandigarh from 10 to 16. Then I moved to another part of the world for my postdoctoral training at La Jolla Institute. And currently I'm working as a research scientist at St. Jude Children's Research Hospital. So that's my journey. And I want to give you an idea because I came from the biology background. I was a master's student of biotech, and then I moved in biometrics or my PhD. So it just to open up your ideas or your understanding that you can switch your field and still you can do good in the other field as well. And as I said, like I'm mostly working on the immune response understanding. And if you go the base of the vaccine design or any immune response for that matter that is generated against infection, initially when infection encounter our body react to it, and we call it primary immune response. And we measure the immune response in case of like antibody concentration, or it also like cell mediated immune response, which is measured in terms of T cell population, which I'm going to touch upon in the next slide. So our initial encounter with the pathogen rises to the primary immune response, which is a small peak. But when the same antigen or the same infection comes again, it's called secondary exposure. The immune response react not only quicker, but the magnitude is even large. So you can see that's the base of the vaccine. So we are trying to achieve this kind of immune response with the vaccine, so that when actual virus is attacking our body, we can have a quick immune response and we can eradicate the pathogen in a small amount of time. So with that understanding, the immune response can be classified into different immune cells, and these are called innate immunity or the adaptive immunity. Innate immunity, that is, we are getting once we are born, so we are getting inherited this immunity, and these are different cell type that are still there, and they are non-specific. They'll be reacting in the same way for every infection, and these include the dendritic cell, natural killer cell, or protein component, complement protein, or neutrophils, eosinophils. These are the part of innate immunity. And there is another part, the adaptive immunity, which is obviously the slow in response. But if you see, this is very specific in nature. And if we classify the different immune cell type, the B cell, which generate the antibody, which we have seen in the last, like how we can measure the immune response in case of antibody concentration, or the another is like T cell, when it's called cell mediated immunity. So here you can generate the T cell immunity, and it recognized by further diversify by CD4 cell or the CD8 T cells. So this is a bigger picture of immune response, and every immune response, they are trying to identify certain epitope, and I'm working mostly over the epitope data. So, and one of the points that I want to touch upon the recent advances is about pre-existing immunity. Pre-existing immunity, even when the infection is new, but we still see a kind of existing immune response because of some kind of similar pathogen that our body already has encountered. So that kind of immunity is called pre-existing immunity. That means immune response already exists even before we got infection. And I will explain this pre-existing immunity in terms of coronavirus in our upcoming slides. And why pre-existing immunity is very important, because it explains the disease severity in different individuals. And let's take an example of coronavirus. You see some people are not getting infection at all, and some people even get infection. Then once they get infection, they recover so quickly. And in some small set of the population, it becomes very severe. So pre-existing immunity is one of the way to explain this different individual, why they are having different disease severity. And it also helps in understanding the vaccine outcome. Obviously now like 3 or 4 vaccine have been already approved. So how we are going to react to this vaccine is the one of the reason that we can explain this understanding is pre-existing immunity. So when we are talking about too much about the pre-existing immunity, here I would like to describe my contribution in pre-existing immunity. So when we say something is pre-existing, it was existing even before the infection. There are 4 different paradigms that we need to understand. And these 4 different paradigms are explained here in different 4 quadrants that you can see in different 4 pictures. I will start from the left top here, like what exists so far. We need to understand what immune response already exists. So for that, we have created a database called Immune Epitope Database, and which was published in 2019. Obviously, the database was there even before 2019, but one of the update was published in 2019. And you can find all the different epitope and how did they react in our body, or not only in our body, how did they react in different, let's say, different species. How did they react in mice? How did they react in rabbit? So that we can understand a bigger picture of the immune response. And you can find all the different epitope and how they are reacting to different species is already been cataloged there with the help of bioinformatics. And this is called IDB. Similarly, once you know all the immune response, then you need to find out there is a patterns, like what kind of pattern is there in existing data. And I involved in one of the app that can show you the immune response over a length of the protein, and which is called Immunome Browser. In this Immunome Browser, you can see where we can see the peak of the immune response and where we see the knowledge gaps where nobody is studying that part of the protein. So we can connect the link in the pre-existing immunity if we have such an app. And similarly, if we go from existing data to extending this information to the new data, like let's say for novel coronavirus, how we can exist this kind of information. And for that, we need to have two different sets. We need to first cluster, and I was involved in one of the tool to understand the clustering of the known data with the unknown data, and which was published in Immunology in 2018. And similarly, how to fill the gap in the knowledge. Obviously, not everything will be known there. Once there are certain epitopes which are overlapping with the known data that you can find out with the clustering, but you also need to know what kind of immune response they will be having. And for that, we have a compendium of different resources that are constituted together as a called immune epitope database and analysis resource. And it has different sets of the tool where you can find out the immune response of certain sequences like T cell epitope or B cell epitope, and you also have the population coverage kind of tool in your epitope set. So in pre-existing immunity that I was mentioning about in context to coronavirus is understood by these different four tools. And if you go in detail of this tool, these all or the bioinformatics software that are contributing to this understanding. And one of my colleagues in the lab took this challenge, and she find out all the epitopes that are there from the SARS coronavirus, and she concluded they are not matching with the other species. Let's say to the MERS species, they are not matching with those species, but they were matching for the common cold viruses or the previous coronaviruses using the protein homology methods. And then they came out with the conclusion how we can find out the exact epitope. And they say took all these different four tools for B cell and the T cell epitopes and find out there are 4 different epitopes which are a part of this novel coronavirus and were also a part of the previously known coronaviruses. And similarly, she done the same thing for the T cell epitope and find out around 12 epitopes that are there, which were common between the existing coronaviruses and the novel coronavirus. And with all this information, once it was validated in the lab, it was published in the Cell. And now you can see all this mentioned tool that I have shown you earlier, the immune epitope database analysis resource or the IDB or the clustering tool, which were here, which become the fundamental part of this tool. And this research highlighted not even by many individuals, which was highlighted by the top expert in the US, Anthony Fauci, in one of the congressional meeting when he said how we can understand the behavior of this immune response in coronavirus. And he waived this paper. Yes, there are some fundamental paper like that, and we need more paper like this. So it gives us a big boost that whatever we are done using the bioinformatics could become the base of the experimental validation and therefore could be very useful. So I want to stress upon again here, once you are having a good understanding of the biology and you know we can develop this kind of tool, then it could be really useful for experimental validation and could address many of the fundamental questions. So that's all my understanding. And with that all, I would like to mention my colleagues at IDB team. These are the different people in 2019 when I was a postdoc there. And let's move on with the career part. And as I said, when we talk about the career, we need to understand that bioinformatics is an amalgam of 3 different disciplines, the stats, the biology, and the computer science. So you need to have the basic understanding of all 3 if you wanted to succeed in bioinformatics. And the biology part, obviously one should be able to connect the dot in the relevant biological problem. Like you should be able to understand there is a problem in biology and how you can address. And in my understanding, I would say like you must understand how many pathogens are there and how they are reacting to our body. There should be enough understanding that you must carry about the disease and its spread, and it's a physiology and genetic that are playing role there. And if you talk about the second part of his computer science, once you have the biological data in your hand, you must understand the computational architecture, the programming languages, and how you can integrate the data from the different sources. Obviously, one lab will be working on one data set, the other lab will be working on the other data set. So you must have a vision to integrate using the computational technique, how I can combine the knowledge from these two different labs. And if we talk about the stat, obviously you need to show your results are significant. So you have understanding of those different stats there, how they are playing the different parameter, and how I can show these are really significant results, and point out the outcome that you wanted to take further in experimental validation. And let's talk about the current scenario. Obviously, we have touched upon my understanding and the skills required. If you talk about the current scenario, every health company, starting from Amazon, Google, or every name that you know from Facebook, or they are jumping into the healthcare. And their easy way is bioinformatics, because they are very good in understanding the computational architecture and programming languages. So there is an easy way to enter into the healthcare. And that's why they are investing in billions or the trillions, and they have set up their own kind of startup in many institutions. So there is a huge scope that you can jump into this wave. And if you talk about in context to another startup, the Ancestry is one of the startup here, and there is another startup in India called Map My Genome, which is mapping your genome using the bioinformatics technique. And there is another startup at 23andMe are coming up. So there are many more startup are joining, and every day you see their new startup are coming up, which has a bioinformatics at the pace. And if you talk about particularly in concept of India, now I would like to point out some of the like extraordinary scientists in India that I have experience to work with, or I know them in person, and I would like to highlight their research work. And before that, not just the scientists are working in extraordinary effort, but also the government is putting a good infrastructure there, and they have organized this hackathon in 2020 so that they give open this challenge, how you can discover the drug using the hackathon. And once you have your idea or your computational idea, you can submit your model. This was an open innovation model started by MHRD and principal scientific advisor in Government of India. And now I'd like to point out these scientists, what they are saying about the bioinformatics. He is Dr. Vijay Raghavan. He's the principal scientific advisor to Government of India. And he said in this interview, in short term, we want to play in India's strength so that we can get our striving community from mathematics and computer science to address the problem in biology. And the similar opinion is put forward by our chief scientist in WHO, and who was a former Director General at ICMR. And she said the same thing, like we need to invest in population-based cohort and understand the biological specimen repository through partnership between government and private sector. And time has come where we need to harshen the data science. So she's pointing to the bioinformatics and the IT expertise to look the biological data. And that's the exact definition of the bioinformatics that I was saying. And similarly, a similar thought were put forward by Dr. Renu Sobrov, who is a senior advisor at DBT. She said we undertook an extensive landscaping study, and she has also launched Innovate in India program, which has recognized bioinformatics as the main skill that we need to build on. And then I would like to mention if the few of the institution or the few of the scientists who are working in different aspects of the bioinformatics in India itself. And his name is Dr. Akhilesh Pandey. He published a Nature cover page, Human Proteome from Institute of Bioinformatics in India. And similarly, a great agriculture genomicist, Dr. Rajiv K. Varsne, who is working at the ICRISAT and developing all the different crop resistance varieties using the bioinformatics tool. And he is Dr. GPS Raghav, with whom I did my PhD. He's working on computational aided drug design and has developed more than hundreds of the software and web server and databases. And he is Dr. Vinod Skaria, who is working on the medical genomics part at IGIB. And he is the pioneer in understanding this SARS-CoV-2, how this genome is varying and how we are coming up with this new strain and how we can address or design new drug against it. And these are the pioneer scientists that I mentioned briefly, but there are way more that I could not mention here. And now I would like to touch upon some of the practical issues being in in biometrics. I'm saying it's a big dark side, but obviously there are some dark side that I would like to aware you before you think your career option would be very, very bright in biometrics. So I don't want to talk about let's say Choti Ganga bol ke naale mein kuda diya or kind of that kind of meme, because whenever you say like biotech le lo bahut scope hai, obviously there is scope, but you need to build on certain skills. And I would like to say, what are the dark side of being in biometrics? So obviously there are some dark side, and this dark side includes there is a niche problem. Obviously this is interdisciplinary, so none of the discipline are actually accepting it as such a full-fledged science. And there is also a question about the status, where a scientist, when you are working in biometrics, should be treated as an assistant or you should be treated as a scientist. So there is also a niche gap still we are facing. There are some ethical issues about the credit sharing. They said like, oh, this was easy enough, this was quick enough, so you are not deserve to be author in our paper because you are biometrics and you just did using some software. So there is also some ethical issues. There are also some citations, because many of the time I have seen that in my life that people are using this biometrics tool, but they are not giving them citations. And there is also some issues about the grant application. So when we are in the research, we need to submit our proposal or the grants. And when we get the money, then we can pursue our research. But in biometrics, there is a big debate. When you submit a biometrics proposal, they said, oh, this is not large enough. Or when you submit a large enough proposal, they say, oh, this is not that you should lead, because that has a small part of the biometrics and the big part of is the data generation, which is coming from the wet lab. So you need to understand that there is still a gap. But it's not something that you should worry about. Once you work hard, you can easily overcome this dark side. All right, so let's come with the take-home message with my understanding of why you should care. You should care because in India and US, we are having a big opening for biometrics jobs. And you can see here in US and India from different websites, LinkedIn and all different sources, there are many more websites which are putting the biometrics job post. And more than thousands of people are being employed through this website, and India and US are at the top in this one. And they are also like offering excellent job opportunities and giving a good understanding of a higher paid job. And obviously there is a big data. If you are interested in science, there is a big data is coming up that need to be decoded, and we need more people that we are currently, there is a kind of demand and supply gap still we have. And with that understanding, I would like to move forward with the other take home message is why biometrics is hot, because there is a gap in supply and demand. And with the sequencing technology like microarray technology, a large amount of data is coming, which we cannot handle manually. So we need to have a big understanding of the computer science. And it leads to the important discovery that I briefly mentioned in my understanding. There is way more that I'm small enough to mention all those stuff. And it obviously saves time and money. And lastly, like my quick tips, when you want to opt for the biometrics, you must understand there is no alternate of programming. You must understand the programming stuff in biometrics, and you need to have the good writing skill, and which can come through clean thought process. So the easiest way to like start telling stories, so that you can tell a story and you can finish from start to end, and it give a complete picture to the listener. And networking is must. Obviously, you should improve your LinkedIn and get connected to the right people. And you must believe in grow together policy. Once you are growing, your circle should grow. And be open up for the challenges and put your best. Follow the happiness around, make new friends, learn new language, stay lively, and keep your sense of humor alive. Obviously, it's going to be a very stressful job, and this should not make you stress in your life. You should keep your humor all the time and make new friends, start new language, and staying lively is one of the tips that I would like to give. And be prepared to new places, because not everything you will get in your city or in town. You must be open to move to new places, and there are some fundamental that you must learn, like the cooking stuff or the socializing, or get some there are some sports activity that you can involve in, so that you can make new friends and you can socialize and make your new circle in new cities. And with that, how you can build all this biometric skill. So there are two different startups that I'm attached to. This is called Decode Life and the Nexen Helper. These are offering services to learn this skill, and they are open for collaboration, and they are also giving personal attention to your question, and so that you can translate your research effort into publication. And they are organizing workshops from time to time that if you're interested, you can check out these links at Decode Life and Nexen Helper. And with that, I'm happy to take questions now. And if your questions are not addressed here, feel free to reach me out or my email, or feel free to connect me on any networking site. If you Google my name, you will be find those networking sites. With that, I want to thank your attention and happy to take questions now.
So it's fantastic to start the day with a bioinformatics expert like Dr. Sandeep has started his presentation with immunology. And, then he explained very nicely about his contribution regarding a recent epidemic work like COVID, and then explained some personal stories like how the next generation should be ready to take up challenges and take it forward. So with that, we thank Dr. Sandeep for the nice presentation and very time, it must be very late in the US, right?
Yeah, it's 11:30 in the night now, but that's fine.
So the funny thing is bioinformaticians, 24 hours is same for them. So it's not like whether it is 10 a.m. or 10 p.m. doesn't make huge difference. So I think we have a couple of questions which is there in the chat box. The first one is by Dr. Manohar. He has a very nice question about what is the role of serotype as coronavirus alone is claimed to cause disease all over the world. So maybe to rephrase that question, like why so much of causality with this particular infection? And then how the serotype variations, if any, is known, if you can comment on that.
Yeah, surely. That's a very good question. And obviously, this is called novel coronavirus. So we are also understanding at the same time when it is spreading around. So we do not have enough science to go through all the details of this virus. But whatever we understand that there are new strain emerging. Like we have seen the recent case in UK, but the rate of this new strains is not that fast that vaccine won't work. And fortunately, these are the changes in other protein than the spike protein where vaccine is targeting. That's why we are going to have a good immune response for the vaccine even with the new strains. So that's about questions about the vaccine itself. And if you say like the new serotype, yes, the virus, every virus goes through certain mutation. The rate of the mutation varies from one virus to the other virus. And if you take an example of HIV, which mutates very fast. So there are new strain coming up every time. And if you say another example of many viruses which are not changing that fast, so we could have a very good immune response there. And if you talk about the serotype are actually defined by how they are recognized in our body because of like different antibody that can recognize them. So still, I'm not sure we have classified these as a serotype, but we have obviously classified them as the strains. The strains are a smaller hierarchy than the serotype.
Yeah, that's, I hope, Dr. Manohar, you got your answers. The next question from Sai is, I think it must be a student. And the question is, which specific programming language that are needed to be learned in bioinformatics, whether Python, C++, or C or Java? And I'll just add whether R language is a very common common thing, and whether each one, whether they do bioinformatics or not, should learn. Maybe you can comment on that.
Yeah, definitely. That's a very good question. And I would say once you are comfortable in one programming language, you can easily switch from one programming language to the another. And if you want to just start, you can start with Python, or you can start even with Perl. So the fundamentals of programming is to make your mind clear up how you should write a code, how you should start. And the only thing that will change from one programming language to the another is syntax, how you are writing that particular chunk of code in other language. So there will be little bit change, but there will not a fundamental change that you won't be able to understand. So I would say start with Python, and then you can easily move to any other programming language.
So just to add, do you suggest each one of us, whenever we are free, should start learning our language?
Yeah, definitely. You can start with any other language. Like I recommend Python myself because I choose it, but there is no good reason why don't you start with R. So once you have your time, and R is very good in stats. So I would recommend whenever you have free time or whenever you can wanted to have productive time, start with the programming language. It will not only help you in your research, but it also help you making decision in your life, because you will be clearing up your mind. You will be having a clean thought process to write a code, and that will help not only in the science, but also in your personal nature.
So there are lots of questions from your old friend, Dr. Rath, Jyoti Prakash Rath. So you raised one concern about, bioinformaticians generally sidelined by basic researchers.
Yeah.
And that happens, if that happens in the United States, I feel like it happens everywhere in the rest of the world. But there is another point you highlighted, like bioinformatics work. Always, like you, maybe the sample size is very high and you send it for, communicate it for publication. People raise like, where is the validation?
Correct.
Right. So Dr. Jyoti's question is, if bioinformatics works good, then why it is taking so much of time to get a COVID vaccine?
That's a very, very good question. And I must appreciate for this question. So there are two different points. If they are sidelined, they are trying to be sidelined to the bioinformatics people, the other researcher people in basic science, but they could not do it anymore because of the large data coming in. We are obviously coming as a front worker now. And coming to why it's taking, how far it is from the validation. Yes, there are also some kind of internal validation that you can do using computer science as well. You don't need to always go to the experimental wet lab to verify your tool. You can also have internal cross validation that you can run on your computer, and you can verify how good it is going to work in real life. You can be as close as wet lab. And in my experience, many of my tools which I developed has been experimentally validated by other people in their publication. They said, we use this tool and then we come up with this particular set of peptides or epitope. And when we test it, it worked. So yes, it is not that close that you can every peptide or maybe every outcome of the bioinformatics you can take to the validation and it will work, but it will give a sense of accuracy, like this percentage of the hits is going to work. And obviously, let's say you start with 100 molecule and you test only 10. So you are still saving 1/10 of your time, like 9/10 of your time and 9/10 of your money. And out of the 10, there is a accuracy that every software is supposed to give. Out of 10, when you are testing, it is going to work on 9 of them or 8 of them or 6 of them. There is kind of accuracy associated with every tool. And the last point, why it is taking that long for coronavirus vaccine, I would say, I want to correct you here. This does not take that long. The virus, we have vaccine in less than 1 year, which is remarkable. We did not have any other vaccine in less than 8 or 9 years before COVID. So we have progressed so much in case of coronavirus, and it did not take that long. Obviously, you need to test your vaccine in 70,000 or 80,000 people that we did in Moderna or Pfizer vaccine. They have tested in more than 70,000 people. So you need to have that number of people to enroll in the vaccine, and that takes time. And you need to have like 3 or 4 months of period. Once you have a vaccine, it was designed only in 2 days, the Pfizer vaccine. So you can see the speed.
So you are absolutely right in explaining that how informatics expertise can be used to filter out lots of hits. Like if you have 100 in, you have a trained bioinformatician on board as a collaborator, not as a competitor. And they help you to reduce it to top 5, top 6, and then you don't waste your postdoctoral time, research time, research resources to get your data on time. So there is one more. There are a few questions from Dr. Jyoti. It's about like, is this India like the strain which is circulating in India, the COVID-19 strain which is circulating in India? Is it very different from China or Europe or UAE or United States that is circulating there?
Yes, there are like 7 to 8 different strain in coronavirus, and we do see some differences. But fortunately, these differences are not in the antigen that we have picked up for the vaccine.
Right.
So if you're asking in context of your vaccine efficacy, then we are very positive that is going to work widely. But if your concern is about if there is strain variation, yes, there is.
So exactly, that was his second question, like whether the vaccine will work in other contexts, like in Indian context or not. So I think you have answered that question. Then there's a very nice question from VRDL Kasturba that maybe you have mentioned something about the mutation rate and error rates. So the question is, how this new variant that is reported in UK, how it has evolved? Can you comment on that?
Okay, yes, it has evolved and they see a different kind of mutation, and I'm not sure, like there could be very different angles to address this question. This could be addressed in terms of how these drugs are going to interact with this new strain, how this is vaccine going to interact, and it need to be experimental validation. But if you compare computationally one to them, you can find out these are the part of the epitope that we have in our vaccine, and these are the part which are conserved even in the new strain. And with that kind of hypothesis, we are assuming, obviously there need to be a experimental validation, but we are assuming is going to act on them as well.
So there's a similar question by Deepak Kumar Panda asking, sir, can you tell us how the COVID virus changes its strains?
And okay, when you call them a strain, these are accumulated different mutations, and once they have functionally different and different virus, they call it a different strain. So yes, these are accumulating the mutation over the time when you are being infected with the virus, and it propagates inside your body and thousands of virus comes out in your breath. So these could be different in one amino acid or two amino acid or maybe a number of them depending upon the mutation rate. So once there are enough mutation in the virus so that it can be functionally different from the original virus, we call it a strain. So yes, new strains are coming up and we are looking very seriously about them.
There's a very nice question from Dipankar Bhattacharya. The question is, when the rate of mutation of corona is high, is it advisable to design the vaccine candidates in just 2 days?
And that's a very good question. And I'd say, like, if you compare all the viruses, we did not still have a vaccine against HIV because the mutation rate is so high. But if you compare those kind of viruses with the coronavirus, the mutation rate is not that high. And most of these mutations, again, I'm stressing, are not on a spike protein, which is a main component of most of the vaccine. And yes, in fact, it could be a change in spike protein in some other strain, not in the existing strain, in the future strain. Even if we have some mutations right there, we are going to have different type of vaccine. Right now, there are around 3 or 4 are approved, but there are going to be many more in the clinical trial running in phase 3. Like the one from the Oxford is running in clinical trial phase 3, and also the Covaxin from India. So obviously, if these current vaccines are not going to work, we can assume the other vaccine, which are not based upon just one protein, but they're also based upon full virus itself, the attenuated virus or heat-killed virus itself. So those kind of vaccines are supposed to work because they are containing the complete virus and all that, changing all the, like we call it immune escape. This is not an easy way for viruses as well to develop the immune escape mechanism.
There's a very nice question from Nisit. I think I'll rephrase his question. It's about, like, is the strain present in an immunocompromised person is susceptible to get more or acquire more mutations? I hope Nisit asked the right question, what you wanted to ask. Yeah.
Yes, I understand the question context, because when our immune system is not reacting, it allows virus to propagate in large number of copies. So that could be one of the reason, because once you are creating large number of copies, the mutation rate, even though it might be low, you might get a new mutation, right? So
Right.
that could be a reason, but that cannot be just ruling out like this is going to be the reason. No, even in the healthy individual, not immunocompromised, the virus is still propagating, the virus is still dividing. So it is not dependent just upon the immune compromised individual. Yes, there could be more number of viruses in those individual because of the immune compromised nature, but it is not completely a one answer that I say this is because of the immune compromised individuals.
So hope Nisit, you got your answer. If you have any additional questions, still you are free to put your question. And in between, I think there are two questions left. One is by Aliva Naik. The question is how bioinformatics activities is benefiting our society. Maybe you can reiterate something you have showed where Anthony Fallaci was presenting your research work and how informatics work is directly helping the society, especially in this COVID-19 situation.
Not only like I won't take all the credit because there is a lot much contributed by other disciplines. So bioinformatics is giving just a starting point, and it is giving starting point based upon the existing knowledge that was generated by many different disciplines like the biotech or the botany or the zoology. They all generated the plethora of data. So bioinformatics is actually finding a pattern in those data and giving this data knowledge from the existing data to the new sequences or the new infection coming in. So it's kind of linking all the existing information to find something new treatment or novel treatments. The drug repurposing itself is one of the reason that bioinformatics played a leading role, because when you know the drugs are there and these are from the related viruses or pre-existing immunity, this is the case there as well. So you can easily link the missing link between these two disciplines using the computational sciences.
So a similar question was also recently raised by Rupali Kalpana. How is bioinformatics used in biotechnology? I think you have already highlighted with multiple examples. There is one question by Dr. Priyajit Sinha, that which bioinformatics books should be recommended for beginners. Maybe you can, if you still remember those books, you can just tell few of them.
In bioinformatics, when we say this is very interdisciplinary, so it's hard to just pick the one book. I would say pick your research question and start looking for the answers. So let's say your question has some plant research associated, right? So you are not supposed to read the bioinformatics application in medical sciences. So the application oriented research should be there. And once you know, like I'm going to move in this direction, read more about the biology and then say how much data is available and how I can find the data that I can link this to my research. That this is the role of the bioinformatics. So I won't be able to suggest just one book there.
Yeah, I think your point is like for doing research activities and all, it's better to select the biological questions first and then find out the tools you require to develop expertise and move on. But I think there are lots of private bioinformatics books are available, which Dr. Priyajit Sinha can recommend to his students. And there is one more question is, if RNA sequence, but it's by, again, Dr. Bhattacharya, if RNA sequence of a virus changes due to mutation, and we want to design a single vaccine, which part of the RNA sequence needs to be considered?
That's a very good question. And I said one of my application was Immunome Browser. Immunome Browser is an application where we can see along the length of the protein, which are the immunodominant region. And if you find the immunodominant region is not only immunodominant, but also the conjured in many strains. So that's going to be the best possible selection.
There are two questions, last two questions, I guess. One is, now lots of advertisement in YouTube comes where the cricketer or a film personality is telling I have sent my son who is going to school has to learn coding. Okay? So now it's a very common advertisement you see in YouTube channels. So Dr. Jyoti's question is that is it okay to send a 11 years old for going for coding classes or not?
I think first of all, that's very, very personal decision. I should not classify that you should do it or you should not do it. You should do it based upon the interest of your children. And if they are interested to go into something related to computer science, or not only computer science, but they are interested to go something where computer can play a bigger role. So programming is a good way to start.
So I think like one of the person who is watching your presentation from YouTube has also raised similar question that who can start biological coding? What prerequisite knowledge needed for them? So
interest.
Okay, yeah, I think it's a single word answer which explains a lot. And
Yeah.
I think we'll go ahead with the last question, which is quite interesting because lots of people started talking about neutralizing antibody, and then lots of biobanking started. So can you please comment on what could be the reason behind waning of neutralizing antibody in COVID-19?
I think when we say neutralizing antibody, these are the antibody which are neutralizing the virus replication. So one of the reason is and these are conformational in nature. Even though the virus is not changing that part where antibody is binding, if the virus is changing some other part, that part could significantly play the role in changing the overall structure of the protein. And with that reason, it could lead to the change in conformation. And therefore, those antibody might not be recognizing the virus anymore, and they are not able to neutralize them. So that's one of the reason that these antibodies are waning, but still there is a hope.
I think there is a very imaginary, like it's a very interesting question. Srimaya Pradhan has asked, how can we use bioinformatics in taxonomic research?
In taxonomy research? Yes, obviously, once you are trying to relate, like if you say taxonomy, you are having different species which are having the same protein, and how this protein is evolving in different species. The 16S RNA is one of the most common reason to do taxonomy in bacterial sciences, the prokaryotes, and you can find out just by sequencing the 16S RNA, which is a protein encoded by 16S. And if you sequence that particular stretch of the genome, you can find out how you can place your species into the original taxa.
Right. So just to add that, actually, if Srimaya Pradhan, you have gone through any metagenome related work or the microbiome related work, what Dr. Sandeep has given a very nice example, which is very commonly used for pulling out what are the taxonomic distributions are present in the fecal material or oral microbiome or respiratory microbiome of healthy and disease subjects. Okay, so with that, if the organizer allows me, I'll take this opportunity to thank Dr. Sandeep, who presented, who introduced the bioinformatics subject with few examples, and then he shared his career path. I'm sure students will get much benefit out of it, and his open-minded presentation has definitely will help also the students and the younger faculties who wants to venture out. And with that, I'll thank Dr. Sandeep and it goes back to the organizing committee. Thank you.
I want to thank all the organizers.
Sandeep, and as well as the chairperson, Dr. Nanda, for a very nice and vivid discussion. Of course, bioinformatics is the, and you have struck the nail at the right position. Thank you, Dr. Sandeep, for waking, not sleeping and waking up till 11:30 to make the things work out at your motherland, India. Thank you once again. So the organizer sincerely acknowledge your effort and your work and your presentation as well. Thank you, Dr. Sandeep.
Thank you, and thank
So-
you giving this opportunity to me. I want to thank all the organizers here. Thank you.
Thank you, sir. So moving on, our next speaker is Dr. Sonalisa Pandey. So she will speak on identification of gene expression, logical variants in Arabidopsis. So let me give a bio sketch of Dr. Pandey. She received her master's from School of Life Sciences, Sambalpur University, and MPhil from Gangadhar Meher College. That was the oldest college in the Western Belt, Sambalpur. Then she joined University of California, San Diego as a research intern in 2015. She applied Boolean analysis framework to Arabidopsis for understanding shoot apical meristem development. She then published many papers. Her research interest mostly focused towards understanding the fundamental mechanism of plant biology. Welcome you, ma'am. A hearty welcome from the organizer, Dr. Pandey. So the stage belongs to you. I do not want to be a hindrance between you and the audience. May I hand over the platform to Dr. Pandey as well as Dr. Nanda.
Hello.
Yes, ma'am, you are quite audible.
Yeah, thank you for the introduction. I am Sonalisa Pandey. It is great because I hear from Dr. Sandeep Dhanda, and my subject is also on bioinformatics, and we something is similar here. And okay, so I would like to start now. I worked at UCSD from 2015, and I write this paper, Identification of Gene Expression Logical Invariants in Arabidopsis, with Dr. Devasis Sahu. I work on Department of Pediatrics, and yeah, this is about me. Let me see. Okay. So my paper is based on Boolean analysis. This term is coined by George Boole, and it is basically the mathematics of two values, high and low, 1, 0, true, false, like that. And Boolean operations are commonly and, or, and not. But we are focusing in this paper on implication, that is X implies Y. This is the truth table that shows X implies Y and when it is true. So if X is false and Y is false, then X implies Y is true. When X is false and Y is true, then also X implies Y is true. When X and Y both are true, then X implies Y implies true is true too. But only when X is true and Y is false, this implication result is false. So here are some common examples, these four, and the most fascinating one is the comment by the counsel Robert Swan Muller. If the president didn't commit a crime, we would have said so. It's like if X, then Y must be true. So for this, we first did the threshold calculation. I wanted to tell you that I am a plant biologist, but I'm first focusing on the biostatistics part to explain my paper. So I used a step tool here to differentiate low and high, because generally gene expression are continuous. So the intermediate is the threshold, below that is low, above that is high. there are 6 Boolean implications. 2 of them are symmetric, equivalent and opposite, and 4 of them are asymmetric, which are more complicated. And generally maximum biostatistical problem mention the equivalent and opposite, but the good thing about the Boolean is they discuss about this asymmetric relationship. That makes it more unique. So equivalent one, these are some genes written ATY2 and PHOT2. These genes are equivalent. So when ATY2 is high, PHOT2 is also high. And also when ATY2 is low, PHOT2 is low too. The interesting one is the asymmetric one. For example, the case number 3, when AP3 is high, FRU is low. And but when AP3 is low, then I guess let me see. But when FRU is low, AP3 can be high or low both. So this is just the statistics part, which tell me, coming to the previous slide. Here we see these are some, in first picture, the quadrant, the lower right quadrant and upper left quadrant are sparsely distributed. That makes it computer to understand that this is a this is the equivalent equation, because 2 of the quadrants are sparse and 2 quadrants are more dense. And this is the statistic thing that computer knows and decide which Boolean implication it is. So another important term is logical invariance. Invariant means something that never change. So whatever rule is followed in every cases, that is logical invariance. PCNA and MCM2 genes are both the fundamental cell cycle genes, which are equivalent to each other in human, mouse, Drosophila, rat, Arabidopsis, and Botrytis. It's a marine organism, rat, like in every different kind of species, starting from human to fly, my Arabidopsis, and Botrytis, rat, everything. So this gene is invariant, which means it is the relationship between these 2 genes are common in almost all the species we know so far. So this is data normalization. For this paper of the Arabidopsis, we studied all the publicly available data, and the most popular one was GSE 6995, and they used the MAS 5.0 normalization method for this. And they take different kinds of like tissue type, growth conditions, developmental stage, and other criteria. And in 2018, we use Boolean analysis and RMA normalization method, that is robust multi-chip average, and we find a very good result. We also find some of the duplicate file that was thought, like they were thinking there are more numbers of samples, but we just remove some extra duplicate from the above one. So this is the proof of how this normalization method, RMA, is better. Oh, sorry, MAS. Yeah, Yeah, we use RMA. I was sorry. So if you see, these are the Boolean figure where every point in this plot is a probe set ID. And if you see, each point says about the relationship, the Boolean relationship. Like, for example, in the first one, the GSC 6995 and our one, you can see there are more dots in the upper side, like in our side. That says that more genes are, uh, we are showing more equivalent relationship in the gene than the the mass one. Okay, there we see one exception, that is A low, X high. But I will explain that later, what's the reason behind it. First one is, yeah, we see in this picture, there are too many of X, this portion is very high, whereas GSE 6995 has very few samples to show this relationship of high to low. And now we need to prove that if these things are, how much correct these are. So this is, we have their data, our data, and here is one RNA sequencing data. If you see, here is AP3 gene and FRU gene. So it is, FRU gene is mostly found expressed in root and AP3 is expressed in flower. In this picture, you can see that AP3 is found in flower and very less in root. But in this picture, that thing is not at all clear. Like here, even AP3 is high in roots. So this quadrant is also high. But in here, this quadrant is like there is nothing. And we really like see FIU high, AP3 low Boolean implication relationship is consistent between RNA normalized data and RNA sequencing data. That I was saying. Yeah, so that is. And for the situation that where we see that our low to high expression, we saw more data in this side, too many, yeah. So we again we have their data sets and our and the RNA sequencing, and we took the, we see the COL4, COL4 and APK1 gene and found that COL4 is low, implies ARK low. Boolean implication relationship is consistent between RNA normalized data and RNA-seq, but that is not here again. So ARK expression is low in the root, actually, but that is not shown here. That says like, we don't know. Summary is Boolean analysis discovers a lot of invariant relationship between genes, and RMA is a better um, normalization method than MAS5 if we take like with Boolean. And we also have one web server and software package, that is STPP Hegemon UCSD.edu plan. If you go here, you can find you can go yourself and find many publicly available data sets, and many things you can just go and see which data are equivalent, which are opposite. You can do all those comparison by yourself, and you can, and if you go to Hegemon, that UCSD page, in place of plant, if you put other things, you can see many important stuff to try on. So basically, Boolean implication network is a very effective tool to study plant biological processes and many other processes, but I am very attached to plant because I have been studied that for a long time. And I worked here also at Yanovsky Lab at UCSD on Arabidopsis. And so, yeah, that is it. What is the time right now? Um, okay, let me see. Oh, this is right. Mm. So these are my acknowledgements. Boolean Lab at UCSD. These are who I work with, our co-workers, and this is Martin Yanovsky Lab, where I worked on Arabidopsis, and I learned from some two universities about the Arabidopsis there too. Yeah. Any questions, please?
Yeah, thank you, Dr. Sonalisa Pandey, for this nice presentation. And it's a good shift, like from bioinformatics used in health research. Now you have was done by Dr. Sandeep, and now you gave nice example of Arabidopsis and your passion towards botany, and still you are continuing with that. So we'll wait a few of the questions from the chat box. I'm yet to, I think we got the first question by Dr. Subrat Thanapati. So what are the biological significance of the genes you have studied in plants? I think he might be talking about those two genes you have highlighted, no, where you have done those correlation analysis like MCM2 and PCNA.
Yeah, cell cycle. Yes.
So what is this biological significance of these two genes? Maybe that's what Dr. Thanapati is interested in.
So these are cell cycle genes like PCN and MCM2. Now, this is like, I just wanted to show these genes so that people will know what is an invariant means. These are fundamental genes.
Right.
And like it's it is found in every species. Like I want to say, as I showed, like human, mouse, Drosophila, rat, Arabidopsis, Bacillus.
I hope, Dr. Thanapati, you got your answer, but if you have still additional queries, please raise it. There's second question by Dr. Dipankar Bhattacharya. He is interested to know if the number of data is less than the relation may appear scattered. However, for more data, we may identify a relation. Thus, is there any minimum number of data acquired to reach to any conclusion? I think his question is more about the number of variables you use to classify the class, different class you undertake for study. So what should be the ratio between number of variables and the number of class you want to classify?
It should be more than 200.
Okay.
We look for what it is, just GSC 6995 is one, but while we did this experiment, we go through whatever publicly available data is present there. If I go to the, I can show more. So we go through many, yeah. So there are maximum like 6,000 data we took, right? And mm, okay, I would like to give
Okay,
it to someone.
let me join in answering these questions. This is the Bashis Sahu. Um,
Hi, Dr. Sao. Yeah. Please feel free to, join in the discussion. Yeah.
okay, so this is a really important question. The number of samples dictate, how whether you can identify a particular relationship. And usually, if you have less than 100 samples, it's really hard to come up with a really good relationship, like Boolean implication relationship. We have done simulations that suggest that you need at least 200 or more. Around 500 is optimal, and more than 500, it's like really good. If you are looking at human data, there is a lot of data out there for in general, but if you're looking into specific tissues, then the data would be limited. But if you collect everything, there is going to be a lot of data. But the thing is, it has to be normalized. The data, if you can't normalize them together and put it into one big data set, if you have data that are in different platforms, they are not compatible with each other. So you can't pull them together.
So
Yeah.
just to add that, is it these numbers are specific for RNA-seq experiments or?
No, it's in general, like any data set. It doesn't necessarily limited to gene expression data. Also, if you have RNA-seq or microarray or even proteomics data sets, right? Look at any data set, you have a table of values, and if you're looking at relationship between variables there, you need to know, how many different variables that you can play around with. For RNA-seq and microarrays, the numbers, they behave very similarly. RNA-seq is of high quality compared to microarray, so the noise would be somewhat less. But there, you're looking at a big picture, and these big picture relationships needs to be established with large data sets. And when you have thousands of data sets, thousands of samples, it doesn't really make a difference between RNA-seq and microarrays.
Great. So I think it was quite interesting to see the effect of data normalization, which very rarely people focus on. So on that, there is a question that what is the threshold? What is the algorithm used to normalize the data?
So for microarrays, the most popular algorithm are here. It's MAS 5.0 and RMA. Many people use MAS 5.0. Many also use RMA. There is all kinds of analysis that shows that MAS 5.0 is better than RMA, and some people show that RMA is better than MAS 5.0. There are other variants of these algorithms that came up later. But the thing is, there are thousands of other approaches that are out there. The normalization is a really vast area of research, and it's really important because if you don't normalize well, you can't compare samples to sample. In this case, we use the most popular one and show that there's a big difference between these two. So people need to be careful about what they want to use if they're interpreting these data sets in different ways. As we have given some example here, you can see that really a dramatically different relationship appears when you normalize the data set in different ways. For example, in this one, the COL4 and ARK1, you're looking at on the left side, and it shows that COL4 low implies ARK1 high. but exactly the same data set. This is the same data set. If you normalize with RMA, it shows call for low implies ARK1 low relationship. These are two different relationships, completely different relationships. In one case, you have a different interpretation whether ARK1 is high or low in a particular sample compared to other data sets. So these will create a lot of confusion. And this is what is going on in public domain, that when people normalize in one way or other, they get drastically different results, and the results are not consistent with each other, which is a big problem.
Right. Right. I hope, Jyoti, you got your answer. Jyoti has a second question about what is the threshold used and how far Boolean works good. So I think maybe the question is on threshold, how you select a threshold.
Yeah. So if you want to do Boolean analysis, you have to convert all the values into this Boolean domain, which is two different values. Gene expression data is continuous. So you have to somehow convert these into two different values. And the way we do that is by fitting a step function. So you first sort the values from low to high, and you model there must be some threshold that separate these two high and low values. You fit a step function and compute the threshold. You have a noise margin around this threshold, and that defines your low and high values. This is the threshold that you use to define the high and low values for every gene independently of each other. And when you look at a pair of genes, you have 4 quadrants, and you apply the statistic to identify whether a relationship is significant or not.
Yeah, I think question came from Pratibha Lenka, requesting you to explain simply what is Boolean implication. I think you have done that in last 2 minutes. But if you want to add anything, feel free to.
Yeah. This is the part where everybody confused. They get confused. Even though it sounds really simple, but it's not really intuitive for many. What is not intuitive is, so this is the truth table for Boolean implication. X implies Y. X implies Y is true when X is actually false. So this is the part that is confusing for many. But the other thing, this one, whether if X is true, then Y has to be true in order for this statement to be true. So the way to think about Boolean implication is, how do you, if somebody tells you that there is a Boolean implication, there's only one way to falsify that statement. And the way to falsify the statement is to set X to true and Y to false. So if somebody says there is an English sentence that says, if X, then Y, if you want to make that statement false, then you have to set X to true and Y to false. In every other situation, the statement is true. Because if you say if X then Y, then you don't really care when X is false. So if X is for all possible values are possible, every value is possible. And here there are some examples, and this is a powerful use of Boolean implication. As you saw, maybe some of you have seen how Robert Mueller, as a special counsel evaluating President Trump, he didn't want to say that the president committed crime. But there is a really nice way to say that in using Boolean implication, which is, it says, if the president didn't commit a crime, then we would have said so. Which doesn't mean that the president actually commit a crime, but it's possible. The possibility arises, which is really powerful way to put it together. So this is what Boolean implication is all about. And if you look at the relationship we look at in the gene expression data, we can summarize the entire expression pattern into this really simple Boolean formulas. Very simple Boolean formula. And many people ignore this and they focus on symmetric approaches. The asymmetric relationship are the one that everybody miss, which actually has a lot of information that is really important for us to know and come up with specific hypotheses about complex diseases.
Okay, so on this plot, I think there are two questions by Dr. Dipankar Bhattacharya. One is how to differentiate signal from noise, or how you detect noise and remove them from the list. And the second question is, if you change this X and Y axis, if you increase, you move the axis towards right and left, extreme right and left, the conclusions will change, or it may change the way we visualize it. So what parameter we select to select the axis, the range of the axis?
The range of the axes are determined by the values of the gene expression. So this is microarray data. And the microarray data, when you normalize them, it will give you a range of values. So for AP3, the range of values you get is from, let's say, 4 to 12. And that's the range you work with. If this is the values you get, if you look at all kinds of samples, if you look at only specific tissues, for example, then that range will be really low because AP3, the range would be low for a specific tissue. So to get the entire range, you need to have diversity. If you don't have the diversity, you can't see the all possible values that can arise for a particular gene.
Right.
So that Boolean implication is obviously sensitive to these parameters. That's why we put everything we can to establish that there is a really fundamental relationship. You could also take this and then apply it on a specific tissue, but you need to have enough samples there to come up with the significance, statistical significance.
So we have two last questions. One is by Dr. Satyabrat Nanda. The question is, transcript variants can result in contrasting effect of a particular gene. For instance, in insect effector genes. So can we use Boolean analysis to identify the transcript variants in insects and their potential downstream effects?
Yeah, I mean, so if you look at, I think one of the last one here, we have fruit flies. Fruit flies is insect, right? So in this case, you're seeing the genes PCNA and MCM2 are equivalent in the insect. And these are data set from different kind of experiments, knockouts or cell lines or any kind of torture that people have done to insects. They give this kind of diversity. If you only look at a specific condition, it may not give you that much of diversity. And diversity is really important to understand the relationship. So if you have diversity in a specific insect that you're trying to study, that can provide this kind of relationship, the Boolean relationship at a big picture level. And if you're diving down deeper into some mechanism, there you have to come up with new analysis tools to get much deeper level information that can give us hypotheses.
You can add on, Dr. Sonali, sir. Yeah. Okay. So the last question is from Rajeshri Panigrai. The question is, can we use this Boolean application for any field of biology? Maybe you can explain with one or two examples. I think you have already done that, but it's okay to repeat one more time.
Yeah, I think it can be applied to any field of biology. Doesn't it's not restricted to plant, for example, or human or mouse specifically. In fact, you could expand it beyond biology. You could apply it to finance. You could apply it to other kind of data. Any kind of big data analysis can be viewed in this term. The beauty of Boolean analysis is it simplifies the data to the very core level, the most simplistic version, which is convert everything into two values. Most people are scared when they do that because they might think that they will lose information. But what I've seen, and I'm continue to advocate this, is that you actually gain power in doing that. You get information that are beyond reasonable doubt, beyond certain kind of statistical significance that people really fight about. And these are really important for coming up with hypotheses that and things that go from bench side to clinical side. It's really important to see how the information that we receive in the bench side is applicable in the clinical side, because you can't test it in the clinical side beforehand, right? It's really expensive to test any clinical data or any clinical sample. These kind of tools help establish this invariant relationship, which are helpful, in establishing the invariance, as well as how it's likely to be translatable in the clinical domain. And we have examples from cancer. We have examples from different kind of diseases, human diseases. You could apply this to specific biological processes like cell cycle, or macrophages, or cell type behavior, development, differentiation. In fact, all aspects of biology can be explained through many of these Boolean implication relationships. So it's broad, and it can be applicable in general to any fields, I would say.
So that's great. Like, it's fantastic to listen Dr. Sonalisa and the explanation of both Dr. Sonalisa and Dr. Debashish. It's fantastic. I think the students, the participants, faculties all raised wonderful questions, and we discussed a lot. We learned a lot. And with that, I thank both of you for taking out your time, in the mid of night or whatever may be the time now.
Yeah, no, thank you. Thank you very much for inviting us. I think this is also,
yeah, we enjoyed it.
Right. So it goes back to the organizer, and in between, I may need 2 minutes break. Yeah. Thank you.
Okay. Thank you very much.
Thank you, Chairperson. Really, we are privileged to host you people, still people from US. Thank you, Dr. Sonalisa Pandey, as well as Dr. Sahu, for joining us to make the event success, and also waking up late night to make up the things. Thank you. And of course, we enjoyed a lot, particularly if I say from my context, I'm a bioinformatician. So to me, I learned a lot from you people how to use the things in plant biology. So it works seamlessly with the plant sciences. So botany and bioinformatics, that's what the agenda, it fits to the agenda. Thank you, Dr. Sonalisa Pandey and Dr. Sahu for for a nice presentation. And of course, also our chairperson, I would like to thank Dr. Ranjan Nanda for hosting the session and taking up all pen to take up each and individual question and put up to the speaker. Thank you, Chairperson Dr. Nanda. So, thank you, Dr. Pandey and Sahu, Dr. Sahu,
Thank you very
and
much.
thank Dr. Sanjay Dhanda for a nice session. So now it is time to go walk into the second session where we have two eminent speakers joining from ICGB New Delhi. So Dr. Ranjan Nanda from a group leader, Translational Research Group, International Center for Genetic Engineering and Biotechnology, is the next speaker. Then we have Dr. Niharika Bhattacharya from the Premier Institute of Indian Institute of Technology, Kharagpur. She will be speaking on the IPR. So, and the session will be chaired by Professor Ashwini Kumar Rath. It is our privilege to have Professor Ashwini Kumar Rath, a household name in cartoon throughout the world. And Professor Rath has over 30 years of teaching experience, and starting from he was a gold medalist in both BSc of Samalpur University, but to serve the Bolangir community, he stick to here, and he taught many various batches. So, and again, the flip side, the other part is he is the holder of Limca book record for the cartoonist brother Ashwini and Abani have many cartoons. They are the acknowledged people in the cartoon world as well. So welcome you, sir, Professor Ashwini Kumar Rath, to chair the session. So
I think it's not audible.
yes, some technical glitches. So yes, just a minute.
I extend my full thanks to Dr. Jyoti for the first introduction. Secondly, I welcome considering invited speakers, Dr. Nanda, as well as Dr. Bhattacharya. Let the session begin with Dr. Nanda. I actually welcome Dr. Nanda. His presence is talk on urine metabolism and serum extracellular vesicle proteome in tuberculosis patient. Welcome, Dr. Nanda. Please.
So before going to the speaker, let me introduce the speaker once more. There is no more, I don't believe that there is any other requirement of introducing the speaker, but for the today's journey, it is formal to introduce the speaker. Dr. Nanda is the group leader of Translational Biology Group, International Center for Genetic Engineering and Biotechnology. Before joining ICGB, He had his master's from Sambalpur University, School of Life Sciences, Sambalpur University, 2001. Then he moved on to Indian Institute of Technology, Kharagpur, did his PhD. Then he scaled up to International Center for Genetic Engineering and Biotechnology, joined as research scientist, staff research scientist, worked in tuberculosis, and later on, he climbed the ladder and became the group leader of translational biology group. He is having a rare distinction of getting Bill and Melinda Gates Foundation Award, travel grants as well. So I welcome you to the session, Dr. Nanda. And now I won't be any more standing between you and the audience. He'll speak on urine metabolism and all. The stage belongs to you, Dr. Nanda.
Good afternoon, Dr. Rath, and good afternoon to all the dignitaries of Centurion University of Technology and Management, Odisha. And it's a unique privilege to be part of this interesting meeting. And today we'll discuss about some of my lab activities, what we are doing at ICGB. I hope is audible and the screen is clear to everyone. Is the screen is fine and
Yeah, it's visible. Yeah.
Yes, sir. Yes, sir. It is quite audible and visible as well.
Okay, so it's great, like that, you know, I did my MSc from Sambalpur University, visited Kharagpur, then did my PhD, came here. It's very, I just wanted to say that before coming to ICGB, I never worked on tuberculosis. So my PhD was on a different subject. I shifted my gear. And when I visited ICGB for a position, then the then director mentioned that we have to start a tuberculosis program. So the message what I want to say is that irrespective of your background, sometimes you can take some faith of leap, you can change your subject, and you can try to do something completely new. So I hope tuberculosis as a disease might be very, like we might have come across it lots of time. But again, it may be, and the problem is still there. Although we all know that tuberculosis related drugs are available in the market, it's government is taking lots of care to initiate the diagnosis, early diagnosis, and that's what, like, we are partly successful in last, especially in last few years, because of some quick diagnostic solutions that has come up very recently in last 10 years. Now the number of cases detected are high, and the number of cured rates are also high. So what we expect in a TB patient, let's start from the suspected patient. A person who comes with some cough, the symptoms are mentioned on the right. You can see that the person comes with cough of 2 weeks or more, and then weight loss and fever, and then night sweats. These are all the common symptoms. And there are certain risk factors like HIV infection, malnutrition, post-genetics. There are few post-genetic genetic composition makes us susceptible to infectious diseases. Then diabetes, as in the first half, Dr. Sandeep talked about COVID cases, it's more in immune compromised conditions. So like that, diabetes also provides a very fertile ground for TB bacteria to convert from latent TB to active TB. So a compromised immunity helps lots of things. So including the conversion of latent to active TB. So a TB patient throws aerosol during cough and all, and those bacteria which comes in the cough goes to a healthy subject who is around the subject, and then gets those bacteria into through the respiration. It goes to the lungs, it gets lodged in the lungs, and whenever the person is a little bit weak in the sense like immunologically less efficient at that time, the bacteria starts multiplying. And then if you'll see, like in a latent state, you see the top one, it's like it forms a granuloma. And then in the middle one, like when it, at that time, actually it's the subject is called latent TB. And latent TB subjects doesn't come up with cough and weightless fever, night sweats, all these symptoms. So the bacteria is present in the system, but we don't develop the disease. So we don't go to the doctor and get ourselves tested and do the necessary therapeutic interventions. But whenever the immune system is weak, we develop active TB. And active TB in the sense, this granuloma, they breaks open, and those bacteria which were inside, which were cornered by the host immune system, they come to the respiratory system, and through the respiratory system, it comes to the aerosol, and as we discussed, it affects healthy subjects. And is TB only affects lungs? It's that's what we always think, but it's not true. It affects central nervous system. It can affect eye. It can it's present more in lymph nodes. It can affect pericardium, adrenal, GI, gastrointestinal tract. Whenever we take milk from TB infected, Mycobacterium bovis infected cow, we don't sterilize it well, we can get GI, gastrointestinal TB. And it can also affect our bone marrow. It can affect genitourinary system. Sometimes the women, they don't get pregnant. It's because the granuloma stops the migration of the sperm to go and fuse with the ovum. So just anti-TB treatment helps to reduce those granuloma size and makes the subject to be fertile. So what we expect in a disease condition? We do expect lots of variation at different molecular compartments, like in genes, it might get altered. Dr. Sonalisa mentioned very nicely about the RNA-seq data, that is mRNA expression profile might get altered. MicroRNA profile can get altered. And then other molecules like proteins, metabolites, volatile organic compounds, lipids can get altered in a disease subject. And what we expect is that because of this pathological effects, we do expect lots of alteration. And when we take effective therapeutic interventions, we do expect that the subject will go back to its healthier stage. So to diagnose a disease condition, we can select few of the genes, or few of the mRNA, few of the protein, or few of the metabolites, or lipids, or VOCs as molecular signature to diagnose disease condition. And I don't have to talk about omics, like the inauguration session, Professor Bhattacharya explained about, it's more about a omics related presentation. The bioinformatics, the initial two presentations were on informatics. Today, I'll discuss a little bit about proteomics study and one example of metabolomics study, and how it helps us to identify molecular signatures that is specific to TB, and how we can better understand the disease condition and then take it forward for developing some translatable solution. And that is what is my team's long-term interest. And we're quite lucky to have wonderful clinicians, clinician collaborators from different hospitals they are on board with us. What we do is collaborate with them. They help us in recruiting the TB patients, and we get their biological fluids and do their diagnosis, as well as we take them for understanding the pathophysiology. And then there's few questions like whether the COVID strain which was present in Europe is same in what we see in India. So like that, the MTB strains, now lots of MTB strains are developing MDR, like drug resistance. They are gaining drug resistance. So we go to different parts of the country to recruit TB patient so that we can take care of the host genetics as well as the pathogen genetic diversity and their resultant outcome. So we have a good presence in Northeast. We have established lab there, and then the rest of the country. So today's discussion, I have selected two topics. One is on urine metabolite analysis, like how the TB patient's urine metabolome is different in a TB patient and how it is different in other disease conditions and healthy subjects. And that was done by Mrinal. Dr. Mrinal finished his PhD in Delhi University Botany Department. He came to us. He got trained, worked with us for 4 or 5 years. Now is a very good immunologist working in UK. And the second story, like the serum extracellular vesicle proteome, how it is different, the proteome composition is different in TB patients, that we'll discuss. It's a very recent finding. 2020, we published a paper on it. And Rakesh has completed that story. He is now searching for postdoc, and he's going to be a scientist in his lab. So quickly, just to highlight, as I have explained just a few minutes before, it's a very busy slide, but I just wanted to convey what we are doing can be done at any place, including Centurion University. So in this study, what we have done is we have taken TB patients from CMC Vellore. CMC Vellore doesn't need any introduction to this gathering. And then we have also gone to the eastern part of India. It's like near Calcutta. There is we have recruited TB patients from there. So what we did is, like as I explained in the first slide, a subject who has high body temperature, the first point is like 2 weeks or more of cough. And then if it has experienced body weight loss, and then night sweat, and if it is producing sputum is the commonest diagnostic matrix used for TB testing. So we recruit those symptomatic TB suspects from these two geographically distinct places. And as I explained, like in last 10 years, we got one scientist have developed a very unique system. It's called nucleic acid amplification test, is otherwise called Gene Expert. That instrument is there inside the arrow. You can see that. It gives results in just less than 90 minutes. So it just checks the presence or absence of a particular gene present in the Mycobacterium tuberculosis. So what we expect is like we take the TB patient sputum sample, we process it, and after the test results, if it says that it has MTB DNA in it, then we group them as active TB cases, and that is in short form we have written as ATB. And if the subject is symptomatic, but in his sputum sample, if MTB genetic material or the intact MTB through a microscopy test, if it is absent, then we group it as non-tuberculosis subject. And what is this non-tuberculosis subject then? These subjects could be pneumonia patients, could be asthmatic patients, could be chronic obstructive pulmonary disease patient, could be lung cancer patients. So they share similar symptoms with TB, but they are not TB cases. So as mentioned, it's NTB as control. So it's a case control study. So we have active TB as case, non-TB as control. And what we have done, we have collected their urine sample. These are all random urine sample. So we take urine sample as we all know, that in urine you have highest amount of water, and then you have a lot amount of urea is there, and urea will affect metabolite analysis. So what we do is we use urease to degrade urea to carbon dioxide and reduce the concentration of urea to close to 0. And then we added 5 microliter of lysine D4. So how lysine and lysine D4 are different? It's mentioned, the fragmentation pattern is mentioned on the right-hand side corner. So lysine D4 will have 4 mass extra. So that we use as an internal control, as an internal standard. And then we extract metabolites present in the urine using methanol. And that metabolites can be run directly in a liquid chromatography mass spectrometry system. But here we are using a gas chromatography mass spectrometry system. So these molecules needs to be derivatized. So derivatization is a simple technique by which we add a side group to the micromolecule so that it can be now made volatile. We can volatilize it so that a mass spec can identify this molecule. And it's a very commonly used method, and it's very detailedly mentioned here. You want to repeat this experiment, you can do it. So the activity, you see this green total ion chromatogram is the representative total ion chromatogram of a non-tuberculosis case or control group. And the other one is the red one is the total ion chromatogram, which results out of a GCMS run of a urine sample. The red one is of an active TB patient. So we talked earlier, Dr. Sonalisa mentioned that we have to use normalization, right? So for urine metabolite analysis, people use creatinine as one of the normalization factor. But in TB patient, because of muscle mass loss, creatine itself gets deregulated. Deregulated in the sense like it is higher in TB patient. So we cannot use creatinine as a normalization factor. So what we use, and then another point, if you just to remind you that we have taken random urine samples. In general, when doctor says that you have to give your urine sample for any testing, they always tell you that please give early morning urine sample. Why? It's because it's the most concentrated form of urine. Whole night we don't drink additional, majority of us don't drink additional water. So the urine what we get in the early morning is the most concentrated one, and it's rich in molecular information. But let's say if you want to do a diagnostic solution or to represent, we cannot expect that every TB patient has to come early morning. So in this project, intentionally we have taken random urine sample. So if we are selecting random urine sample, then these samples will be very diluted. The dilution level will vary from person to person. So in urine metabolite study, to take care of the dilution factor, here we do total area normalization. I'm just repeating Dr. Sonalisa and Dr. Debasis mentioned very clearly about normalization is important. So here we use one molecule like lysine D4 as the internal standard to monitor the instrument associated variations, and total area normalization to normalize between samples. Then question comes like, let's say one of the peak is highlighted as glucose. Why we are claiming it as glucose? It's because if we run a commercial standard of glucose, the retention time will be same as we have seen here. And we have already discussed that let's take example of lactic acid. So here when we are talking about lactic acid, it's actually TMS derivatized lactic acid. So it will have additional trimethylsilyl groups that is mentioned on the top panel. So if you have questions, please feel free to raise so that we'll discuss about it so that at least these two things should go, like why we need internal standard, why we need normalization for urine metabolite analysis. And in this study, we have done both case control, in that we have taken 21 active TB patients and 21 NTB subjects. And their mean age is very similar. There's no much statistical significant variation. But only thing is, one of the limitation of this study is that we have included only male. Why male? It's because urine metabolites will vary based on the reproductive cycle in female. So to reduce the variability, we have just selected male, but in our subsequent studies, we are including both the genders for our analysis. And like in the first presentation, Dr. Sandeep told, we need validation. Informatics work needs other wet lab validation. So in the second part, what we have done is, we have followed up TB patients till they complete their therapeutic intervention. TB patients undertake 6 months of tuberculosis treatment if it is a drug susceptible case. And if the patient is suffering from a MDR TB, the patient belongs to MDR TB, like it has drug resistance TB, then the therapeutic intervention can continue up to 18 months to 2 years. 12 to 18 months. So how we achieve to identify important molecules? Before that, in the lower part, lower left, you see a principal component analysis. In that, we have mentioned those black dots are the TB, is the urine metabolite profile of a TB patient. The blue triangle shows the urine profile of a non-TB cases. And this red one on the right are actually QC samples. And what is QC? It's a quality control sample. So what we do is that we pull all the samples of active TB, non-TB, means all study samples has to be pulled together to be prepared as a QC, quality control sample. And as this, like we have 42 samples to run, so we cannot run 42 samples in a single day. So it can continue for 5 or 10 days. So here we have taken 10 days to complete this analysis, at least data acquisition. So we started day by running a quality control sample. And then we run our test samples. If the QC is not coming proper, that means we need to condition the instrument first and then run our biological sample to identify important molecules. So the common steps, what we do, is mentioned in the left-hand side flow chart. The raw data files, what we get from GCMS, we align them, we prepare a data matrix, and then we use MetaboAnalyst 2.0. It was available in 2015. Now it is MetaboAnalyst 4.0. Here you can upload all your data. It's a free online software for doing lots of work, including the statistical analysis. And it doesn't need any licensing. Each one of us, if we have interest, we can use this tool. So if we have a data matrix, like we identify, let's say 400 odd molecules in every sample, urine sample, then how to identify important molecules? So we follow two methods. One is univariate method. In that we check fold change value. So either it has to be present in higher or lower abundance in the case, like here in TB patients, with respect to the non-tuberculosis controls. And then we do multivariate analysis, like principal component analysis, to see what is the grouping pattern. And on the middle, you see those green dots are actually the non-tuberculosis patient urine samples, and the red dots present the urine profile of active TB patients. And you see they are very unique, except they are clustering away from each other. That means the urine profile of a TB patient and a non-TB cases at the time of presentation when they are yet to start any treatment, they group separately. So let's say we have 442 molecular features we have identified, that is resulting into this group separation. But all 442 may not be important. Like the earlier presentation, in the presentation, they have mentioned that there are some important molecules which are present on both the side of the corners. So like, based on two parameters, the fold change value, if it is more than 1.5, and then we have done a additional statistical analysis, is called partial least square discriminant analysis. It's a statistical algorithm to identify important molecules. So from that, we selected 42 important features, which are mentioned on the right. And it shows that all these active TB and non-TB subjects can be grouped separately. And from this, the predictability is close to 95%. And the area under curve is close to 0.85. So the up to 85% sensitivity and specificity, it can classify TB and non-TB. But these molecular features, then we wanted to identify what are those molecular features. So what we identified is, out of 42, we identified 21, but we just selected 5 because they were presenting, they were part of phenylalanine and tyrosine degradation pathway. So if you'll see, 4-hydroxybenzoic acid is higher in ATB patient. 4-hydroxypuric acid is also higher. Norepinephrine is higher. Gentisic acid is also present in very high amount in ATB subjects. And all these are representing or part of the phenylalanine and tyrosine metabolic pathway. And if we go back to textbook, phenylalanine is an essential amino acid, tyrosine is also essential amino acids. And those blue colored arrows, these enzymatic steps are governed by few genes which are yet to be reported in human. So highly likely these genes or these enzymes are contributed by the gut microbiota. And if we'll see, the correlation value of 4-hydroxybenzoic acid and 4-hydroxypuric acid is close to 0.74. So they are very much linked. And from this story, and then if we'll see norepinephrine is, it's also an immunomarker. It's not only as a, it also works on immunological changes or immunological molecular profiling, and it has work in immunology other than neural biology and all. And if you take a beta dopamine hydroxylase mutant knockout mice, it is more susceptible to TB. So it seems like the tyrosine phenyl metabolism is getting altered in TB patients. But what happens to this urine metabolites upon treatment? So here we have shown that if we have taken 11 active TB patients, we have followed them at least once in every 2 months till they complete their therapeutic interventions, up to 6 months. And then you'll see the urine metabolite profile is getting altered with treatment. And these pink dots are actually representing healthy subject urine profile. So the 6 months clinically cured TB patients' urine profile, what we assume that it should match 100% with the healthy subject, but it doesn't happen like that. So till 6 months, the subject has taken several grams of antibiotics. So those grams of antibiotics might have altered the gut microbiota, and it might have influenced lots of host metabolic activity, resulting to a change like better than the active TB patient, but it's not sufficiently has reached as far as its biochemical profile is yet to be identical to a healthy subject. And maybe if you could have taken a 12 months, like after 6 months of completing therapy, if you would have taken 3 months or 4 months extra to collect urine sample and profile, maybe that could match to a healthy subject's urine profile. And as we discussed, geographical sites represents host genetics as well as pathogen and genetic diversity. Age also impacts, age, body mass index, And smoking habit also impacts the urine metabolite composition, but the important molecules what we have identified doesn't change with respect to age or BMI or geographical sites from which we have recruited TB patient or smoking habits. So quickly, from our study, we have shown that tyrosine phenylalanine metabolic axis is altered in TB patient, and there might be a possible role of altered gut microbiota on which we are working on it. And we have data from mice and human subjects suffering from TB has shown altered gut microbiota diversity. So we are working on it. Then quickly we'll go to the second part of the talk. So extravesicular vesicles, if anyone is interested, there is a very nice paper, discussed very well it came in Science. It talks about what is the exosome. And we all know that exosome has an endogenous origin. It comes out of the affected cell, or it's not always the affected cell. Healthy cell also contributes exosomes. And these exosomes have several functions, including the most common one is to transmit molecular information from the affected site to the other parts or the organs. And they are rich in nucleic acid, they are rich in protein, they are rich in metabolites. So they are rich in many things. So what we thought is like, let's profile the proteome component of it. And this paper came in proteomics clinical application, and it came in the cover page. All work is, major work is done by Rakesh. So what we did is, as I mentioned earlier, we didn't take healthy and active TB patients for comparison. We took healthy as one of the control, but our primary control group is the non-tuberculosis cases, which are suffering from other TB, other disease conditions, having similar symptom to TB. And then after isolating their exosomes, there are multiple methods of exosome isolation. We have used ExoQuick TC method, and for validation, we have used also ultracentrifugation sucrose density gradient method. So after isolation, the first thing we have to do is characterization. So there are lots of there are lots of surface marker molecules like CD9, which can be monitored on the isolated exosomes. And then we have used a most commonly used proteomics tool, quantitative proteomics tool. It's called iTrack. We have two sets of group. The healthy and ATB we have compared in iTrack 1. And in the second one, active TB and non-TB was compared. So the first question is, like, we know that TB patients or non-tuberculosis cases will have higher degree of inflammation. It's just like COVID patients, they will have higher degree of inflammation, like with respect to the healthy subjects. So we find there is no, so before we do go for a comparisons study, we should ideally take age and gender match subjects. So here we have taken, you see, all the subjects were taken, we have taken male subjects for serum exosome preparation, and their age is very similar between case and control groups. Their BMI, as we have mentioned, that active TB patients have low, they lose body weight, so they have very low BMI, significantly low BMI than healthy subjects. And NTB is an in-between group from, it's between active TB and healthy, it's in between. Its BMI is low because it has some infection or other disease condition, but it is definitely not healthy. So, so we have characterized these the exosomes isolated from the urine samples, and through electron microscope and dynamic light scattering, it shows like the range is 80 to 200 nanometers, and in some cases it's higher. Irrespective of whether we have isolated using ExoQuick or ultracentrifugation based method, we have isolated similar exosomes. And in the inflammation conditions like TB patients and healthy subjects, we do expect inflammatory response protein will be very high in the serum. But if we compare the total protein in the serum between active TB, non-TB and healthy, we don't see any significant difference. But in the exosomes, isolated from ATV patients has higher protein amount. That's presented in the last right figure. And this is all, similar observation is also reported in other infectious disease conditions and cancer conditions and all. So whatever these microvesicles we have isolated, we need to characterize that it's really exosome or not. So we monitored lots of marker molecules like PDCD6IP, NAPSA, spondin, aquaporin, CD63, CD9, irrespective of whether we have isolated from active TB patient or non-TB subjects or healthy subjects. All exosomes has these marker molecules. So it seems like we have identified these molecules. The exosome preparation is good. And then we wanted to know, like, if exosome has lots of molecular signature, like has molecular information, can we use as vaccine, or before we use it for or exploit its biological function, we need to also know whether they are biologically active when we store it in -80 or -40. So what we did is, like, we isolated this micro exosomes from different case and control groups, stored it in -80, and then we took A549 cells, which are type 2 epithelial cells present in the human lungs, and then these labeled exosomes were introduced or incubated with this A549 cells. And then we incubated from 1 hour to 5 hours, and we see these exosomes are still viable. Still, their molecular information goes inside this A549 cells and gets delivered. And this below is the video. I'll not spend much time on that, but you'll see this expanded images shows that earlier isolated exosomes still can enter to a cell line. So that means it can enter, deliver their cargo, and execute their biological function. So from the proteomics experiment, I'm sorry, it's really a busy slide, but I wanted to say that in the first ITRAC experiment, where we have compared the exosome proteome of active TB and healthy subjects, we have identified 132 proteins. And in the second ITRAC experiment, we have identified, in that we have taken active TB and non-TB cases, there we have identified 68 proteins. And those red one are the upregulated protein, or their abundance is higher in the exosome of that particular study group. And the blue one are reported to be underexpressed, or their abundance is little low. And if the fold change value is more than 1.5, we have selected them as important molecules. So to classify two groups, it's not necessarily that we have to select all the deregulated molecules. What we can do is we can just select the minimum possible number of molecules to classify the important study group. So here we have taken haptoglobulin, HP, KYT3, SERPINA1, and ApoC3. These four are sufficient enough to group healthy from active TB, and we have taken it for validation. And these exosome proteins, if you'll see their biological processes, the proteins what we have identified, a subgroup of it which are present in exocrata. They contribute to different biological processes like endocytosis, vesicle-mediated transport, innate immune response, which is expected. So out of those four proteins, the reviewer asked that, and then we went with an independent sample set, like Dr. Sandeep mentioned, that if you have an informatics conclusion, like if you have narrowed down from 14 to 4 molecules, then you need to validate it using wet lab experiment, using an independent patient population. So exactly that's what we did here. We took an independent patient study population. We monitored the expression of these 4 molecules. And then we have shown the fold change values, what we have identified using mass spec is matching with the Western blot ratios. But the reviewer wanted that we should also use a commonly used method of exosome isolation, which doesn't need antibody and others, that is ultracentrifugation, sucrose density gradient method, to use it, isolate exosome, and then monitor whatever the results we have identified in the initial 2 sets, whether it is matching or not. So here, if you'll see, we have just probed 3 molecules. We missed ApoC3. It's because we need to load lots of protein, so we avoided that. And the reviewer was happy to accept this paper. So after, let's say these 3 molecules shows altered expression in active TB patients, but is these molecules can also be used whether the TB patient is responding to treatment or not. So how we'll know that subject is responding to treatment or not? So at 0 months, the person is sputum microscopy positive, nucleic acid amplification gene expert test positive, it's culture positive. After 2 months of completing the intensive therapeutic treatment, if the subject's sputum sample has still viable bacteria, or viable in the sense like if it is microscopy positive, then it is a non-responder. So after 2 months, still if the bacteria is present in the sputum of a TB patient, it's a non-responder. So the clinician has to give a different treatment. And if it is absent, then it is a responder. So we wanted to see like whether these 4 molecules can also predict responding status or not. Or the limitation of this study is the limited sample size, like we have just taken 4 subjects. And from this, it seems like ApoC3, apolipoprotein C3, which inhibits lipase activity, it's actually in the responders is getting upregulated, whereas in non-responders, it's getting downregulated, and maybe that is contributing to a compromised system, host system. And that needs further investigation, and we are working on it. So to summarize, we have identified a minimum set of 4 proteins, KYT3, which works, it's on the right-hand side corner, it's mentioned like tryptophan gives rise to kynurenine. Kynurenine breaks down to convert kynurenine to kynurenic acid. And this kynurenic acid happens to be little bit, has psychological effect. So we do expect maybe that is one of the probable mechanism that a probable molecule that might be contributing depression in TB patients. That needs further validation, and we are working on it. Then SERPINA1 is involved in neutrophil degranulation and all. It protects the host. And in TB patients, we do expect degraded hemoglobin is higher, and those degraded hemoglobin also leads to, gives a fertile ground for the Mycobacterium TB for its multiplication and all. So host increases HP production to limit the access of iron to the mycobacteria. And apolipoprotein C3 involves in lipose, it controls lipogenic gene expressions. So it higher ApoC3, when it is high, the lipolysis is high. So so this exogenous proteomes plays a critical role, and it explains a detailed pathophysiology of a tuberculosis patient. So these are all the few observations we have. We know our limitation that we have very limited samples. These are all observations from few sets of subjects. This needs expansion. And the message I wanted to share is like, we can do all this activity staying in this country. And all this work is done by our brilliant research scholars. And we get the opportunity to talk, but the hard work is done by them. So, and the funding we have received from the Department of Biotechnology and other international funding agencies. And all the credit goes to my clinical collaborators who help us to reach to this stage. So with this, I thank the organizer for giving us this opportunity. Thank you.
Thank you, Dr. Nanda, for your nice presentation on the subject. The discussion is now open, and we are taking the questions in this session. Dr. Jyoti, please.
Thank you.
Thank you, Dr. Nanda, for a nice deliberation. Of course, we enjoyed a lot. Hopefully, I am audible there.
Yeah. Yes, please.
Yes, please. So there are lot of questions coming from various participants. So whether Ranjan sir, whether it is feasible. Okay, let me try out. Let me go with the question, or will you comfortable reading the chat?
If you want, I can take it from the chat box and start.
Of course, sir. Of course,
one
sir.
by one.
Of course, sir.
Thank you. So Dr. Jyoti asked a question like, what are the driver genes for TB? I think there are a lot which can influence the immunity of a system. The simplest example is vitamin D related genes. So whether we can tell them as driver genes or not, I'm not sure about it. But there are lots of genes which affect the human immune system, can compromise the immune system of a subject, leading to conversion of latent TB to active TB. So I hope that answered. The next question is from Ritu. What are internal standards? So as I have shown that lysine D4, we have used as an internal standard. So generally, like if it is a mammalian system, you can take a molecule of plants to add it in the sample so that sample to sample variation and day-to-day instrument variation can be taken care. So that is called internal standards. And then internal standard can also be used for validating a molecule. Like if you say glucose is getting altered, you add glucose as internal standard and you see the peak area is increasing. Then you tell like what I am claiming as glucose is really glucose because when I'm adding glucose as internal standard, the peak area is increasing. So I hope that answers. Then the next question is NTB and active TB, how to categorize it. So it is purely as we have discussed that both these groups have similar symptoms. So when they go to the clinician, the symptoms are identical. But if in the sputum sample, if we identify presence or absence of mycobacteria through microscopy, then if it is present, it's called active TB. If it is absent, then the subject is given additional taste like culture taste or nucleic acid based amplification test. If it is like, if we still find MTB genetic material in the sputum, then we also group them as active TB. So the non-TB subjects are lung cancer, COPD, asthma, pneumonia patients. Then Deepak Kumar Panda has a question like, how long one can live with untreated TB? Which type of food help to cure TB? Which age of people is most chance of affected by TB? It's a wonderful question. All three are interesting. I think we all should have healthy food irrespective of whether we want to cure TB or cure COVID or anything. We should have a healthy, we should take healthy food to have a healthy immune system so that we can fight any kind of infection to the best possible way. Then how long one can live with untreated TB? If it is a latent TB, we may live till our last day without treating it. Or like we follow the different guidelines which has been put by different agencies. But it has been, just for your information, there are lots of subjects which has never been reported having TB after their death when they get their skeleton sample. And in skeleton, they identify that the person was suffering from bone TB and all. So maybe we might have TB, but we don't know, but we lead a healthy life. That can also happen. And that goes to the credit goes to our healthy immune system. And of course, like as you mentioned, we should have good food. So type of food depends on lots of variables, so I may not comment on that. Then which age of people? It's like all kinds of age, all age people, irrespective of the age type. You see, if a child doesn't take vaccine, BCG vaccine, he can get miliary TB and the person can die. So, as far as reporting is concerned, majorly 35 to 55 years of old people report TB cases because they work. That age is for workers, so they have to work. If they have issue, they go to the hospital. Old age people are more susceptible to TB, but but majorly, I think it affects all kinds of age. Okay, till time. Then next question is by Dr. Jyoti Prakash Rath. Till time we have age-old therapy of dots. Why new therapy is not available? It's a very fantastic, simple but very difficult question to answer. TB drug therapy is not easy, and developed world has not invested good amount of money on TB drug development. It's because it was a poor man's disease earlier, but now after HIV came into the picture, then HIV patients started developing TB. So the developed world started waking up and invested money on drug discovery. And as we all know, that drug discovery always takes lots of time to validate and come to this stage. But we are lucky that at least few drugs are in pipeline. Lots of drugs are in pipeline. Few drugs are in trial, especially for drug resistance TB. Then again, Dr. Rath's question is, India leads the world TB population. Is there any uniqueness in Indian genome? Any reporting for that? We don't have much report on Indian genetics, but I think our economy system and the lack of food, lack of access to medicine. But I think we have done a great job in last 5-10 years, and I think the country is doing a lot to improve this situation. Sarthak Kumar has a question. How exosome preparation is done? I think, Sarthak, I have explained to you that ExoQuick method we have used. It's an antibody. There are multiple methods of exosome isolation. We have used ExoQuick TC as well as ultracentrifugation, density gradient ultracentrifugation method. So there are multiple ways of isolating exosomes. Then Jayant Kumar Barik had a very nice question, like, is there any kit to detect the positivity of TB through blood sample? Unfortunately not. Actually, there is a test for checking latent TB using blood samples. That is called IGRA test, interferon gamma release assay test, but that is not for active TB diagnosis. And but unfortunately, in some clinical settings, people use it. I think that is not good. Then okay, then Rajeshri Panigrai has a question: how to detect the TB except the sputum test? Do we have TB bacteria in exhaled air, just like detection of alcohol in mouth air? Can we do it for TB? It's a fantastic question. My team works on electronic nose development, where we check the exhaled volatile organic compound analysis, which are present in the TB patient's breath sample. We don't detect TB bacteria because, again, if you want to detect TB bacteria from exhaled air, then also you have to trap it on a medium that has to undergo a microscopy test or a culture test or a nucleic acid amplification test, which will be effectively similar to what we are doing with sputum test. So I think you are right. Detection of alcohol, the way we do using electronic nose, can be done using, and there are lots of companies are into it. We have lots of competitors. I wish I could have presented some work on electronic nose, but maybe some other time. Thank you. Thank you for the nice question. Okay, then Sarthak Kumar has a wonderful question. Is there any study between relation of TB and gluten allergy. As for my child, Montux was positive, but all tests were negative. Even bulb test was negative. Later, after a few years, he was diagnosed with gluten allergic. I'm sorry, I don't have an answer to your question, but I don't, but IGRA, sorry, the sputum, this Montux test is nothing but a skin taste. And skin test is positive in maybe 1/3 of Indian population, is because we have lots of environmental mycobacteria and we are exposed to it, so it gives an immune reaction. So it doesn't mean that we have active TB. Then Dr. Subrat Thanapati has a nice question. Host genetics have roles in development of resistance in TB. That's a very tricky question because then we are telling that the TB bacteria, susceptible bacteria goes inside the host, and when due course is develop resistance within the host. I don't have any, I have not come across such papers, but I'll not be surprised that if something like that is happening. Similar question somebody asked for Dr. Sandeep in the first presentation, like whether the COVID patient in a weaker immune system, weakening system, subject with weakened immune system can be a fertile ground for COVID, new strain of COVID. So like that, I don't have an answer for that. Then Sarthak Kumar has a question. He was diagnosed as mediastinal glands were swollen and was put on TB medication for 9 months. Actually, as we have mentioned, lymph nodes, lymphatic system actually attracts MTB very nicely, and sometimes MTB goes, and sometimes you see people with a nodule on the neck, and that nodule is highly likely because of TB infection. That's the reason the doctor might have given TB medication. And as we have lots of TB in Indian continent, I'll not be surprised that doctor might have done some test, like some test to check presence or absence of MTB in those swollen glands. And that's the reason he might have given TB medication. And it's good that if the subject has completed it. Then, don't you think, Dr. Jyoti Prakash has said, like, don't you think the sample size is little less in the first study to generalize any conclusion? I completely agree with you. It was a pilot scale study. And whenever we do pilot scale study, we do it with available sample size. But, and that is a limitation. We accept that limitation. But patient-based study always needs lots of money. And sometimes funding agencies has to be generous enough to recruit that many subjects and do such studies. And I think we are into that business, so we'll be able to do it in coming future. Then Ayesha Abhilipsa has a question. Can we include TB as an infectious disease? Of course, Ayesha, TB is an infectious disease caused by Mycobacterium tuberculosis. So of course, we can include it. It is an infectious disease. Then Dr. Jyoti has asked how far bedaquiline holds true for its promise. Yeah, it's working really nice, and I think, yeah, it is doing its job. Yeah. Then from Galaxy M20, somebody has asked, what is the best way till now to diagnose TB? I think, as we discussed in the first slide, TB is not only a respiratory disease condition, which majority of us think like. So if it has affected the ovary, if it has affected the skin, if it has affected eye, then there are different methods to do it. So I think a clinician is the best judge to recommend the suitable diagnostic TB test. Then Dr. Santanu Bhattacharya has a nice question. What is the possibility of patients with asthma and bronchial disease towards TB? Respiratory diseases might compromise immune system. So any disease that compromise immune system can lead to conversion of latent to active TB, especially in TB endemic countries like India. So Dr. Rajyasree Panigrahi has a question. People are having TB in spinal cord and uterus and other areas. What are the symptoms and tests to detect it? Spinal cord, people do scanning, and from that the clinician think that okay, there might be some issue, so they take CSF sample. Tuberculosis meningitis is a big problem in our country, and it's very difficult to diagnose. There are lots of established TB researcher, not lots, there are few cases, established TB researchers has tuberculosis meningitis and they didn't have any clue about it till they suffered they suffered some incidents where they lost whatever happened, and then they were admitted to hospital, and then they got to know they have spinal TB. And uterus, sometimes if the women is not conceiving, that time doctor do certain tests, and then through biopsy it can be checked. Eye has other areas when you asked. Eye and skin are other organs. We have some good work from Bhubaneswar. People have done eye-based TB infection studies and all. And nucleic acid amplification test is the best method to quickly diagnose the TB, presence of TB. What are the different types of TB apart from lungs? As TB vaccine exists, why we are still fearing TB? TB vaccine do exist only for children, not for adults. That's the reason we are fearing for TB. And I think it affects all other organ systems. Any method to minimize mother-to-child transmission, it doesn't happen much. But again, if an index patient inside the house is having MDR TB, we do expect the compromised immune system might, whether it is a child or an old person, can get disease from the index subject if they are staying in the same house. Dr. Rath, does TB transmit through mother's milk? I think no, but I could be wrong. Dr. Subrat Thanapati, can you please give ideas on the differentiating genes in different kind of TB? Are there any specific genes expressed in active while no or low in latent? Actually, gene expression, of course, will change when the bacteria is in latent stage, because it will definitely use minimum of its gene activity for maintaining its basic function. But when it is active, it overexpresses everything, including genes responsible for taking food and for its multiplication and all. Dr. Santanu Bhattacharya, hematogenous spreading to infants. I don't think so, because there are reports from AIIMS New Delhi has shown that if a subject's blood is now having MTB, that subject will die in a couple of days. So hematogenous spreading to infants, I don't have knowledge on it to comment, but yeah. Manish Paul has a question. Can tuberculosis stimulate the development of any other diseases? Yes, Manish. If the immune system is weak, whether through TB or not TB, it can develop other diseases. Yes. What is cutoff used for PCA? PCA, Dr. Jyoti, we don't use any cutoff. There are other parameters we select, if the molecule is present in more than 50% within the class, we select it as a variable and we include it for PCA. Elena has a question. Is it rare for a TB patient to be put in isolation? TB patient, if it is really MDR or XDR patient, keeping them in a specific area is better, but I don't know whether we can use the word isolation here or not. Dr. Rath, so next generation will not be bearing any TB germ as vaccine exists. No, vaccine is only effective for pediatric cases, and after 17, 18 years, the BCG vaccination loses its efficacy. So adults have TB, adults will have TB, or may not develop disease, but it may have problem. So vaccination for adult TB is an ongoing process. We are yet to be successful in that. Correlation between diabetes and TB, that's the Dr. Rath's question. HIV makes 30% susceptible for latent to active TB conversion. Diabetes makes 3 times more susceptible for TB conversion. So diabetes has a strong correlation with TB. It makes 3 times susceptible. Gene level correlation, we are working on animal model and human subjects having tuberculosis and tuberculosis diabetes comorbid condition. Gene level correlation, expression and correlation data, maybe next year I can share those data from our lab, from Indian tuberculosis and diabetic conditions. That's fine. I think I'm stepping over to the time of the next presenter. So thank you.
Thank you, Dr. Ranjan. Sir, please.
Thank you.
Thank you, Dr. Nanda, for your nice presentation. So nice to hear you about all the aspects of tuberculosis research. I especially thank you for your interactive session on the which were being answered, the questions being answered by you in a so lucid way. I hope the audience, they have enjoyed a lot. They have gained a lot from your presentation. Thank you once again. Now, in the second phase of this session, I welcome Dr. Niharika Bhattacharya for his lecture on the on her subject protection of biotechnology innovation. I once again welcome Dr. Niharika. Dr. Jyoti, please have the welcome note, Dr. Jyoti.
Yes, thank you, Dr. Nanda, for a nice deliberations. And thank you, Chair, to allow me to introduce our next speaker. Hopefully, I'm audible there.
Yes.
Yes, okay. So, thank you again. So now it is time to march on to the next speaker. We have an eminent speaker joining us from IIT Kharagpur right there. And now we start with bioinformatics. Then we went on to the wet lab. Now we are thinking that how to safeguard the intellectual property. So that's what next speaker, then that's what we wrote in Dr. Niharika Sahu Bhattacharya. So to safeguard the intellectual property. So let me introduce Dr. Niharika Sahu Bhattacharya. So Dr. Niharika Sahu Bhattacharya presently working in School of Intellectual Property Law, Indian Institute of Technology, Kharagpur. Before joining the Premier Institute, she had her PhD from IIT Kharagpur in intellectual property right. Then she worked in University of Cincinnati, Ohio, US as a research fellow. Then she served as a scientist at National Institute of Science, Technology and Developmental Studies, that is NISTADS, the CSR Institute. And presently she is much engaged in the IPR protection and all. She is a quite busy fellow in intellectual property, safeguarding intellectual property of the motherland, India. So she had many publication and patent to her credit, and she will speak on the how intellectual property can safeguard the biotechnological product. So I should not be any hindrance between you and audience. So over to you, chairperson. Over to you, sir. So over to you. So may I request Dr. Bhattacharya, Niharika Bhattacharya, to share her slide and start the session.
Okay. First of all, a very good afternoon to all of you, respected chairs, like Chairman sir, and Dr. Jyoti. Thank you for inviting me to this session. It's a real pleasure to be a part of this forum, and I have been hearing from morning. You all are having some technical sessions in different aspect of the science and technology, particularly in biotechnology and plant sciences. And even now we heard Dr. Nanda talking about his research. So my topic would be a little bit different. So different, but I would say much relevant to all the discussion. Just to give you my background, so I'm also a MSc in Life Sciences from Sambalpur University, and then I did my MPhil from Sambalpur University with specialization in microbiology. Then I was initially in IIT Kharagpur, part of working as a junior research fellow in Department of Agriculture and Food Engineering. But suddenly at that time, IIT Kharagpur opened its first school, which is known as Rajiv Gandhi School of Intellectual Property Law. It is something unique of its like kind, for one and only school in whole India, which takes the technological student and teach them the legal aspect. So I also changed my track, and for the first time, like I ventured into a totally new discipline, which I also never read or heard about before, but kind of a bet on my career. But that I think I have successfully now defended my thesis and earned a PhD in intellectual property right. And particularly, I worked in the interdisciplinary area of biopharmaceuticals and herbal medicines and intellectual property rights. And after that, my journey started, and I learned how this subject, intellectual property rights, particularly you all might be hearing about patents, trademarks, copyright, and everything like that has to do with us as a technologist or biotechnologist or a botanist or a plant biotechnologist. So very relevant area, but unfortunately, we all are not aware about many of the facets or aspects of this thing. So I thought it would be a nice interaction with you all, budding technologists and already teachers of Centurion University. So if I can give you some basic preliminary idea regarding the various aspects of the intellectual property law. So on that aspect, my presentation is particularly on protection of innovations in the area of biotechnology. I'm just sharing my screen. I hope it's visible. Is my screen visible?
Yes, ma'am.
Okay, thank you.
It is visible.
Yeah, thank you so much. So I would be short, and I know we are already a little bit of having paucity of time. So in this presentation, I would be basically focusing on what is this IPR we are talking about so much. How to identify the different IP, which as a technologist or as a scientist or as a teacher, you all are generating during your day-to-day work in the lab or somewhere in the table also. And what would be the most relevant form of the protection for the same, and then how we can protect the different aspect of this intellectual property. So the thing is that we sometimes work and we gain knowledge, and being in the field of education and academics, like we think, okay, knowledge is like it's coming and it has a potential value, and we keep on working on it, still we develop something good and we keep on percolating that to the other persons or other students who work along with us. But sometimes knowledge as such does not have a value unless you realize what is that value of the knowledge. This potential energy has to be converted into the kinetic form that would give you much more benefit in the other way, which is not achievable. So in that domain, the intellectual property rights is a kind of asset or a instrument particularly, which will not only impart value to your innovation, it helps in exerting the ownership over the particular type of innovation or inventions which you are working upon. And it also motivates the other fellows to create further innovation. And overall, the innovation management, the key to our this current knowledge economy, that you keep on innovating, you keep on harnessing the benefits of your innovations and invention. So in that way, the intellectual property plays a central role. So if you look basically the functions of the intellectual property right, so it gives you basically two forms of the right. One is the moral right over your expression, over your invention, over your thought process or work. And also that moral right allows you to reap the economic rights or the benefit over that thing. Secondly, the indirect benefit which we as a whole towards our society or the public is that it acts as a kind of incentive to promote or to create more and more innovations or more and more such good innovations which would be helpful to the human being. So this intellectual property right and economic development are like closely related to each other. You talk any of the things, whether the issues related to climate change, whether the issues related to the global health problems, or whether the issue related to food security. Inventions are being there, but unless and until that invention gets an incentive, it's not going to again develop. Let's take the example of this COVID. So many companies were working on this COVID vaccines. Only now few are in the clinical trial phase. Only one or two have succeeded. Why it is so? Because unless you exert that power or exert that monopoly or exert that what you called that right over the things, you are not you will be not able to harness the benefit of the innovations in a fullest manner. So it is very important to have the intellectual property in the field of scientific invention. And intellectual property and the public interest, the access benefit, the incentive to the other inventions, and overall the whole development of the intellectual property are linked with each other. And that's why it is crucial for, as a scientist or academician, for us to understand this intellectual property right. And like in India, like obviously I would say from the last 10 years, we are trying to be more efforts are being given to educate more and more on the intellectual property aspect, but still at many levels we are unaware. And even you get the exposure to the intellectual property, if it is not done at the right time, it won't be that beneficial which is expected from it. So my presentation will be a little bit of not theoretical but information based. So you might feel that sometimes why I'm talking about it. So later on you may realize the importance of this thing. So anyway, moving forward. So as I mentioned, intellectual property is essential, not only exert economic rights, but also help you to commercially exploit your inventions. It gives or allow the funding or the venture funding or investment on your technology. It allows you to transfer the technology to various other domains or other persons so that the technology can be utilized fully. And further, the overall cultural development of the society can take place with the help of this intellectual property rights. Now you can ask me, so what should we do? So sitting in the academic arena, so we are creating a lot of intellectual property with our day-to-day life. whether you write a thesis, whether you write a paper, whether you do certain scientific experiment and develop a new thing, whether you sing a new song, write a lyrics, anything, you try to develop a new plant variety, or you try to develop a database, you are creating certain software, some bioinformatics tool, even you have certain confidential information along with you. All these are different forms of the intellectual property. It is not a thing that only patent. We as a scientist hear more about patent, but no, patent is not the only one.
Me, Dr. Niharika.
Yes.
Could you please full screen the slide?
Okay, sure. Just give me a second. Yeah. Is it visible now?
Yes, ma'am.
Yes.
So, what I was talking that, so we can create a lot of different types of the IP, intellectual property. Now, the problem is that, okay, how do you identify that? Okay, which one is what? How do you protect what? That is the very next questions. So if you look into the broad category of the intellectual property rights, the intellectual property rights can be categorized into two forms. One, which we call as the industrial property right, and other one most popularly known as the copyright. But apart from that, there are few other things like which we call these allied form of the intellectual property right, or the other forms of the intellectual property right, like we have geographical indication, we have trade secret, we have semiconductor and integrated circuit design protection, we have plant variety protection. So these are different forms of the intellectual property right. And if you specifically move towards the industrial property rights, which is mainly comprised of these patents, trademarks, and designs. Why these are called the industrial property, as you know? So they have the direct implication towards any industry or creation of the industry or the maintenance of the industry. So, and most popular, like patents are one of the highly popular form of the thing. Like lot of patents has been filed by many companies, academicians, and inventors all over the world. So these are the basic forms of the intellectual property rights. Now comes to the next question. Okay, I now know these are the different forms. So where should I proceed? So if I give you the broad overview, so when you talk about a patent is something which is given for an invention. Okay, and it is a kind of a monopoly right, or a right, or a kind of a property to you, so that you can say that this is my inventions and I am the owner, and apart from my permission, no one is going to use it. And copyright basically is something which protects the expression of the literary or the artistic work. Okay, it's a kind of an automated right. You don't have to really go for a registration, and it's not mandatory in India. So whatever you write, whatever you create, whether it's your thesis, whether it's a drawing, or whether something, that's automatically gets copyrighted to you. And if you come to trademark, so trademarks, as you popularly know, it's a name, logo, or signs, which particularly helps you in identification, which gives you a kind of a brand name or image. So those are the trademarks. Designs are a kind of outline or the exterior shape or the appearance that gives an aesthetic value to a certain invention or a product. So in that respect, the design becomes very important. Then we have different forms like various databases. So databases may be copyrightable, but these days the databases have many other functionality. So this arrangement of the information in a novel manner, so that can also give you certain right. Then another important thing is the trade secret, where like the Coca-Cola is the best example which I can give. So nobody knows what is inside that, what is the true composition of that. And that secret, that crucial information gives you all the benefit over your product or the invention. So these are the basic forms. Now I would like to explain each of these, and particularly patents, a little bit more, so that will help you in going forward. So as I said, so patents are given for inventions, and patents are an important form of the intellectual property right. So now You said, okay, we are doing a lot of experiments in our lab, so this is all inventions. Maybe, but you know, this is intellectual property law. So as a definition, as a like the law is very what you call science and law differs is that laws go by the definitions which has given. So already there is, if you go look into the meaning of the invention in a dictionary, so there are three things. It may be an invention, it may be a discovery, or it may be an innovation, or sometimes it may be a know-how. So what is that you are doing? What is it does it qualify to be an invention? So what is the basic difference between an invention and a discovery? So discovery is something that which is usually there. Okay, you know, okay, there is a microorganism in the earth crust, or there are few algae, few bacteria, there are lying deep inside the sea. So if I isolate a bacteria from 10,000 meter down from the sea level, so that cannot qualify as an invention. That bacteria or that microorganism was already present in the sea. What I did is that I have isolated. So that bacteria is something, is not that which I created. It is already been created. I have just isolated. So can I claim a patent over that bacteria? No, not really. That won't qualify for you for that invention. So you have to now understand what is the difference between an invention, an innovation, or a discovery, or the know-how. Innovation, sometimes innovation is what? Any new thing which you have developed. So that can qualify as an innovation and also moving forward to an invention. Know-how, what are know-hows? Know-hows are the, okay, I know there is certain technique by which I can, like you can pipette, or you can use the pipette, or you can pour certain inoculate the microorganism. That's the know-how. So our teachers have taught us how to do this. So there is a technique, or if someone going for a large scale manufacturing, there are certain procedures or certain things which help you in achieving the things in a more efficient manner. So those are known as the know-hows. Patents are applicable for the inventions only. And inventions means, as per the legal definition, it can be a new product. It application. Okay, so we have basically three types of the invention. One is the product invention related to the product, invention related to a process, an invention, a process leading to a product. And but again, it is not that whatever you are developing may qualify for the patent. So in order to have a patent, you need to have a substantial new thing which meets certain criteria. So this is just an example of few of the innovative things which we come across these days. So like this surgical robot, where instead of this manual operating things, now the robot can carry out the function. So the Samsung is now claiming a patent over the surgical robot. NASA's weather predictor. So they have adopted different devices through which they can predict the weather, how it is going to change in a minute by minute basis. The Apple is coming up with a self-deploying screen protector, where if a screen of the mobile phone is breaking, it will automatically rejoin. Then in the US, we have this pollinating bees, where what you call where in US, where acres and acres of land is a single homogenized type of a crop, where the land diversity of the plant biodiversity is really coming down. This pollinating bee would help in sustaining the biodiversity, in generating new species, in helping this pollination of the different plants. So this is the pollinating bee which they are coming forward. So like these are the various inventions which we see. Now you may have the question. So I am working in my laboratory. I am developing some or I'm working on some project, developing certain new things, new protocols, new product. So how I can get a patent? So before you come to that questions, you have to first identify what is the thing, or what is that thing which can qualify as an invention. Okay. So to understand that, you have to have the proper answer to the following question. What is the technological area where am I working? Does my subject matter of the invention falls under the category of the patent? how it is different from the available knowledge in that area, and what is the utility of the invention. Can my invention be again commercialized, or can it have a potential market? So all these questions have to be answered before you can think of about a patent. So as per the legal intellectual property thing, or we have a patent law. So as per the Indian patent law, in order to have a patent, the patentable invention must satisfy few conditions. Basically, it has three conditions. The invention, or the thing which you think can be given a patent, has to satisfy three main conditions: novelty, non-obviousness, and industrial applicability. So what is that? Actually, what was the patent? The initially, like if you date back the 18th century or the 17th century, so in the United States, what was happened when someone is coming up with a new thing or a new discovery or a new anything new which was not there, so they were granted a kind of letter from the kings, so that kind of a certificate, like, okay, this person has developed something new or useful. So from that word, the patent has come. You know, the patent can be as simple as a septipin. If you look, if you anytime get a chance to search over Google, if you try to find a patent over a septipin, you will find number of patents. The simple, the James clip which we talk, okay, it has a patent over it. All of you might be using this laser Gillette shaving blades. Do you know how many patents it is covered with? Just if you are purchasing some Gillette blade, it is one of the costliest blade which is used by male persons. So if you see the packet, they will give you around 10 to 12 patent details. These are the patent granted. These are the patent files. So over that blade itself, we are having so many of patents. So it is not that the patent has to be like, okay, this is extraordinary, out of the world research, then only I will get a patent. No, the patent has to be something very unique. And the uniqueness of the invention is established through the few of this concept called novelty, or the inventive step, or its application. So moving forward, so why this patent so this patent basically gives you kind of a monopoly right, or a right over 20 years of time period. A patent lasts for 20 years from the date of filing. Okay, and it is a kind of a right, like if you have a land, if you have that property over your land, then you can say, okay, no one can trespass into my land. I can put a boundary. I can put a gate. I can allow whom I want to, who can enter into my premise or not. Similarly, the patent also gives you an exclusive right to prevent others from making or using it or selling it or importing, even exporting it. Okay, so that's why we all are so far about this patent. But this criteria has to be properly judged by you, then only you should be going for a patent. So when I say about this, so the important thing which one should know about the patent thing is that patent cannot be given for anything and everything over the earth. That is, if you are thinking that whatever I am working can be eligible for a patent, you may be wrong. Okay, so there are many things which can be patented, but again, the law itself poses certain restriction. So instead of, the patent is generally considered as a negative right. It prevents someone from using that thing. So as per as our intellectual property laws, or patent law in particular, so it says, okay, what cannot be patented. In the negative way, it denotes what cannot be patented. So there are a list of substances which cannot be patented upfront. First, any invention which is frivolous inventions, means which cannot exist, truthfully cannot exist, that are known as the frivolous invention, that cannot be patented. Inventions which are contrary to the law or morality or injurious to public health, that cannot be patented. If I say, okay, someone has developed a human clone, or trying to develop a human clone or a superhuman, can I patent that? No, not really, because I don't know what will happen with that super clone. Will that be destructive in nature, or will that be harmful to our society? Maybe positive, but that might have a harmful thing. So that cannot be granted a patent. So that is what known as the contrary, like morality or injurious to public health. Suppose if I can chemically synthesize a poisonous substance which can kill the rat, or which can kill human in a second or in a few minutes. So can that be considered as a patentable thing? No, because that is not good for public health. So those kind of things cannot be given patent over the that kind of application even cannot be accepted by the patent office. Contrary to the law. So if I say, okay, this machine works against the principle of gravitation. No, nothing can work like that on the surface of the earth. So that cannot be contrary to the law or existing principle, scientific principle. So that is also not allowed. Third, mere discovery of a scientific principle or formulation of an abstract theory. So we now know, okay, E equals to mc square or F equal to ma. Like these are the various scientific principles which we have been studying, the gravitational force and other things. So already that is there on the earth, and that is the basic principles through which our earth or we as a human being are functioning or running. So those kind of that it will be considered as the scientific principle. Mere discovery of the scientific principles cannot be given a patent. If you have any theory over any of the principles of working on earth, that cannot also be given patent. Another prohibitive thing which is prohibited under the patent law is the mere discovery of the new form of a known substance. If I will give you an example, simple example, suppose now we know paracetamol, all of us use paracetamol for fever. So tomorrow I will say, okay, it's for fever by bringing the temperature, which helps in bringing the temperature down. And then I will say, no, paracetamol can be used for body pain also. That's a new use of that same thing. So it is not something new, or that is okay. This can be relatable. Or tomorrow I will say, okay, body pain, or it can be helpful for stomach ache also to some extent. Suppose I again did some study and I found that, okay, that can be helpful for the stomach ache. So can I get a patent over that? Not really, may not be possible. But yes, tomorrow if something can say, no, this paracetamol can be used for dementia or used for any other disease. I'm just giving you a very vague example just to get to the point. So that's a mere use. So mere use or the new secondary use of the substance would be like, okay, body pain or head pain, leg pain. These are all related things. But if I'm talking about something normally used for fever and something which has been used for a lesser known disease like dementia or any other thing, it's substantially different. The parameters which is applicable to study fever cannot be applicable to dementia or any other thing which is completely unrelated. In those circumstances, there might be chances that your invention may be considered as a patentable thing. But if it is not, then it would be considered as the mere discovery of a new form of a known substance. So that is not possible. Then mere arrangement or the known arrangement of the devices. So if my thing has 4 parts, A, B, C, D, I know I have to put A first, B first, then C, then D, then, and then just I do permutation and combination and now put the other things in different order. And I will say, look, this is something different I did. This is not the same which was already existed. Then it won't be again qualifying for that thing. So that would be considered as the mere arrangement or the rearrangement. This is applicable more in the field of instrument or mechanical apparatus and all those things. Then very important to our thing, in India, methods of agriculture or horticulture are not patentable. So any other way in which we are doing or doing agriculture, maybe the way you sow the seed or you cut the seedlings or the other horticultural processes, that is not patentable. Any process for the medicinal, surgical, curative, prophylactic, diagnostic, therapeutic, or treatment of the human being or animals cannot also be considered as a patent. Means suppose tomorrow, suppose you imagine a situation. So patent is a monopoly, right? So we have best heart surgeon and other things. So they do the operations very nicely, or any implantation or something. So the first doctor who has performed those kind of surgical or methods would claim patent over the technique. So what will happen? They will be the only one who will be carrying out that thing. No one apart from their permission or the license cannot carry out the same experiment. So then it won't be serving the purpose of this patent. So that's why these method or process for these surgical purity prophylactic treatment methods are not patentable. Any treatment methods are not patentable. So you might, so treatment methods and pharmaceuticals or medicines are three different things. So treatment methods are not patentable, but yes, if you have developed a new product or a compound which is useful for the treatment of anything, that can be patentable. And plants and animals in whole or part of other than the microorganisms are not patentable. So you cannot patent any plant, you cannot patent any animal, nor they are any part of the plant, even seeds or different varieties, or even essential biological processes. So the photosynthesis, respiration, so you say, okay, so why Krebs cycle was not patented, why Krebs, he discovered that thing. No, those things are not patentable in India. Okay, so plants, animals, but yes, microorganisms are something which we can think about the patent, provided there are few other criteria which need to be fulfilled for that. So in that list, we have other things like mathematical or the business models are not patentable. Then literary, dramatic, or musical works that are coming under the purview of the copyright, so they are not patentable. The rule or the method of performing mental acts or playing games. Okay, so you say, okay, I can play solitaire in my thing. So the way you play solitaire in your system, that is not patentable. Yes, the person who has developed that solitaire might have employed certain algorithm or certain what you called codes. So that may be part of other intellectual property, right? But the way you play the game is not patentable. Okay, and then the presentation of the information. So may I today I am presenting a slide over in front of you, or tomorrow if I perform some other things, that may not be given the patent. Then topography of the integrated circuit. Topography means the layout, like you all might have seen any chip or this motherboard of a system. So you might have seen this green color thing, green, and various transistors and diodes are put over that. So those things are also not topography. The design of those things are not patentable. And very important from our biological science or botanical perspective, the traditional knowledge are also not patentable. Something which has been existed earlier cannot also be patented. So I just gave you a broad list of the substances that is not patentable. So if you are thinking of a patent, so first you have to understand my subject matter does not fall under this purview of this non-patentable things first. So if you are working on some of this and think that that can be patentable directly, that may not be truly applicable. So this is the broad thing. Having said that, now this is the first net you are lying on. Then again, you have to meet certain other criteria that may lead to the patenting of the substance. Okay, so another thing I must say here, all these intellectual property rights include whether you say patent or copyright or any other thing are country specific. Whatever I am telling you is specific to India only. Different countries have different parameters for their thing. Like I say, in India, plants cannot be patented, but in US, plants can be patented. Okay, and what is morally wrong or injurious to public health in India might not be the same in US or in Europe. Okay, so different countries have their own standards for all this. The foundations are same. Okay, the criteria of novelty, inventive step, or other things which I mentioned, as such, the wordings are same, but the interpretation of those things may not be same, and it varies from country to country. And these patent rights are not international rights. The patent rights are country specific. if you have a patent in India, that would be only valid to India only. So that point you should be careful about. Now, first step, you are aware, okay, so my invention, whether it falls in the purview of a non-patentable thing or not. Okay, so if suppose you are working on a method of or a preparation of a recombinant microorganism, or a inserting a gene to a plant cell or an animal cell, suppose a new kind of a, so that expresses a new protein which is helpful for human disease or something like that. So that is not falling under the purview of the non-patentable subject matter. Then what would be the next criteria? So the next thing would be that the patent office. So what is the patent? So basically what is, so you apply the patent to the patent office. So in India, we have 4 patent offices in Kolkata, Mumbai, Chennai, and in Delhi. So now you can go for e-filing. So you file basically an application for the patent. Okay. So for that thing, what the examiner will look, first you will say, okay, what you have written. Then after ascertaining that this invention falls in the criteria of the patentable subject matter, he will check whether the other criteria are met or not. So what other criteria? I first told novelty. So he will check what is the novelty of your work, means how your work can be considered as a novel. So what is novel something? Means which did not existed earlier. So something very new. Okay. So means today I'm wearing a new dress. So how it will be considered new? If my friends have not seen it, then only it will be considered new. Is it so? May not be really. Maybe I had visited other country, there I have used that dress. So my friends have not seen it. And then I came to India and then I saw, look, this is a new dress which I'm wearing. That doesn't mean that the dress was new, but yes, for my friends, it's new. So here, similarly, just a simulation I gave. So similarly, the invention can be considered as a new or a novel invention, provided it is for the first time coming to the scientific domain. It is not that it was not in India. So this concept is, so the novelty of the invention is considered in an international basis. It should be novel throughout the world. And how do they judge their novelty? Basically, the patent examiner, they check what is that thing which is, so is the same invention was there in the prior art. So what is a prior art? So prior art is something, means was it already published by any other scientist or any other worker? Maybe, or it is used. So prior art means anything which is existed before the application. It may be in the form of a publication, maybe a research paper, maybe a magazine article, maybe a blog in the internet, or it may be the product itself. May not, that may be possible the manufacturer has not written anything and he has started manufacturing the product and the product is directly in the market. So what is that thing? So that prior art has to be taken into consideration. So basically the patent examiners are well equipped with different databases. So you say Scopus or other thing, or even patent. So the prior art can be a patent database or a non-patent literature, which is the scientific articles. So the patent examiner checks all those things to find whether the same thing was already existed or not. So that would be considered as a novel invention. So, okay. So for imagine for the first time, someone has developed a marker. So marker is what? It is different from the pen. So marker is a permanent marker. So the permanent marker is something which I will, if I will write on a surface, so that won't be easy to erase. I can erase with some alcohol and other things, that's different. But still, unlike the pen, it can work on any different surfaces and it would be same. So what will this thing, the examiner will check whether there was something called permanent marker or not. Marker was there, white marker is there, but permanent marker was not there. So this permanent marker will be a novel thing. Okay, so the criteria would be met. The second thing would be, okay, non-obviousness. So how do you judge that a thing is non-obvious or not? Basically, non-obviousness means like if someone is developing a permanent marker, is it the technique is so different that none of his peers can really produce it. So what they will do, So I said, okay, I have whiteboard marker, I have breadboard marker, or I have other markers, but not permanent marker. Marker concept is there. So permanent marker was the novel thing. But in order to make the permanent marker, what is that needed? Okay, it has some ink, which is basically making it permanent. So how my other marker ink or whiteboard marker and this permanent marker are different? It is something, so by addition of certain chemicals, it is making that ink to be permanent, or what is that? So the inventive step associated with it. So suppose the inventive step is such that like none of my peers can, or the person who are working in that field would not be able to get that, then it would be considered as the non-obviousness. Or how that they think that is judged basically. So they will see, okay, there was a paper which talked about marker. There was another paper which talked about how ink can be made permanent. So is it that by reading those papers, I just created a marker which can be made permanent? So then it will be considered as an anonymous. It's quite obvious that, okay, I read two papers and then I found out, okay, how to go about that. But if it was not that, there was no correlation between that. It was just a some paper was there, okay, some chemicals which is like the stability of the chemical can be increased in some way, and I somehow related that thing and inculcated that technology to my normal whiteboard marker and made it a permanent marker. So in that case, it may be possible that it is satisfying that inventive step criteria or the non-obviousness criteria. Means it was not so easy. There was no teaching or suggestions or motivations which is readily available, which led me to create this kind of invention. So that is known as the inventive step associated. So if your invention is novel and very new, okay, permanent marker for the first time, and it's like it has inventive step, means nobody thought how to make a permanent marker. I'm the one who is making. So that is making it a patentable invention. Yes, there is a third criteria which is known as the what is called industrial applicability, means what is the potential of this invention. So when I say I have a permanent marker, so it is like what you say, that thing, like for the first time it is coming to the like so it can be used. So so that is the industrial applicability of the same. So basically, you have to understand, means what is the problem which is being solved, what is the problem you are targeting, or what are the solutions to the problem. So all these questions will lead you to understand the whole innovation process, and that would make you understand how to like whether the invention is patentable or not. Just excuse me 1 minute, please. There is some. Excuse me, sorry for that disturbance. Yeah, so basically, so you have to now be careful about the different parameters of the thing, like how your invention or the thing which you are working is very different from the existing literature. So that's why like you always might be suggesting to your students that, okay, you should be doing a good literature review before you write a thesis. So what is the purpose of that? So basically, in order to understand the existing work or the available literature, basically we motivate our students so that you don't repeat the same work, and you come out or you basically find out the lacuna in the literature, and on that lacuna you start working, so that that gives you a fruitful thing. Otherwise, what's the point in repeating the same work if it has already happened? So in that way, you have to, similarly in the case of patent also, you have to be very sure that your thing was not there in the prior art or the literature 1, 2, 3 or whatever is there. So that would lead you to a patentable invention. So if different things are like, there are three different aspects of the invention which are present in different literature, and then you just taken them together and put it and say that I have come out with a invention, that is not really going to be work for a patent thing. Okay. And then, as I said, okay, industrial application is also very important. So then only you can think of about having a what you call this patent. So in order to have a patent, these three things are very important. As I mentioned, the invention should be novel, it should have certain inventive step, and it should be of industrial application. So in IIT Kharagpur, I would just wanted to show you this one. So here in IIT Kharagpur also there are many scientists who are working on different things, but still we do not have that much of patent, because what happens as an academician, many of the time we do not really understand, or it's very difficult for us to understand what can be patented. So this is a kind of invention disclosure form of which we ask our professors to fill out, and they themselves tell us, okay, what was the earlier references they have been using, what are the state of the art, whether there are some patents in this area or not, or there are some research papers in that area or not. Then we also ask them, okay, now you yourself tell how your work is different from the existing references which you have taken. And if they are able to now find out the main points on which their inventions are good enough or distinct enough, then we allow them to go for the patent filing. So all these things must be taken into consideration, because why? Because patent is something that will give you a monopoly, and it is not that comes for free. There is a cost associated with a patent. So there is a filing fee, there is again maintenance fee for the next 20 years. So before you incur that cost, you should be very careful to understand whether my patent is a patentable invention or not. So once you are quite sure about that, then you can get a go ahead and go for the patent filing. So this is about all this patenting thing. But to just give you an example how the things can, this is a timeline which I wanted to show you regarding this CRISPR-Cas9 gene or CRISPR technology. So you all know now this CRISPR-Cas9 genome editing tool has been has been given the Nobel Prize this year. So it's a long history. Okay, so like from 1987 onwards, the work in that area has begun, and currently we are in 2020. And many, like more than 100 patents have been granted in the field of the in the field of the CRISPR. So what is that CRISPR? It is a gene editing technology. So here, CRISPR is basically found as a repetitive sequence. So this sequence is not patented. It's the thing how you can cut a genome and then put certain genes, and other things are patented. So in patenting thing, or this thing becomes very important to identify the technique. So as I've said, microorganisms as such cannot be patented. For the first example which I gave you, that you isolate a microorganism from the sea and you then go for patent, you cannot be, so it's difficult for you to get a patent on that thing. But you can claim how you have isolated the microorganism from that thing. Or if you have developed a culture media where the microorganism can be grown properly, that can be a patentable thing. Or you can modify the microorganism by certain recombinant DNA technology, or you can put the some essential genes in that and secrete certain protein. So that can be patentable, but as such, the microorganism cannot be patented. So for example, earlier what was happening, because the science is ever evolving, the biotechnology also evolving every day. So earlier what was happening when the Human Genome Project has not started, so if anyone is isolated a gene sequence, he may go for the DNA sequence or the RNA sequence and can go file for a patent. But these days, simple isolated gene sequence cannot be patented. It has to be certain modified forms of the gene. Like if you have developed a cDNA, suppose, or if you have developed certain expression sequence tags or some things which can be used for the diagnostic or any other probing purpose, that might be a patentable thing, but not the gene itself. And another important thing for the patent is that the patent is given to the true and first inventor. Okay, so basically the problem with the researcher remains is that sometimes we as an academician are more inclined to publish rather than patent. Also, if you find something, okay, write a paper and let's get it published. But if you have something which you think can be patentable, please don't publish it first. You just file an application first, then you go ahead for the publication. Because as I mentioned, the patent is checked with respect to your prior art. So if you publish something that may, even though it is your own work, that may act as a prior art for yourself. So it is advisable that first patent, then publish. At least patent means you from your end file the application there, because the prior art will be checked from the date of filing. So once you file the patent, it's no problem. Like that would take your patent thing would take its own course. Then you keep on publishing or writing papers over it. Not a problem. Similarly, in this Cas-Gene also, there was an issue. Two groups of scientists were working, one along with this MIT and UC Berkeley, and another group of scientists from the Broad Institute, Boston. So both of them had filed a patent, and there was a litigation saying who filed the first application. So the persons who are working in UC Berkeley were the first one who filed the patent, and their patent was and they were considered as the true inventor. Means they were the first one who reported this Cas-Gene editing technology. So their thing was granted patent. And as I showed, these are the scientists, these two ladies, this microbiologist Emmanuel Carpenter and Jennifer Doudna. So both of them were awarded the Nobel Prize for this Cas-Gene technology. While the on the above photograph, you can see that third person, Mr. Zhang, who is another prominent person who has worked in the area of the Cas-Gene. Even though he is also working in the same area, but he is not considered as a pioneer in this thing. And all of them were associated at some point of time. So this patenting is like very interesting thing where, like, if you have filed the patent first, you can be given the patent first. So, and the patent has larger implication. You know, this this CRISPR technology, it's just a 10-year-old technology, and there are more than 10 companies working in this thing, not only in the area of human therapeutics but non-human therapeutics, and like millions of dollars has been invested in this. So patenting gives you not only economic, the moral right, but also economic right. So the person who has the patent first would be, if he licenses the technology, then the other company can be able to work on it. So that's the importance of the patent, why we always think of patenting. So this is all about the patent. So now as an academician and a scientist, if you feel like patenting something, so better you judge yourself whether your invention is truly can be patented or not. How different is your invention, and how it can be helpful. I believe I'm exceeding the time, so I should stop here. And there are other intellectual property rights like copyright. So if you are writing something, that can be given copyright. And as I said, the other forms are there, which may be applicable as and when situation arises. So that's what for the day. Thank you so much for having me. So I would be happy to take certain questions if you have any.
Thank you, Dr. Bhattacharya, for a nice session. Of course, we enjoyed from the first to the last slides, and that is not at all that you are get more from your side. So many questions pop up in the chat box. So whether you will take it on your own, or shall I read it out as your convenience? You can take. You can tell.
Well,
Okay, I can read it out. If I miss someone, then please point me
of
out.
course,
Okay.
of course.
Sai, okay, so Mr. Sai has asked me, if we consider X as did it rain and Y did people use umbrella, with a response of false from both the sides, what will be the end result? Sorry, I didn't get the question properly. So are you, if that question can be explained? Look, so if I'm really unable to
Yes, okay, yeah, of course. So
just the question.
we are also
So
handicapped
if
to understand the
so if you
questions.
can just explain me. So basically, it's depend on the interpretation. Definitely, if you are asking me from the patenting perspective, for example, when I say a patent which is injurious to public health or morality cannot be given a patent. So suppose tomorrow I invent, this is a very example which I give in my classes also. So you devise a pickpocketing machine, or I devise a scissor which can cut a pocket in such a precision that even that person cannot know I can take its purse, or I can basically that purse can be stolen easily. So that's something which is bad actually. But if I represent that thing in my patent application, a cutting device or a efficient scissor which can be helpful for any other purpose. So it's very important how you write a patent application. Even though my intention was something bad, like, or can be used in a different way, but if I don't represent that thing upfront, or it is very difficult to understand from the patent, chances are that you might get patent. So it also depends how you are interpreting on that thing. I think this is where there's a lot of discussion. I did not getting it. Dr. Ratha, if you can just read that,
Yes,
because I
yes,
think there is a lot of
lot of questions for the prior speaker, so a little bit confusion. Let me, of course, let me start with. I am scrolling up. There are a lot of things.
Okay, that's what.
Yes, people have congratulated you for excellent deliberation, excellent talk. Of course, many people and our other speaker, Dr. Narendra Pradhan, also congratulated you because he learned a lot from the deliberation. So I believe this may get patent.
Sorry, timeline for get a patent.
For getting an Indian
Okay.
patent.
Okay, timeline for actually the patent application would be counted from the day from which you have filed the application. So just to give you a brief, so there are basically two types of application. One is known as the provisional application and other one is known as the complete application. So what happens, as I told you, sometimes even if you are not like sure and you want to publish it, and your invention may not be ready. So suppose you are working on certain product, but the product is not ready as yet, but you want to publish it at the same time, and you don't want to lose the time also, because you know the first one to file a patent will get a patent. So you have to be prompt enough. So in that case, there is an option, like you do not, even though the invention is not ready, you can just file a provisional application, and that application does not need much details. It's only that the title of the invention, and then what is the basic, you can give an abstract and some description of that. So what is the faida, what is the benefit of that thing is that you get 1 year of time period within which you can file a complete application. So once the complete application is received, then after 18 months of the first filing, that is published to the public. Okay, so that is known as the prior publication of the application. So that is to invite any opposition from any public. So if suppose in two institute, two people are working, so one may say, look, I was working first, so he may claim that you are not the true inventor, so I have a opposition, I can question your patent. So to get all those queries, application is published. If the patent office does not receive any query, then after 18 months only it can be examined. Okay, so basically for the examination, you have to file a request. So overall examination until the grant of the patent, it may take minimum 3 to 4 years, because you know from before 18 months it cannot be examined. And also if you don't file a request for examination, you have to file the request for examination at least within 4 years from the filing date. So it takes generally a long period of time. Earlier it was taking 7 to 8 years to grant a patent, but these days within 3-4 years also you can get a patent, provided your patent application is written properly. Otherwise you will get lot of response from the examination, then you have to again give the feedback, answer to the queries, so that may lengthen the time. Earlier, since the number of applications are quite high and we do not have many patent examiners, so that was taking a lot of time. But anyway, these days the patent office is working very swiftly. So 4 years, 3 to 4 years is the minimum time. Maximum such no limit, to be frank.
Okay, thank you, ma'am. Hopefully that answers the questions. So next question popping up from Dr. Satyanarayan Sahu, who is to know what are the basic requirement for the patent? What is the process,
Okay.
and what is the process?
Process is that nothing. So basically you have to understand. If you want to file a patent, so first you understand whether you are eligible for filing a patent or not. In India, anyone, means any major, not a minor, can file a patent. Any person, any company, whether it's an academic institute or an industry or any other single person, can individually or jointly can file the patent. So suppose you all are working with some organization, or I am working in IIT Kharagpur. So today if I invent something, so I am the inventor, not the patent owner, because I am working for IIT Kharagpur. So in that case, IIT Kharagpur will be the owner of the patent, or what we call in legal term is the assignee. So my name would be there as, but the patent would be filed from or in the name of the IIT Kharagpur. So if you, that depends on your employment agreement with your institute, or a student suppose working in some lab. So even that student may not directly file, he will be an inventor, but the institute in which he is working can file the patent on the behalf of the student. One difference between the research publication and the patent is that in research publication, what we can have 10-15 authors also. So the first one is the first author, and there is a corresponding author, and in between we have numbers of author. But in patent, the inventors, there is no thing called first inventor or second inventors. Whatever name which are mentioned in the patent application are equally inventors. So it is not that to please your friendship or to please your teacher, you put a name of your teacher in the patent application. You know why? Because the patent is something which you are applying for your commercial benefit. Otherwise, what's the point? Look, no, if you want to just score it for your academic performance, that is of no use. I will never suggest that you just for your academic endeavor, you go for filing. If you feel that can be commercialized, then only you should go ahead with the publication or patent thing. And you should be very careful about putting the name of the inventor. Like that inventor must have contributed to that work. Otherwise, someone can invalidate your patent. So you have to be very careful who are the true inventors and who can file the patent application. And in terms of the other requirement is that your invention must be ready. Means all the experiments, all the things which you have, which you are claiming must be ready. So that has to be written in a proper format. Okay, so in patent, it's a techno-legal document. So we have a proper format. You have to give the abstract, the figures, and you have to completely describe. So you have to describe the invention in such a manner that anyone or any person who is skilled in that, means I'm a microbiologist, so I can understand microbiology. So if I want to repeat that experiment in my lab, I would be able to do that. So that is called complete specification, detailed specification. So if it is missing that much of detailed thing, it may be rejected. And then the patent always ends in certain points which are known as claims. So basically the claims are something which defines the scope of your invention. So you have to write it properly, means what are you claiming? So if I am claiming a product, I have to describe the product in such a way that it would give me protection on each of its aspects. So the drafting of patent application is very important in that way. And for the biological thing, there are few other things which is required. Like suppose you are working on a particular strain of microorganism. If it is not commonly available, you have to deposit those microorganism. If you are, so we have depository institute like MTech Chandigarh or NCCS Pune. So you have to deposit the organism there, or if you are working on a new plant species, you have to deposit that there, and you get a accession number that you have to provide. And if you have a certain sequence work, so you have to give the whole sequence listing along with your application. Drawings are mandatory, like if you are using certain kinds of the drawing, examples of the thing. So all that has to be drafted properly. These are the written requirement, and there are few forms. Okay, so I would suggest there is a site called ipindia.nic.in. You can just visit that site. So there are a number of forms, so that has to be filled before you go for the application. But yes, as an inventor, you must be careful about the whole application, because it is no one would know the invention better than you. So that has to be written properly. I hope, like, this is a very big thing actually. So in a nutshell, I told you, it's another, like, it will take me 2 hours if I keep on explaining all the parts of the thing.
Of course, that is a big story. So hopefully Dr. Satyanarayan Sahu has got his answer. So next I have a query, means I have a little bit curiosity, how much money is required to file an Indian patent?
Okay, so look, as I said, so suppose your invention is not ready, you are filing for a provisional application. So that is like initial stage or a complete 4,000 rupees for an individual. Or for if it is a startup company, there are few exemptions, like to promote more and more patenting. So there are exemptions. And if it is a academic institution, like again 4,000 to 5,000 for filing. And but if it is an industry, it will take 8,000 rupees for filing. Okay, so there are other fees also. So there is an examination fee. If you want to have an early publication, there is a publication fee. Then there are, after the patent is granted, again there are certain charges for granting. The whole examination fee is there. And then because a patent right is valid for 20 years, so you have to pay the renewal fee for every year. So overall around, I will say that the price changes time to time, but still 30 to 40,000 would be a cost for a patent to be maintained. But having said that, this is only for the normal process. But the main crux in the patent is that how you write a patent or draft a patent. So if you are hiring a patent attorney, so that patent attorney might take you 30,000 or 40,000 rupees. So that is a big cost. But if you are sufficient enough and you can write a patent by your own, then it may be 20 to 30,000 sometimes. And only the maintenance fee is the one which should be higher. So there is a list of a list is given for the fee. I may share that link with you, so you can see how much is the total cost. And otherwise, like, it sometimes cost lakhs of rupees for the companies to file the patent. So it depends who is writing your patent, because you have to validate your patent. So that the writing of a patent is very crucial, and for that the attorney charges a very high amount. So that is the major cost in India. So
Thank you, ma'am.
Yeah.
Then the supplementary question is, what is the difference between Indian and US patent?
Okay, look, if you want a patent in India, you have to file in India. And if you want to have a patent in US, you have to file in US. So you cannot file a patent in US if you are not filing in India. At least you have to inform the controller. So difference is that in US, the patent is what you call very flexible or very linear. The criteria in India, we say the patent provisions are very hard to achieve. There are certain reasons, because as you say, plants cannot be protected here or animals cannot be protected here. In US, you can have a plant patent apart from tubers like potato and the main, because those are their staple food. Any new variety of plant can be patented there. So the condition, the grant of patents are little bit, the criteria, examination criteria are little bit different, or even sometimes very linear. Business methods. In India, we do not have patent over any business method. In US, you can have. Sometimes the computer software and associated hardware, those are not patentable in India, but you can get it. So it difference is only the examination criteria. Otherwise, it's more or less same. And the fees, obviously, very high there.
Professor Dipankar Bhattacharya has specific queries. He has put 3 queries. So the first question goes like this. So if I read it the word by word, modify a molecule. He has written if I modify a biomolecule by a functional group and use it as a drug, that is in bracket he's written no one has used it such a molecule earlier for a particular disease. So like example, if one add hydroxy group at a particular position, then to inhibit one molecule, so whether it is be patentable.
Okay. To be specific over that example, so he has suggested curcumin. Okay, so in India, one of the thing is that traditional knowledge cannot be patented. So curcumin is basically isolated from the haldi, as we all know. So first question would be, it's a general knowledge, they will say, because something you might have heard about this biopiracy, like this turmeric, neem, haldi, where other countries are trying to like claim it that it's their resource. So to circumvent that, actually this traditional knowledge is not patented in India, or not allowed as a patent. So first objection would be on the curcumin. Anyway, suppose you have now isolated it as some substance, and you are saying, for example, you are adding a hydroxy group. So in case of the chemical substances, we have the standards are different. So it's possible if you change a moiety or add a side chain, the nature of the molecule may change. So what happens when a person claims a chemical substance, he doesn't claim the chemical structure as such. So there in this, like as you suggested, a OH group, he may write a R group. The R can be a hydroxyl or a methyl or any other group. So in that case, the claim means he had already claimed all this substance. But again, you are saying that, okay, by that you are treating or treating something, or like inhibit one microbe. Yes. So you cannot claim that compound, but you can say the use of that substance to inhibit that microbe. Maybe it is, suppose you are using it as an antibiotic, for example, a new antibiotic, then the process of making that new antibiotic can be claimed rather than claiming that product. Maybe that product may be already claimed, you never know. Even though you are adding certain side group to that, so that compound may be existing or very easy to make that compound. But yes, the use of the compound to treat certain groups of the microbe, that thing can be considered as a novel, as a process. Or a product by process thing, or can be used as an antibiotic. So that may give you the edge that you can patent of that thing. But you have to, that's what I'm telling. Drafting is a very critical aspect. So if you put something upfront, curcumin or traditional knowledge, something, they would upfront reject, no, this is not, that cannot be granted patent. So you have to be very careful in writing those things. If I just take the second one, there is a known equipment or product. I used it for a particular application where it was never used earlier. Product A bed is used for water purification. Use 3D printer bed under microgravity condition. Yes, application, if it is not, maybe again you have to be very careful. Considering the you have to consider the other prior arts. So if that porous clay bed is used for 3D printing bed, yes, sufficiently different application, and if you can prove that it is having a highly industrial use, and again something very different, that can be considered. But new application, it not be very general, then it may come under the clause of section 3D and may be rejected. But the application, if it is substantially new, that have a chance of grant of a patent. Synergy in case of use of 2 or more biomolecules for a particular application. This again, synergy or the efficiency is again a debatable thing. How efficient means, again, that would be specific to the domain. So if I suppose, if I'm taking example, if a medicine was working or taking 4 hours of time period to work on a particular condition, and now I have again modified it in such a form that now it is working in half an hour, then that would give me, that may be considered as enhanced efficacy, or like the time is so short, 4 hours and 30 minutes makes a huge difference. You can save lives. But if I say 3 hours and 4 hours, then again that may be questionable. So how much difference it will make if it is working within 3 hours and working within 4 hours? So the enhanced efficacy and all these are very debatable things. Again, will be very case specific. So you have to prove it by example that it's going to be more effective in overall manner. So that would be in that case, like it's case by case. There is no, so 20% efficacy or 80% efficacy would be considered. So again, that is very specific to the technology. So the invention has to be novel to get the patent. Then how the safety pin as you use, okay. So the safety pin patent which I'm talking is around 1800 something, the time period, if I'm not wrong. So what happened? So earlier there was pin. So pin was, okay, suppose a lady is using a pin on his dupatta or sari, so that may hurt that lady. So, or if you are using for some purpose. So for the first time, the pin mechanism was devised where the pointy end was put inside a covered thing or something, so that it is not hurting. So the invention is not regarding the, we gave it the name safety pin, but it is not safety pin as such here. So, and I said the invention has to be novel, and the novelty is always judged with respect to the prior art. So when I'm talking about 1890, the patent was granted. Before that, whether the safety pin already existed or not, that was checked. I'm not, if you file for a safety pin, it will be definitely rejected if you file it for today. So it was, so the application is always judged the day on which you filed, whether something existed before that or not. So at that time, the safety pin was very, what you called new. That's why it is granted a patent. Can we patent primer sequence designed by us? Yes, you can, if it is some substantially different, like, because you know these days in the like you already have a session on the bioinformatics. So already many sequences and primers has been put forwarded. So you must have to like sequence as not as such, primer sequence along with the usage. That would be give you a higher. Otherwise, the normal gene sequence can be a part of copyright also if you are just giving the sequence. But if you are using a primer sequence and also its use, that would be patentable, maybe patentable. What kind of plant products are patentable? Yes, plant products, suppose you have isolated certain phytochemicals and then you are specifying a different use of it. So those kind, but again, that use cannot be very general. Okay, it's inhibiting certain microorganism or this or that. No, so like all these medicines, how they are getting patent over it. So these are the extract of the plant, how you have purified it, how the other technology has been using, and then you can go for that patent. What is the difference between a patent, publication, and the copyright? Okay. Okay. So patent, as I said, is given for an invention, and it is a kind of a certificate or a legal right which allows you to commercialize your invention or stop anyone from using your invention. Whereas a publication is covered by a copyright. Publication is thing which we have done something, we are writing, and once you have given written something, it is your copyright. But when you are publishing it, so maybe you are giving your copyright to the Elsevier or any other Springer or someone who is publishing, the publisher house who is publishing on your behalf, so that all the benefits he gets. And apart, but when you have a copyright, like you are allowing everyone to read your work or use their thing. Suppose I can quote you or cite you, but when you are a patent, no one can use it, basically use in the form commercial form. Plus, yes, there are research exemptions. So patent is more about the from the commercial point of view, when you think that there are certain economic benefits which you can protect. That is through patent. Copyright is for the literary work. You cannot get a copyright for a technique which you have invented, but yes, when the technique is written in the form of a paper, that is protected by the copyright. Yes, there is a government website. So as I said, so Indian Patent Office website is there. You can just visit www.ipindia.nic.in. All the steps you can get there. What do license patent means and why it is required? Okay, licensing means you have a technology. So suppose I am telling that CRISPR technology is a gene editing technology. So someone you want to use that CRISPR technology for treating beta thalassemia. So like the scientists who have developed that technology, they are not directly working. A company is working. So basically they are licensing the technology from you so for that license, they are giving you some amount of money. So that is the thing they, so you can get the economic benefit that is known as licensing of the technology. Means otherwise you can sue them legally, file a case against them that they are using your patent and they're not taking permission from you. And that's why you will claim, like in US, that's why everyone is going for patent, because you can claim millions of dollars for your technology. So like BT Cotton or Monsanto, and why these big companies are so much about the patent, that is the reason, because you can earn money from that. Thank you, Dr. Narendra. Thank you, Rukmini. Okay, and what is this? Avinash. I hope I have given. When researcher have a publication, who has the copyright? It is the authors of the journal. Yeah. So when you have a publication, basically, if you have like all of you have published, so you may give the you already these days is Elsevier or other things, you write a consent form or copyright form. So you give your copyright to the publisher. But what is the beauty of the copyright is that as an author, you have certain authorial rights. So means the moral right or the personal right with respect to your work always remains with you. So as an author, as and when we want to modify your work, you can do that. And the moral rights are that if someone distorts your work, suppose suppose this and the copyright subsists for a long period of time, depends, like after the death also 70 years or 80 years, whole lifetime of the author, plus after death few years. For example, Gitanjali. Okay, so now it is open to public. So Rabindranath Tagore has written Gitanjali. So if he were alive and someone is trying to distort his work or putting certain like you can say anything bad words or something adding, so he may raise objection. So the copyright gives you that moral right that you can protect the distortion of your work, and as an author you have the very right to use that your work or share your work for academic purposes and other things. So that is important. But yes, these days, this copyright is very important, because Elsevier and other, they charge high price for that. And it's when someone is publishing in the ResearchGate, if you have an account in ResearchGate, before you want to make a publication open, they always ask you whether you have the right to make it open access or not. So maybe, so you have to be very careful with whom you have published. So whether they allow you to publish it openly or not in such forum. Or else, if you want to share your article, you can directly give that person if someone asks you. So that can be done. That is no problem. But if you just put it on a public website, so that might be a problem in some cases. Thank you, Professor Lagacity. For filing a patent, anyone can file or only patent agent? No, anyone. As an individual, you can file. So there are patent agents who are the facilitators. But so for example, an inventor might be a poor farmer also, who is not that much of literate. So in that case, patent agents are there who can help them in filing. So that's the role of a patent agent. Thank you, Dr. Sir. Thank you, Avinash. Again, Dr. Narendra, yeah, that's very interesting. As you said, Uppsala University, in their patent goes to the researcher, not to the university. Yes, it depends upon every institute have their own IP policy. So if you will see the bigger institute like UC Berkeley or whatever institute in US, so they always have a spin-off company from their thing, so that whatever technology is developed, so they may give the technology to that company. In India also, it depends upon the IP policy of the institute. Even though IIT Kharagpur is the assignee, I will take their name, but maybe possible he will give that patent to me. Okay, you can use your patent as you want, or they may ask certain royalty in some cases. Yes, it depends upon the IP policy of the institute. I think, thank you,
Yes.
Dr. Lagassetti. Yeah, I think that's solves your query.
Yes.
Okay, not a problem. My email ID is with Dr. Ratho also. Please, if you have any doubt, any query, you can write to me also. I would try to answer your queries over there also. So thank you all of you for having me with us, and I have passed the time long by. So thank you so much.
Thank you, Dr. Niharika Bhattacharya, for a nice session. And just a last query, if you allow me.
Sure.
Can patent be reverted back if someone learned that whatever he has patented is no more holds true? Could you follow the question?
Sorry, if you can just repeat.
Can patent be reverted back if
Okay.
someone learned that whatever he has patented no more holds true?
Okay. So yes. So suppose the patent is already granted. So there are two ways. So you see, after the patent has been granted, you feel that your patent is not that important or it is not that good. So what you do, you don't pay the fee. So that is like, if you don't pay the fee, renewal fee, the patent will be dead. That is one way. If you feel that that is because not very useful, but you feel think that something wrong has been claimed in the patent, which is really can be disputable, you want that, yes, you can write to the Controller General of the head of the patent office saying that I don't this patent has been wrongfully awarded to me, or this is has certain grounds which should not be, so I want to revoke this patent. So that is called as revocation of the patent. So the patent may be cancelled at that point of time, or if it not published or not examined as it. So anytime before the grant of the patent, you may ask the patent office or not to examine it or not to publish it. So that provisions are there.
Thank you. Thank you again. So we had a very fruitful session. Thanks goes to both the speaker as well as the chair for holding a very nice session, of course. We, on behalf of the organizing committee, we heartfully thank and acknowledge the effort made by Dr. Niharika Bhattacharya, Dr. Ranjan Kumar Nanda, for making things work seamlessly. Thanks again, again also we thank Professor Ashwini Kumar Rath for chairing the session and working things correctly. Okay, so hopefully everybody has enjoyed the session. Now it is a high time to have a lunch. I think we should break, and I request the participant to join back at 3 o'clock. So, and if possible, Niharika ma'am also join.
Sure, yeah. Yeah, thank you so much. Thank you, Chair, and thank you, Professor Dr. Ratha, and all the organizing committee members. It was very nice to be here. Thank you so much. Yeah.
Thanks a lot. Then we can break.
Sure, yeah, thank you. Bye.
Good afternoon.
Welcome back to the the culminating session of this international e-conference. After a happy launch, we'll enjoy another group of avid researcher. So the session has two eminent speaker, rather I should say entrepreneur, Dr. Sukanta Bhattacharya and Dr. Narendra Pradhan. And the session will be chaired by Professor Dipankar Bhattacharya, sir. So let me introduce about the chair. Professor Dipankar Bhattacharya, the mentor for School of Applied Sciences, Centurion University, and manager of Gram Tarang, the director of Gram Tarang. So Professor Dipankar Bhattacharya had his PhD on biosensors from Jadavpur University. Then he went on to do his postdoc at University of Quebec, Canada, followed by joining there as a research professor. His research area, particularly in Canada, he was focusing on machine learning and chemical engineering. He has worked with many renowned aluminum companies such as Aluminary Alloy of Canada, then Hydro Aluminium for Norway, Rio Tinto from the European country France, and many more. Then he thought serving the motherland, took the flight to India and joined, later on he joined with us in developing the Centurion University of Technology and Management, Odisha. He was working as Dean of School of Applied Sciences just a couple of months back, and then he elevated to be the director of Gram Tarang. And now he's working as mentor for and technical director of Gram Tarang, and he's also working as mentor guiding the School of Applied Sciences. He has around 200 publications to his credit in many peer-reviewed journals, and again, he has 5 patents, including 1 US patent. He has students from around 20 countries. He has worked, student from 20 countries he has guided. So welcome you, sir, to chair the session. Welcome you, Professor Dipankar Bhattacharya, sir.
Thank you, Dr. Rath.
So next, it's time to introduce our next eminent speaker, Dr. Sukanta Bhattacharya, Sukanta Sekhar Bhattacharya. So Dr. Bhattacharya had his master's from School of Life Sciences, Sambalpur University. Then he joined the PhD program of the Premier Institute IIT Kharagpur. After finishing his PhD, he had his first postdoc in Cincinnati, US, where he focused on microbial biotechnology work and microbiomes. And during his PhD time, he was working in microbial genomics. And his research areas and academic interest broadly focus on mostly in applied area. That's what we thought of roping in and asking him to join as a speaker, because we are School of Applied Sciences. So his focused area is on applied area of microbial and environmental biotechnology, particularly enzyme technology, with the focus being on the xenobiotics, metabolism enzymes. Then, of course, microbiomes and looking at the toxicity study. Then he has worked in the at the interface of applied microbiology and toxicology, and explored the potentials of microorganisms both in the environment and microbiomes to affect the biotransformation of xenobiotic chemicals. So presently he is the CEO and director for Lignin Biosciences private company. He has started up, and he's working for furthering its work. So may I request Dr. Dipankar Bhattacharya to chair the session, and now the platform belongs to you, sir.
Thank you, Dr. Rath. So now I welcome Dr. Bhattacharya to present his work on microbiomes, mega modulators in health maintenance and restoration. Please, sir.
Thank you, Professor Bhattacharya and Professor Rath, for the kind introduction. So today I would be talking on this upcoming area of research: microbiomes, mega modulators in health maintenance and restoration.
Excuse me, Dr. Bhattacharya. Can you make
Yes.
a slideshow so that it will be visible to all?
Yeah.
Press F5.
In presentation mode or F5 in the lab.
Okay. Got it. Yeah. Is it visible now?
Yeah.
Yeah.
Okay. Thank you. Thank you so much. Yeah, so today I would be talking on microbiomes, mega modulators in health maintenance and restoration. So now when we talk about the microbiomes, a very similar term comes up, that is a human genome. So when we talk about this human genome project, so that caused a lot of a lot of enthusiasm amongst the researchers, and a lot of possibilities were thrown up. But now just to so in the screen you can see this famous scene. This is from the movie Bahubali, a very popular movie last year, and a very interesting one too. So in this picture, it very rightly summarizes the relationship between the human genome and the microbiome. So if we see that this is the human genome, and we were very excited about when the Human Genome Project was completed. But when we explored further, we came up upon we came up with the existence of another possibility with the discovery of microbiome. That is it is a collection of the microbial cells and the microorganisms within the human body, which actually may play a very vital role in the health and disease. So when we look at, giving the outline of my talk today, so we would talk about microbiome, that is the definition and where do we find it, how do we study the microbiomes, how these microbiomes, they impact us, and how the microbiomes themselves may be impacted. We will discuss it with some case studies. That is, we would discuss it with some good case studies of some other groups, and followed by some case studies and the data that was available with us that we carried out. And we will also discuss about the constraints in this microbiome study. So now when we talk about this microbiome, so as we started during the discussion, that is this microbiome the second human genome. So when we talk about this microbiome, this term microbiome which is there, that was coined by Joshua Lederberg to signify the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space and have been all but ignored as determinants of health and disease. So now, these microorganisms which are there, so these microorganisms virtually they are present in many parts of our body, and they are actually they interact with us in more ways than we can even imagine till now. So when we look at this microbiome where they are present, we see that they can be present in the gut. Gut, obviously, this is one of the most well explored area where we about the microbiome, because most of the research groups which are there, so their focus is on gut microbiome. There are already around 500 to 1,000 bacterial species which colonize this gut, and now they have been already, it has been proven that they are playing a very critical role in the human health and disease. Then these microbiomes which are present, they are also present in the oral cavity, of course, and our focus and the focus of my research has been on oral cavity, and then they are also present in this in the lungs. Lungs, previously it was thought that lungs is a very sterile environment, but that was a myth. We would discuss how why it was thought to be sterile and how this was, and how it was found that that was a myth. And this microbiome, it is also found on the skin and urogenital tract. So now when we say that these microbiomes, they're present in these different parts of the body, it is more often than not, these microbiomes which are present in different parts of the body, they interact with each other. For example, the gut microbiome, the oral microbiome, the lungs microbiome, all of these, the skin, the urogenital tract, all of them they work in synchrony. And many a times in a disease, we find that not only one, but many or all of these microbial communities on the human body, they're affected, and they form a very distinct pattern. Now, when we look at this microbiome characterization, or how do we study the microbiomes? So there are two ways in which we can do it. One is, of course, the culturing, which which all the people in this forum, I believe, are well aware about. So this is a very, and the next is sequencing. So coming to this culturing, this culturing is an old school strategy. That is, we culture the microorganisms on different kinds of media, and then we go in for characterization. But the problem with this technique is that that most of the microorganisms which are present in the human body are non-cultivable. And then this culturing, it's also time consuming, because you have to go through different media and then see that which microorganism can grow, grow in which media. The compatible media has to be figured out. But the advantage of this culturing technique is it helps in proper characterization and also in the simulations in vitro. The second technique, or the new school strategy, or the more modern technique which is there, is a sequencing. This technique is, of course, it is rapid, it is accurate. It is accurate because here we do not depend upon that upon the cultivability of the microorganisms which are present. We just sequence the entire microbial community which is there, and then resulting that it captures most of the microorganisms which are present. But on the flip side, it is expensive. You need specialized equipments for that. It is a destructive analysis, because once you get the microorganisms, you extract the DNA, you are not culturing them. So this is a destructive analysis, and as it is a destructive analysis, in vitro simulations are not possible. So when we are unable to culture them or cultivate them, we cannot go in for this in vitro studies, or for these studies. So we have to. So in this case, we have to depend on more on the samples that we obtain from the nature or from the people, rather than simulating particular microorganisms in animal models or like. So now when we look at so this is the overview of the 16S rDNA based sequencing approach. Here I call it rDNA because because this rRNA is a very unstable molecule. So when we are going in for the sequencing, we select this rDNA instead of the rRNA. So what we basically do here is the entire microbial community, it is first collected. That is, it may be a swab, it may be a rinse, or things like that. So we get this community sample which are there. Then we go in for a total DNA extraction. So now from this total DNA extraction, what we do is we go in for an amplification of the 16S rDNA region of the total DNA which is there. Then we go in for sequencing of this of the 16S rDNA which we amplified, and after that what we go is we classify this operational taxonomic units as we call them, these sequences which are there via the database. That is, the bioinformatics tools are used here. So there are different databases which are available for this microbial for this microbial sequencing. That is, the NCBI database is there, Silva database is Silva Green Genes database is there. So we classify the OTUs, and then we look at the OTU abundance and the species interactions if they are there. So this is the way that we move forward with this 16S rDNA based sequencing. So now when we what we will do, so now when we look at the different microbiomes which are present, so first we cannot move forward without talking about the human gut microbiome, because it's one of the most well studied microbiomes. So here, as we were discussing previously, that around 500 to 1,000 species are present, and these are already known to closely interact with the host cells which are there. So these also regulate the immune system, and that's why a lot of interest is being generated in this microbiome even in this post-COVID era. So and these microbiomes, they are actually so these microbiomes, they can affect the human health, and these are affected by the human health. So we can use them for diagnosis, or even we may use it for therapeutic intervention. So here, as we mentioned, that it directly impacts the health and metabolism, and it is known to be affected by diet and other stress or health conditions. Then when we talk about the microbiome of the lungs, as we were discussing again, like the sterile environment in the lungs is a myth, and this and the airways which are there, this is colonized by different phyla which are there, like the proteobacteria, the firmicutes, bacteroids. They form the major the major phyla which colonize the lungs. But however, this initial source of this airway microbiome we do not know still now. So research is going on that. So and what is evident is that this airway microorganisms, so these are very adverse. These are affected by the health conditions of the individuals. Like in the COPD patients, the microbiota of this of the different regions of the lungs, it is very distinct from that of the control from the control group or the healthy individuals. Then this oral human microbiome. This has been my area of interest. So this is another place where we find a lot of microbial species. So we have greater than 600 taxa which are there. Among this, only 24% have been named. 8% they have been cultivated, but they are unnamed, and 68% of them are uncultivated phylotypes. So there is a lot of scope in this area, and a lot of things that has to be explored. So these 13 phyla which are there, they're very abundant, in this human microbiome, which is in the human oral microbiome. Now, when we talk about this microbiomes, so actually the human body, so the human body and its microbiome, actually they operate in sync with each other. Like they are more of a superorganism. Like what happens is that the condition of the human health, it determines the microorganisms, and the microorganisms which are present, they determine the human health. So it's always a crosstalk between them. So when we look at this has been seen in the gut, where the commensal microbiota which is there, they facilitate a breakdown of the food. They contribute towards the development of the immune system. They even modulate the immune system. Some case studies, they have shown that this inflammatory bowel disease is associated with the microbiome dysbiosis or a change in the microbiome. Then it is it is very critical so this microbiome, it is very critical in the maintenance of this gut immune homeostasis. So, but till now, as a lot of study has already been carried out in the gut, so there are evidences which point to this developing and maintaining, this type of interactions which are going on between these microbiomes and the human health and disease. So these type of interactions, they're very evident in the gut, but such direct links, they are absent in the lungs microbiome. But of course, as we were discussing in the previous in the previous slide, that an association is observed between the microbiome and the lung diseases such as COPD or COPD and even in certain cases of lung certain cases of cancer. So those kind of things, they are evident. Now this is a slide where we see that the shaping of the pulmonary immune response by the commensal microbiota. So here what we see is that it can be done in two ways. One, of course, it can be a direct response to the microbial cells, and the other can be a response to the microbial metabolites. That the pulmonary immune response can come up. So here there are a few case studies in which where we talk about this environmental, toxic, and microbiome interactions. So these are the work done by some other groups. So I've just put forward some of them just as an example. So first of all, before going into that, when we talk about the xenobiotics, because the term that we come up frequently in these studies are xenobiotics. So just to mention that these xenobiotics, these are the chemicals which are not present in the natural environment, but they're released in the environment due to anthropogenic activities or the human activities. So the common xenobiotics that we are exposed to, they are the polycyclic aromatic hydrocarbons, the chlorophenols, the dioxins, heavy metals, and pesticides. So many of these are present in the environment, but in a very low quantity, such as PAHs, they are present, heavy metals, they are present, but they're present in very less quantities in the environment. And this human exposure is increased because of the human activities itself. So there are a host of other xenobiotics also, like which we of course come across, like the polythenes and those kind of things. So those are also xenobiotics. So our focus today is on these xenobiotics to which we are exposed during breathing, or the xenobiotics to which our respiratory system is exposed. So here we have here we have a case study on this particulate matter 10. So here so in this case study, actually so in this case study, it has been observed that this particulate matter 10, when it is exposed, so to this mice model, so it actually it alters the inflammatory response in the gut, and also it alters the gut microbiome. This PM10 may have a very high toxicological load and may comprise of this elemental carbon, heavy hydrocarbons, and a large proportion of this pHs found in the fossil fuels and the dehydrated sulfuric acid derived from the from the fuel sulfur. It is this is mainly because of this diesel exhaust particles that these are released. So now when this PM10 exposure, when we look at this wild mice, so we can see that so it triggers a different immune response in the control, as well as the PM in the PM10 exposed mice. We can definitely see a ramped up immune response coming up here, and so it was both in a short term as well as in a long term exposure. We see a pro-inflammatory response coming up. And then when we look at this microbial community, we find that a big shift in the microbial community that occurs as compared to the wild type, to the wild type plus which are exposed to the particulate matter 10. The IL-10 knockouts, or the IL-10 is an anti-inflammatory cytokine. So in an IL-10 knockout, that means the model which will be a pro-inflammatory model. We see that and then this particulate matter 10 plus IL-10, we see the when this IL-10 is exposed to this particulate matter 10, we see the shift in the microbiomes that is happening. So this is a microbial composition in the wild type and the IL-10 at day 0 and day 35. And similarly, we have here a case study with the arsenic. So in this arsenic, actually, a lot of people, they are exposed to this arsenic. Even in India, we find that a lot of people in many mega cities, like even in Kolkata, they are exposed to arsenic. So and when we take it in figures, we see that approximately 2% of the human population, or 160 million people, they are exposed to arsenic above the limits that is set by the EPA, or the Environmental Protection Agency. So this arsenic, it is responsible for a number of diseased condition in human beings. And so now when we look at this bacterial community in the sputum samples, we see that there is a shift in the bacterial community when we look at this moderate exposure, as well as and compare it with the low exposure of arsenic. So this is the different microbial communities which were studied and which were found to be affected in this study. So when we look at taken together in a nutshell, when we look at this study, so we see that the Straxa which are prevalent in this moderate exposure groups, they're Firmicutes, Chamelia, Lactobacillus, Streptococcus, Neisseria, and Pasteurellacea. The taxa, those are prevalent in the low exposure group. They are Rothia, Prevotella, Prevotellaceae, Fusarium, Fusobacterium, and Neisseriaceae. So what can happen here is we can use even this microbiome as a marker of the exposure, that whether the person has been exposed to such toxicants or not. We can look at the microbiomes, and we can look at the shift of the microbiomes, and maybe that we can come up with some conclusions. And also we may use the microbiomes, like we can alter the microbiomes. We can attempt at doing so to bring back the healthy microflora, or the healthy microbial composition as a part of therapeutic intervention. So this is another case study that is with alcohol and tobacco. I'll just quickly skip over this one, and I'll come to our own case study, that is the microbiomes with head and neck cancer. So this study was carried out by us at University of Cincinnati. So coming to this head and neck cancer, or head and neck squamous cell carcinoma as it is called. So what happens is this head and neck cancer is a major health concern. So approximately 60,000 new cases of head and neck cancer or head and neck squamous cell carcinoma, it is diagnosed each year, and the mortality rate of this head and neck squamous cell carcinoma, or HNSCC, is approximately 60% within 5 years of its inception or detection. And the worst part of it is even if people they survive this head and neck squamous cell carcinoma, the quality of life is drastically reduced. Approximately $3.6 billion is spent in the United States alone for the treatment and the medical interventions. This head and neck squamous cell carcinoma, or HNSCC, it is very strongly associated with tobacco or alcohol consumption, and it is also associated with human papillomavirus or HPV infections. But that is approximately just 25% of these cases. So now, when we look at this, at this head and neck cancer, squamous cell carcinoma, so our focus is not on the HPV cases or human papillomavirus cases. Our focus, because human papillomavirus in head and neck cancer has been fairly well studied as compared to the head and neck squamous cell carcinoma or HNSCC cases which do not have HPV. So now when we look at this HNSCC, we see that there are several cases of HPV negative or human papillomavirus negative cases. So these people, as we were talking in this previous slide, so here in what we see is that, in the previous slide we said that that this it is strongly associated with tobacco and alcohol, HNSCC, and it is also associated with HPV cases. But now when we look at several cases which come up in the clinical studies, what happens is that many of these patients who have this HNSCC, they are non-smokers and they are non-drinkers, and they're also HPV negative. So this led us to believe that here, apart from this HPV, and the tobacco smoke and the alcohol, there may be other abiotic and the biotic factors too. Now this pH which is there, this has already been known to play a role on the onset of several cancers, and that is including head and neck cancer. Lung cancer, of course, we know, but head and neck cancer also, we know that the pHs have a very critical role. Then also we also found that an altered microbiome was also observed in smokers, and the second thing and the other thing that was there was an altered microbiome was also found in people with periodontal diseases or it was also observed in people with poor microbial with poor oral hygiene. And it was found that people with poor oral hygiene, they have more tendency to have head and neck squamous cell carcinoma as compared to people with good oral hygiene. So when we take all this information together, that is the presence of the HPV negative cases, this non-smokers, non-drinkers, we see this case with poor oral hygiene, as well as taking all this together, we may come to a conclusion that microbiomes may have a very critical role to play. So now when we look at so our study, basically it revolves around the crosstalk of the pHs and the them as a marker, or whether we can go in for a therapy, we can use them for therapeutic intervention. So our whole study, which is there, so it revolves around this. So now this pHs, when we look at it, so we find that this pHs which are there, they are ubiquitous pollutant, or they are present everywhere. They are present in the tobacco smoke, as well as in the environmental tobacco smoke exposure. We can be exposed to that. We are exposed to the fossil fuels. They are found there. In the smoked foods, we can find them, like in the barbecues. So we can find them in the household heaters. And there can be other sources which are less common, but of course they do occur. And when they occur, they can be very disastrous, such as oil spills, etc. We can find them. So now the question which is there is that, which we asked ourselves was that, is there a crosstalk between all these things? That is there a crosstalk between the pH exposure, the head and neck squamous cell carcinoma, and the oral microbiome? So to answer this question, what we did is we created a study design. So in this study design, what happened is what we considered was the 6 groups which are there. So this comprised of three control group, that is who did not have head and neck squamous cell carcinoma, and 3 patient groups who had squamous cell carcinoma. So in the control groups, we included people who do not have head and neck squamous cell carcinoma, HNSCC, who are non-smokers. Then we had these healthy smokers who were there, and the healthy former smokers. So these 3 groups, they did not have HNSCC. Then we had these group, the other group which was there, that is the patients group. So here we had 3 subsets, that is the non-smokers, the smokers, and the former smokers. So here, so in the 6 groups, what we did is we sampled the oral cavity in the retromolar region near the parotid gland, that is the back of the oral cavity, using the sponges. Now this was the experimental flow sheet that we went ahead with. So we had the 6 study cohorts, or the groups, which we mentioned previously. So what we did was we took the oral sponges to collect the microbiome, and then we extracted these bacterial species or the microbial species from there. Here, of course, to mention here that this when we talk about this microbiome, this includes all the 3 parts, that is, the bacteria, the fungi, and the viruses. But here our focus has been on the bacterial community itself, community only. We have not considered here the fungal or the viral community. So here what we do is we took the oral sponges, and then we extracted the bacterial community which was there, which was loaded on this on these sponges, and then we went ahead with this microbial DNA isolation. Once this DNA was isolated from these cells, we amplified the 16S rDNA, or the 16S rRNA gene. We went ahead with this application, and then we went for sequencing using the MiSeq platform, and then the analysis was done in Loney processing environment. We got the OTUs, and then we went ahead and analyzed it with the software, that is the Mothur software we had used at that time. And then we went ahead with the identification of the pHs and HNSCC specific microbiome. So in this microbiome analysis, so this basically we discussed this in the previous slide. So here this a bit more in details, that is, after the DNA extraction, we had this so this V3 and V4 regions were targeted with the barcode based amplification, and then this bioinformatic analysis was carried out. So now this is the results that we got, and here we see that so here we have this pie chart on the unexposed, that is the non-smokers, the pH exposed, that is the smokers, and the pH exposed, that is the former smokers. So here we see that these groups, they're altered significantly in the pH exposed to the smokers, as when we compare it with the non-smokers. So that group is upregulated, and then we see downregulation of these groups in the non-smokers as compared to the smokers. And then we see that these groups, that they are a bit altered in the former smokers. Then this chart shows the patient versus control. So here you can see that we have this control, that is a non-smoker, the HNSCC patient who is a non-smoker, and we have this former smoker, and we have a control who is former smoker, and we have a control who is a smoker, and we compare it with the HNSCC patient who is a non-smoker. So here also we see an alteration in the Pasteurellacea and the Veillonellacea. And then we see the downregulation of these groups. And what we see is this Prevotellacea, which is there, that is in the smokers, it is upregulated as compared to the other groups. Then this is the chart which shows all these three groups which are there, the three control groups being compared with the HNSCC patient who is a former smoker. And here we see that this Micrococci and the Pasteurellacea, Veillonellacea, Prevotellacea, they are being altered again. Then we have this we have these three control groups as compared to the HNSCC patient who is a smoker, and we see again the alteration in the groups which have been highlighted. So to sum up this one, so what we see that there is a comparative decrease in the abundance of the Streptococcus which is observed in the patients. Then these species which are there, that is Pasteurellacea and Prevotellacea, it actually increases in the smokers. The microbiome which is there, that is partly restored in the former smokers. And in addition to, so here also here we see that here what we have considered is that the exposure to the external environment, that is to the pHs, because when we consider the cigarette smoke, we consider it as an exposure to the pHs. So in addition to this factor, to this external factor, so we cannot rule out the interaction of the different the interaction between the different microbial communities which are there. So which may also be influencing the relative abundance, because there are a lot of interactions that goes on between the microbial communities as well. So this is our second. This is our second study which we carried out. That is, this was based on a professional exposure, that is this occupational exposure or microbiome perturbation in firefighters. So when we consider the firefighting, so this is a profession with very high inherent risks, because here during the firefighting operations, the firefighters, they're exposed to the smoke, which have many carcinogenic and pro-inflammatory chemicals such as PHs and particulates. So many of these chemicals which are there, so they have been linked with many symptoms, such as including nasal, oropharyngeal, and symptoms in the larynx region, and systematic pathologies. And in the firefighters, it has been seen that that there are many diseases such as this cardiovascular, hypertension, and other respiratory diseases which are more prevalent in the firefighter population as compared to the control population. And many of these symptoms which are there, which exist in this firefighting population, they are also seen in the smokers. People people with periodontal diseases. So with dental diseases such as periodontitis, etc. So in this study, our hypothesis was the firefighters frequently inhale smoke constituents like PHs and other carcinogenic toxicants in this firefighting operations. So this exposure which is there, so it may alter their microbiome, which may be similar to that of the tobacco smokers. So our hypothesis was occupational smoke exposure in firefighters alters oral microbiome, which may lead to oral-respiratory diseases, arguably via its modifier role in toxicant metabolism. So this was our hypothesis. Based on this hypothesis, we carried out the study. So our specific aim here was characterization of the oral microbiome in smoke-exposed versus unexposed individuals, and a comparison of microbiome structure in smokers versus in firefighters. So in this study design, we had this control. We had the controls who are non-smokers. We had the smokers, former smokers, and the firefighters. And here our sampling mode was oral rinse. So here again, this was the experimental flow sheet, which was which is basically similar to our previous flow sheet, but here we have included other group, that is the firefighters, and we are comparing it with the control individuals or the healthy individuals who are smokers, former smokers, and the non-smokers. Again, here we take the oral samples, we go in for the microbial DNA isolation, we amplify the 16S rDNA or the 16S rRNA gene, and we sequence it on Illumina MiSeq platform. After that, and here we also carried out another in-house analysis using the discontinuous gradient gel electrophoresis to look at the different to look at the different at the change in the microbial communities which may be happening means overall, and then and then after we go in for the next gen sequencing, we analyze the OTUs which we got, or the operational taxonomic units of the microbial community, and then we look at the relative abundance of the different different phyla of microorganisms which are there. So here, so this was the finding that we got here with. So here we have considered 12 firefighters and 3 control. So three controls. So this was a preliminary study which was carried out, and this is the heat map where we see that the relative abundance, the increase in abundance as well as decrease in many species as between the firefighters and the control subjects. So this is so this plot explains the summarize. So this summarizes the findings which were there. So here we can see that the OTUs or the microbial communities which showed a big difference between the firefighters and the control subjects. And then when we look at the evenness and the diversity indices as an indicator between the firefighters and the controls, so we see that that in this firefighters there is the evenness index increases and the diversity index actually decreases. So these are so here this is so here we see the change which is happening, that is when we can, so this is based on the questionnaire that we had put forward to the firefighters. So we see that it is altered with the firefighting experience, firefighting events, the pHs, the food pH intake, as well as the tobacco use between the firefighters, because this was not a very controlled study. We went for the sample. We went for sampling we went for sampling of random firefighters who were there, and based on the questionnaire we got this result. So to summarize the total findings of what we obtained from this firefighter study, we saw that this firefighters they show an overall decrease in the microbial diversity, and they also show a altered microbial composition as compared to the control population. Now, the firefighting exercise was also marked with a significant increase in the proportion of the bacterial family of Streptococci, and a decrease in Campylobacteriaceae, Wickeslasii, Burkholdeриaceae, Cardiobacteriaceae families. So there was a decreased diversity and an increased evenness in the oral microbiome in the firefighters. Then there were perturbations or changes which were observed in the Peptostreptococci family, which was linked with the length and the magnitude of the firefighting experience. That is, the number of years that the firefighters have been engaged in this profession. Then, of course, the food habits, it showed that that it causes alteration in many bacterial families. When we go in for when we conclude these studies, so what we came up with these conclusions where this exposure to the pHs can result in an altered microbiome. So this was more than evident. Then an altered microbiome was observed in the head and neck cancer patients with Pasteurellacea and Prevotellacea showing relative abundance with respect to the controls. And of course, this also leads us to believe that an altered microbiome can affect pH metabolism in the human body, thereby playing a role in the health and disease. So now, with all these studies being carried out, we were faced with many limitations as well as knowledge gaps. So first was this alteration or are these microbiomes which are there, are they just the cause or they are the effect of that? Are they the effect of the exposure, or they can cause this or the effect of the exposure or the diseased condition, or are they the cause of the diseased condition? So this was one of the big questions that we are faced with. Then there is another limitation that is there with the human subjects is a low sample size. Then in these studies there was there was an issue of lack of animal models to simulate similar conditions, because with human beings you cannot simulate. So or you cannot make them voluntarily exposed to the toxicants or the carcinogens. So here we need animal models to study that. So those evidences were lacking in this study. So those are in progress now. And then this entire study which is there, so definitely these are expensive studies. And then there are limitations in the in vitro cultures, because most of this microbial community which are there, so they cannot be cultured on the Petri plates. So it was not possible. So it has not been possible to increase the concentration or the alter the microbiome voluntarily using this target microbial communities and see the effect. So with this, all these things, actually so these are pointers actually that that these microbiomes, they can play a role in our well-being, or means there is a crosstalk between the microbiomes and the health conditions of the human beings. So now the other thing that comes up is the microbiome in the therapeutics. So now when we look at this microbiome in the therapeutics, so we are actually we are all aware of the term that is probiotics. So this so using probiotics has been it has been used since ages now, but the term and the scientific explanation may be relatively new, but we have been using it for a long means since ages we have been using that. Like we have been using curd, yogurt, so those kind of food we have been using. So those are all probiotics. So probiotics are the live microorganisms that are considered to have beneficial effect on the human health by changing the composition of the intestinal microbiota, thereby contributing to the intestinal microbial balance. So the common examples of this are the lactobacillus and the bifidobacterium. But however, like the intake of the probiotics, how it, it crosses the gastrointestinal tract, and so those are yet, those are yet areas of research. So those things are those things are still now not clear, but still then, the beneficial effect of these bacteria, they have been well recognized. Then we have the prebiotics, or the prebiotics are the non-digestible food ingredients that selectively stimulate the growth and activity of one or limited amount of beneficial bacteria in the colon. So examples are germinated barley foodstuffs and the mushrooms. Then we have the symbiotics. So this symbiotics is actually, a combination of both the strategies of the probiotics as well as of the prebiotics. For example, here we have put forward an example that is, a combination of lactobacillus casei and dextran was found to be beneficial in allergic rhinitis in humans as well as in allergic diseases in mouse models. So those type of strategies, they are being carried out now. So this is a very upcoming field, and a lot of work has to be done. So a lot of work is ongoing, and right now, like the microbiomes, it is a talk of the day, and this is only one aspect that we are covering, but there are several other aspects in which the microbial studies, they are going on. The studies on microbiomes, even in the plants, the crops. So everywhere the microbiome studies are going on. So this is just a way forward. So these are the preliminary findings which are there, and the ongoing work is going on. And of course, then there is microbial transplantation, which is there. So here in this case, here we go on for direct microbial transplantation, such as the fecal microbiota transplantation. It has been used for the past 50 years. And this can also reverse the effect of severe ulcerative colitis in antibiotic treated patients. So this is a very good, this transplant has been going on for quite some time now. So these are the different strategies, which have been employed for this microbiome restoration, as well as microbiome alteration from disease to healthy. And so taken together, what we can say is this microbiomes, they can play both as a role, both as a role in the diagnosis as well as in the therapeutic interventions. So now it is open for discussion, and I would be happy to answer any questions that may come up.
Thank you, Dr. Bhattacharya. It was really a nice presentation, and we came up with a lot of ideas actually from your presentation. I would like to discuss with you in detail after I address the questions here. There are a couple of questions. So the first question is from Jyoti Prakash Roth. What is the prevalent microbiome in smokers, and whether Indian cohorts were taken into consideration, and if any genomic similarities exist between the two candidate microbes?
It's a very good question. First of all, the straightforward answer about taking the different races into consideration is no. They were all American. This was preliminary. This was an American population which was participating in the study. So the Indian cohort has not been taken into consideration. But yes, like the Indian cohort may display a very different microbiome as compared to the American cohort, because because depending on the environmental conditions, the food habits, the health conditions, the Indian microbiome can be significantly, it can be different. Of course, there the Prevotellacea and Pasteurellacea, they did show abundance there in the smokers, But when we consider the different populations which are there, these microbiomes may show, these OTUs, they can, or these microbes, they may show abundance, but at the same time, the total so the total microbiome community, which was the total microbiome or the microbial community abundance, which is there, that can be very different between the different populations.
Right. And he has another question that is logical actually. So how you select the control, as now all are exposed to smoke?
This study actually, yeah, that's very true and very relevant now. But here, this study which was conducted in US, and here what was happening, and in US what cities, what happens is that the place from which this was conducted, there the PM 2.5, the PM 10, and the environmental pH was very low. So so that was one of the factors which was there. So and the second thing which was there is what we did was we had a questionnaire with us. So in this questionnaire, actually we had a variety of questions. Like we had the questions on like it may not be always definitely like the question is very relevant. Like it may not be always the pHs that is present in the air only. It can be pHs present elsewhere. So in the questionnaire, actually we tried to we tried to map very thoroughly the exposure. That is, we looked at the food habit. That is, whether the person was taking the barbecued food or food which had pHs. We looked at the shampoo that he was using, whether he was using the tar-based shampoo, and a number of those factors were considered to see that the person whom we are referring to as control has lower exposure to the pHs as compared to the person who we are designating as a smoker or a pH exposed person. So that we try to take into consideration. But of course, these are human subjects, so absolute control may not always be possible. That's very true.
And he has another question. As we are now exposed to smoke regularly, so do we adapt to it? Like our body is adapting, this microbial balance is changing.
No, I wouldn't say that we are adapting. I would definitely say that I would definitely agree to this, that our microbiome must be our microbiome may be changing. That I would agree with, but adaptation I don't know. But yes, our exposure is increasing, and so are our diseases. So taken together, we can say that yes, we are being exposed, and the incidence of the diseases are also increasing. So yes, our microbiome shift may be happening, but we cannot call it actually that we are adapting to it. We are just being exposed, and we are being victimized. We are not adapting.
And the balance is changing.
True.
And there is Dr. Subrat. He's asking, does microbiome differs in different types of cancer? So for different types of cancer, is it different?
I don't have I would believe yes, because with different types of cancer, it may differ, because all the cancers, they have their own metabolic routes and their own cycles which are there. So I would believe yes, but yes, I don't have data to support that.
Okay. So there is another question from Rajasri. What about the microbiome of a newborn? Is it born, or with time it acquires?
I would say both, because when he is born, he is exposed to his mother's microbiome. So what we see is like the babies who are born who have a normal birth as compared to the cesarean birth, they have a very different microbiomes because of the exposure that happens during the birth. And then coming to the second one, I would again say yes, because yes, the person, he is born with the microbiome, but when he is exposed to the environment, that alters the microbiome. So, so it's yes in both the cases, but that yes, it is both by birth as well as with the environment.
Then Dr. Roth again, as the microbe is living symbiotically, so is there any horizontal transfer of genes from microbes to human to adapt to the situation? So is there any transfer of gene or any influence?
A very good question, but again, we don't have means I don't have data support means about that. It may be like definitely there is influence, that's for sure, but horizontal transfer of genes, I'm not sure about that.
Actually, this question, what I was thinking of, this is one of the things that came to my mind. Like if someone has cancer, then there is some mutation of the cell or something, changes have happened. Now, there are microbes. So microbes is a separate entity. Human cell is a separate entity.
True.
Now, there should be some kind of connection between the microbe and the human cell so that there might be some modification in the cell. And I was thinking, I'm not exactly from this field, so just brainstorming. Now, these microbes have a lot of enzymes. Now, some may be intracellular, some may be extracellular also.
True.
So, probably, and enzymes are nothing but biological catalysts.
True.
And also, when they are exposed to some constraints, they change themselves.
Right.
And in that process, might be they are catalyzing some biological processes of human beings. Is there any study on this, that how these microbes are actually catalyzing the cellular processes in the human beings?
Actually, what happens is that the microbiomes, this is a very nice question, and it is a very insightful one, because what happens is that we see a microbial shift here. So when we are seeing a microbial shift, what you rightly pointed out is that these microbiomes, these microorganisms, they have a huge repertoire of enzymes. That is so what happens is that microbes, there are many xenobiotic metabolizing enzymes which are there, like the cytochrome P450s, and there are other so these kind of enzymes which are there, which can actually which can metabolize the xenobiotics. So what they can do is they can actually they can metabolize the pHs which are there, like they can hydroxylate them, and they can transform them into a more reactive form. So what happens basically is that when we are having cancer, at that time it is now well known that there are oncogenes which are present. So in a cancer model, we see that that cancer is basically like it is something like the total inflammation, the total inflammatory process which is that's gone wrong. So what happens here is that the proactive, that is activated molecules which are there, they can actually, you can say that they can work on this oncogenes, or they can change this immunological environment which is there, triggering the diseases or even triggering the cancer which is there. So even without even means directly going for a gene transfer, so they are altering the genetic composition. So they are causing mutations. So that can happen. But when we talk about a horizontal gene transfer, so yes, that may be possible, but yes, that I don't know.
Catalyze the process somehow.
They can catalyze because they have the enzymes, they have the they have the enzymes to metabolize these xenobiotics which are there.
And another question, see, every individual human being, they're different. So whenever you are performing such kind of study, it is difficult to use the controller, it's difficult to use the study the effect of the parameter.
Yeah.
Now, what I was thinking, why not we can think of using some machine learning process where we will have the composition of the microbes and the parameters of the human being.
Yeah.
Because a person might have a different diabetic level, different blood pressure level, and each of these parameters should have some influence on the microbiome. So if we include all the parameters and develop some kind of machine learning model, there one advantage we might find is that during experiments, it is very difficult to change one parameter at a time. So to really understand the effect of individual parameters, but in mathematics, we can do whatever we want to do. So we can change one parameter at a time and see the effect of that parameter on the particular output.
Yes, that's a big probability which is there, and that can be a very promising probability. But the only thing that is there, that moving forward, that may be the way out. But but for that, what we need to do is we need to train it. We need to train the network.
Right.
So in this machine learning, so the number of inputs which happens is very critical, because say we are having here as in case of firefighters, we have very limited population. So we have only 20 people or 30 people who are participating. So when we go in for a machine learning with a population size of 30 versus a machine learning with a population size of say 1,000 or 2,000. So when the machine is trained with the iteration of 1,000 or 2,000, so it will be much more accurate.
Right. But in machine learning, what we can do, say someone is a firefighter and someone is an ordinary man, but the parameters of those two people, if we combine, then what might happen? We can increase the sample size.
Yes, so that's a probability. Yes, we can mathematically do that, but but the only caveat that I can see there is, suppose there is an error, like errors in case that we haven't considered a parameter, or because in a human population, for instance, I'll tell you that we came across because here this human exposure, when we are talking about, this is a self-admitted one. Because we have the questionnaires, the people, they write that yes, we are exposed to this, or we are not exposed to this. That's what they do, basically. But what we find out is many a times we may run into errors with that, because we had a question. This was this interesting part. That was, we had a question that are you exposed to asbestos? So we had a lot of responders who responded negative. They said that no, we are not exposed to asbestos. But then we looked at their profession, at their profession, that what they were doing. So many of the people, they were in Navy. So so that cohort, they had served Navy in the 50s and 60s. So when we say that they had served the Navy in the 50s and 60s, that that time those submarines in which they were deployed, they had asbestos. So they were exposed to asbestos even without them knowing. So what may happen in these studies is that machine learning is definitely a way forward, but we need a very big population size. So
it's really a challenge for this to remove the noise also.
Of course, and if we don't do that, then what may happen is any error that may creep in, that will be magnified.
Right.
Because with more the iterations that will be happening, so the machine will take that to be true. The error will be taken as a reading.
Correct.
So and it will go on amplifying that. So that may ultimately result in drawing wrong conclusions. So that's what may happen. So that's the only thing. But with the increase in with the population size, we can overcome that.
And there is another question from Dr. Subrat. Is it possible to study the effect of microbiome in different human diseases by transferring the gut microbiome from human to mice?
Yes, that those possibilities, they are being studied now, and it's a very relevant question nowadays, because for studying these microbiomes, we do need, we need animal models. So what is happening nowadays is we are now coming up with the gnotobiotic models, that is the germ-free mice model. So in this germ-free mice model, what is happening is we are able to transplant the microbiomes which are there, which are available in the human population, or the microbiome which we are targeting to the germ-free mice. So using those things, actually using the gnotobiotic models, it is possible to study the effect of certain microbial communities on the health and disease. But again, it comes up with a rider, that is, that that is the mouse model, or the animal models which are there, that may not replicate the human health in its entirety, because their metabolism is very different from the human beings. That's why in the translational research, what we see, that the studies that has been carried out on the mice, they're very less percentage of very promising studies which we get on the mice is actually translated to human beings, because from the mice it has to go to pig, then it has to go to any mammalian species, and then only it can come into humans. So that is the limitation. But yes, with the gnotobiotic models we can do that.
And Dr. Roth has another interesting question. Like us, does microbes are selective to different kinds of smokers, like marijuana or cannabis? So if different kinds of smoking content varies?
I would say yes, because yes, because with the different different kinds of smoking, I believe the smoke composition may be different. Right. So with with different chemical compositions, different microbes would definitely have a selective pressure on the different kind of microbes. So the microbial community, I would expect them to be different. Right. So it won't be a surprise if a marijuana smoker has a different microbiome than a tobacco smoker.
And Dr. Somitri Sahu, is there any method to for early detection of microbiome in cancer? Any symptoms? So it's more kind of sensor she is thinking of, if it is possible.
Yeah, that's what we are trying to do. That is, we are trying to identify the microbial communities, like the Prevotella-C, which we are looking at. It showed a very marked, marked increase in the cancer patients, so as compared to the normal one. So Pasteurella-C and Prevotella-C we did see here. So yes, so that is that is what we are looking at. We are trying to use the microbiome both as a biomarker as well as for therapeutic intervention later on. So yes,
their byproducts also we can use as a non-invasive sensor probably.
Yes, like in this biosensor, yeah, of course there may be metabolites which are there, but here we are looking directly at the microbiomes. So yeah, that's also a possibility. I would say that yes, that may be a possibility.
Dr. Roth commented one thing, it's nice. So human mouth can work as a culture vessel.
Yeah, I would agree to that, because our body is a bioreactor. When we look at when we look at at our body at that time, we see that that we have the microbiome in our mouth, we have microbiome in our lungs, we have it on our skin, gut, everywhere we have the microbiome. And of course, and it's 3 to 5 times the size of the human genome. So yes, we are a bioreactor for the microbiomes. I would agree to that.
Microbiomes, it can be around 5 pounds.
Yeah, 5 times that of the human genome.
So it's a lot.
Yeah.
Writing somewhere. So I think there is no more question. Oh no, there is some. Okay. Oh no, there is no more question. So thank you, Dr. Bhattacharya, for this nice presentation. And maybe later we will have a discussion with you also, so that because I work on machine learning, so I want to explore a little bit.
Sure, we'll be happy to collaborate
Yeah.
and discuss more.
Thank you very much.
Thank you, sir.
Have a nice day.
You too. Thank you.
So now it's time for the next presenter, Dr. Narendra Pradhan.
So thank you, Dr. Sukanta Bhattacharya, for a nice deliberation and a vivid talk.
Thank you.
After
Pleasure.
that, it's our pleasure to host you. Then our next speaker is Dr. Narendra Pradhan from Uppsala, Sweden. So let me give a brief bio of Dr. Pradhan. So Dr. Pradhan had more than 10 years of experience in method development in collaboration with two biotech industries, that is biosciences and protein simple. Then he was keen in developing three analytical methods, that is capillary isoelectric focusing, the NanoPro plus PLA, then digital imaging for immunoblots. It's acquired excellent scientific communication skills, and to his credit, he earned more than 20 papers. And of course, I can see one Nature Communication paper just flashing my screen. So, to this eastern state of Odisha. And of course, he graduated from Rajendra College, Balangir. Then he marched on to Jawaharlal Nehru University for his master's as well as PhD with Professor Alok Bhattacharya. There he published many papers and got called from Uppsala, Sweden. And of course, he's more in, he's a kind of proteomics expert. And he has, then in Uppsala, he developed many skill and equipped himself. And he has, again, to his curiosity, to feed up his curiosity, he has one more MSc in Uppsala in business studies. And he is one of the 5 business incubator being selected worldwide to support and develop the entrepreneur in Sweden, particularly. Hopefully that's hopefully you'll enjoy the session, and we will mostly talk about the NanoPro technology. So now I'll hand over the session to Chair Professor Bhattacharya.
Thank you, Dr. Rath. So I would like to invite Dr. Narendra Pradhan to present his talk.
Hello, can you see my slide now?
Yeah, it's visible.
Okay. Thank you very much for the invitation, Dr. Rath, Dr. Santanu Bhattacharya, Dr. Dipankar Bhattacharya. It's a privilege for me to talk with the people from the same area. Just to brief introduction, like I live here in Uppsala almost 10 years now. And Uppsala is a really beautiful city. You can see from the slide, and this is the Uppsala is famous for many things. For example, this church, or this is the cathedral. You can see that this is the one of the longest or largest in Scandinavian countries. That is one. Of course, second one is the Uppsala University, which is famous. It was founded in 1477, and many things actually. It is not probably possible for me to describe all, but one of the important thing I noticed, like this is famous for the Carlos Linnaeus, who is the father of the taxonomy in the botany, and it is Recently, I have been to the Linnaeus Museum, and then it is fantastic to see how he nomenclature this all the plants he started. He has all the written documents are there, organized in many files and folders. Okay, that's all. And then about this, let's start it with the today's presentation. So, and the topic is. I cannot, sorry, ultra-sensitive and high throughput protein detection techniques for the human cancer. And in this today's topic, I'll just go a little bit more on this conventional Western blot and its pros and cons. And then we'll go to the nanopro technology, which I have used extensively. And that is what is the pros and cons, how it is used. And then this is the one of the recent technique which is developed in Uppsala University with Professor Ulf Lendergren. This is the rolling circle amplification, RCA, or it is also called in proximity ligation assay. And how we merge both of these two powerful technology, this NanoPro and the proximity ligation assay, and to test this if it is work in cancer biopsies. And we tested with the human colon cancer biopsies, and it works fantastically. So, okay, let's go to the first topic. This is the Western blot, and the first Western blot, it was there in 1979 when Tobin et al. They published the first paper in PNAS. And what are the pros? Because this is the technique, that this is extensively used in almost every protein labs across the world. It is so simple, and then it is nicely, you can get a very interesting result with short span of time. And it has so popular, so it has gone to the percolate into the each and every laboratory. So most of the people will know that what is the Western blot and how to do this. Then the qualitative detection of the single protein or a post-translational protein, when you have this phosphorylated protein versus the unphosphorylated proteins, or some other kind of a modification, then it can be easily detected through this Western blot. When you have the verification of protein, let's say you have the cloning and you have you wanted to test whether you get the protein production or not. It is a regular technique that people use and know that whether the protein is there or not. Also, many of the diagnostic purpose it can be used, it was using, such as HIV, and the list is go on and it is on and on because this is a so popular technique. But this is also, it is some challenges it comes with the Western blot, in the conventional Western blot. And let's see, this is the one of the Western blot where we did like many samples were run at the same time. If you see this each of the lane from the starting to end, every lane it is very equally charged protein. It is very well transferred. But what is the problem? The problem comes here. These two places where it was not transferred, this is a bubble and this is a bubble. And if you compare the protein from, simply you cannot use if you are quantifying this particular band. And this whole experiment you have to repeat or re-repeat. And this blot, this is a Ponshu S10 after the transfer of the protein. So it is a PVDF membrane. After the transfer, you can stain with the Ponshu S10, and then you just normally we used to do this with just to check whether the protein has been transferred properly or not. So, and this is another problem, or this is the HeadMac or HuBac primary human cell line was treated with growth factor with different time points, and then checked with the this phospho ARC1, 2 protein or total ARC1, 2 protein. And if you see, this is the with 7 minute, 15 minute, 30 minute, it has a very nice induction of the phosphorylated protein. Here it is also similar. But if you see here in this, because you need to strip the plot to reprobe with the total protein. So that is a kind of requirement we have to do with the conventional Western blot. And when you do the stripping, then this is the problem, that whether you are not able to 100% sure this is the, here the protein was there, or this is the stripped more than more stripping has been done so that the total protein has been removed. So this is the always this is the confusion, and that is another challenge. And this is one of the paper where we where Dr. Rath mentioned this nature communication paper, and we were being asked from the reviewer, like, this is a complicated experiment, and we had to do this for the because this is each sample, each lane is corresponding to mouse long lysate, and each lane was each mice was treated with different time points, and there are 9 mice here, and 9 mutant mice here, and 9 wild type mice here. And we have to do it like 4 different sets of experiment and 18 sample each time. And then we have to do the statistics. So that is where the problem starts. Then when you have this, all the experiment, because these 2 plots were run, because that's what we have been struggling like 4 months. I think 2 postdocs, one senior postdocs, and I was, both we were struggling with this to do the exactly similar. We were trying hard, because these 2 plots, it is the same experiment were done in the same time, same buffer, same setting, same quantity of protein charge in each lane, and still you can see some of the places or some of this part of the protein has not transferred, or something happened in each time. And it was very, very difficult to make the statistically validated. Ultimately, we had done, but this is the problem I'm emphasizing with this conventional Western blot. When it is a kind of yes or no question, whether you have one sample, you have no protein, another sample, you have the protein, it is fantastic, you can see the result. But when it comes to the quantification, it adds another level of complexity. When it comes to the cell lysate or primary cells or any cell line or cell lysate, it is quite easy to do it. And then next complexity, when it goes to the mice lysate or a human tissue, this is really, really tricky if you see the quantification part. So if you see what are the challenges of this, just to summarize, one thing is reproducibility is a major concern with this conventional Western blot, because this has been a lot of different steps involved, starting from the cell lysis, protein quantification, then SDS page, charging, loading, transferring. Each step can add some kind of some kind of human error, and that is what is happening. And then the quantification, when you have this only one gel, few samples, quantification still is not a big problem. Even if it's the principle is semi-quantitative, still you can manage to do it. But when, as I said earlier, we have many samples at a time, like more than 20 or 30 samples, it is kind of very difficult to do the quantification. And then it is the time consuming. As I said, we spend like 4 months to do only one experiment to make it like statistical significant or statistical analysis. And this is hugely time consuming as well as it, when you have the time consuming, that is also, it becomes more expensive. Then you have the limited number of samples. You cannot analyze more than 30 samples at a time. And when you have more than 30 samples and many gels, then it is hard to control, like what sort of control it, one blot to another blot, how do you control that one? You have to do many times. So, and then also the limited isoform detection, it means that when you have the phosphorylated ARC1 protein or phosphorylated ARC2 protein, you cannot do like a single phosphorylated ARC or double phosphorylated ARC1 versus the single phosphorylated ARC2 with the double phosphorylated ARC2. So it will be tricky. And always you need more quantity of samples. And then when in case if you have the biopsy samples or patient sample, and then you cannot spend all the sample because this is the patient, you cannot get the sample, that is so crucial. And then as we know that there's the stripping problem. So, and the last but not the least, this is the cost associated with each blot. And then if you repeat like 3 times or 4 times, you just have to add the cost. This is just approximate cost of a Western blot here in Swedish crown, there around 500 crown each time. But generally we forget to calculate the cost of the time. Each Western blot it takes almost 3 days to complete and then to know the result. So now the question, do we have a solution to these challenges of this conventional Western blot? And the answer is yes, we have. And it is a recent technique, which is I'm going to talk more about this technique and how we solve all this, like most of these challenges, and how it can be, and this is the NanoPro technology. So I just want to ask you that did you know about this NanoPro technology before this webinar? If you know that, it just type in the chat box yes, just to know. If you don't know, don't write anything there, so it will be easy for me to see that how many you know or not.
When I was checking your profile, I saw something about NanoPro. It uses some kind of capillary thing to separate the protein.
Yes,
and
yes.
Can analyze lot of proteins in very short time.
Yeah.
But earlier I didn't know about this.
Okay. I don't know if I am able to see this chat box or.
The answer is no.
Okay.
Now all the answers are no.
Okay, then I know that this is this is the recent technique, and then also the many people, they don't know that this is quite obvious. And
it is probably NanoPro, no?
Yeah, NanoPro. So I'll just explain this NanoPro, what are the steps it involves, and this is the one in the. Happens in everything happens within a single capillary, and this capillary takes only very few microliter of samples. Not even microliter; it takes only 400 nanoliter, less than half a microliter of sample, and then do all the analysis. So each capillary goes through the several rounds of, like, for example, it is the single capillary. Same capillary goes from first step to second, third, and to get this result. Everything happens within the capillary. So each capillary, you can imagine that one capillary is the one experiment. And then this is the first step where you take the cells or the tissues. You can take either cells or tissues or any other protein, make lysate, and put this protein lysate to mix with ampholytes. It is their buffer and the fluorescent isoelectric standards. This all get just mixed in the capillaries in a centrifuge, and then you just aliquot this sample into in a 396 well plate, and then this plate will go to inside the machine, and from there automatically each capillary which takes this fill with this lysate, ampholytes, everything, and then when electric current passed in the separating tray, Then this all the proteins. Here it only the difference with the STS space is here the separation takes place on the basis of isoelectric point of the each protein. So this is the, if there is a positive protein or a negative protein, this moves according to the cathode side or anode side. And when it's the charge is matches with the isoelectric point, there it stops. So this focus is automatically, and then it goes for the 40 minutes. And then the next step is after this separation or after the isoelectric focusing, you can allow the protein to immobilize to the capillary wall. So you put the UV light, and this is also automatic. The machine will put the UV light. You can of course set up how many seconds you want to put UV light, and then this all the proteins, separated protein, will fixed into the capillary wall, and then this all the material will flush out from the capillary. And the next step is to incubate with the primary antibody, and then again after 1 hour or half an hour, it's throw all the primary antibody, and then you put into the wash. How many washes you want to do, it depends on the experiment. And then you wash all the primary antibody, the extra primary antibody, I would say, and then put the secondary antibody. Again, with the secondary antibody is always like with the HRP level secondary antibody, which can give you the signal later stage. And then you put the chemiluminescence as substrate, the ECL, and then you get the signal here. And as soon as you get the signal or the light, then you can get a peak. And this graph of the peak or these peaks are recorded by a camera, CCD camera, and you will get always the result in the form of this graph. So you can get the several peaks, and you can quantify these peaks, and then you'll know how much proteins are there and particular to these peaks. And yeah. So this is the representation of capillary, with each capillary go through several rounds and to get the result. And this is why high throughput, because you can do like 96 capillary at a time, and you just run the machine in the afternoon, and in the morning you'll get all the results. So this is really, really very convenient and very high throughput. You can get huge amount of data in a short time span. So if you are still not clear, then it will be a little bit slide movie to see that how it happens in the real terms, and then probably it will be much more clear for all of you. Just a second. Oh, sorry. Can you see the movie? This one, is it working there?
Yeah, it's visible.
Okay, so this is the machine, Nano Pro machine, and this is the capillary. It is taking the capillary. So 12 capillary it takes at a time out of 96. So it goes for 8 rounds. And this is where you can see the sample. I put already the sample in this one. It's a different combination of lysate here. And then each capillary takes the protein, lysate, ampholytes, everything into this inside the capillary. And then now it goes to the separating tray, where already it was filled with the cathode and anode buffer. So this is the capillary tray, and here you can see this. And now the electric current pass, and all the protein are separated, or we can say focused on the basis of isoelectric point of the protein. So then it is the immobilized by UV light, and all this protein gets stuck into the wall of the capillary. And then it will wash, and then primary antibody incubation with the primary antibody. Here you can see this is the schematic, not the real one. So after that, the secondary antibody will come and bind to the primary antibody, which is HRP labeled. And then when you add the ECL, then you get this signal. And this will come in the form of a graph or peak. Okay, I hope it is clear.
Do you pass hydrogen peroxide here? Because you are using HRP. So is there hydrogen peroxide and some coloring material like ABTS or DTNB, something like that?
No, this is, I don't remember exactly this ECL, this is HRP, which what is the ECL?
Horseradish peroxidase you use.
Yeah.
So that's the enzyme.
Horseradish peroxidase.
Yes.
So it needs some hydrogen peroxide and some coloring material, actually, like there are some amines which produce red color.
Yeah, I think this is the kind of typical enzyme substrate reaction to get this color. Okay. Yeah, that's true. So, and this is the typical example of the conventional Western blot, and as well as the NanoPro side by side. You can see this is the when we do the conventional Western blot, you get this ARC1 and ARC2 protein in the two bands. And here, if you do the NanoPro, you can see this peak, double phospho ERK1, then this small peak, the phospho ERK1, then double phospho ERK2, and then ERK1, phospho ERK2, and then ERK2. So you can get the 6 different peak, and you can really quantify each peak, how many percentage of the peak is there, and then it will be much more. Here you can get 2 peaks and also the probably 2 another peaks are the phosphorylated peaks. But this single phosphorylation and double phosphorylation is you can see only here in the NanoPro. So what all the disadvantage here in this conventional Western blot is an advantage for NanoPro system. So this is the comparison between the conventional blot and then NanoPro. And here the main disadvantage what I found with the NanoPro system is every antibody does not work in this system. So you have to test the antibody, or you have to make sure that before running the final experiment, you have to optimize your antibody to get this peak, whether this peak is real or the false positive. So you have to make sure to run before this before going further for the experiment. So just to summarize the advantage: no gel run and no transfer, no stripping. It is all automated. So and it is a high throughput and high reproducibility, because you actually the human interference is very less in this procedure, and that's why you get this high reproducibility. So if you standardize and you have a method ready to go, then you don't need to worry, like you go just put the sample and then see the peaks. And exactly you can see this, you don't have the problem of this one capillary to the last capillary, like we have seen this in the conventional Western blot, like first blot to the second blot, there is a quite variability you can see. So one can run 96 samples. So in each day you can get double, like in the morning you can run one and then in the evening. So it is really a high throughput and you can get a huge amount of data. And if you have the valuable sample, for example, biopsies or isolated cells from the mice, or some of the, I know that this is some of the, when you isolate the cells from the mice, it is very low in number. And then it is, you cannot spend all those cells to do a simple Western blot. So that is useful. And these are the papers where it shows this, all this technique works perfectly. And you can also see that this, from the 25 cells, one can get a very qualitative signal through the NanoPro. Okay, now this is a busy slide, and then I would just tell you a little bit more on the how we did in this technique, how we used by using the human colon cancer samples from the patient. And this is the conventional Western blot. So we did once, and it takes so much time, not time, but it waste like at least 10 microgram of each sample. And then we stop this using this conventional way. And then we did, I think, 20 times. You can do one Western blot sample to do 20 or 30 times with the NanoPro. And this is the analysis with the ARC MAP kinase protein. And then you can see the peak, and all these peaks were quantified. And then the peaks were actually plotted against the, like, for example, here you can see this ARC1, and this panel is the normal sample, normal biopsies. Here the stage 2 cancer sample, and here is the stage 4 cancer sample. So, and you can do a lot of analysis from this ARC1, ARC2, total ARC, phospho ARC1, phospho ARC2, and phospho ARC versus ARC. So all this kind of analysis, but the point I'm mentioning here is like each peak you can take from here. And then if you analyze like number of other relevant proteins, and you can get also the many other peaks depending on the protein, what proteins you are analyzing. Also the post-translational modification, like phosphorylation, and other peaks you can see. And then if you take all this, all the protein peaks and analyze in a like systematic or system biology, this is the one like convex Hall, what we did. And then this was the interesting data. What we found is like when you see this total phospho ARC1, not the ARC2, and then this is the P70SK kinase and peak 3. Like when we say the peak 3, this is there were many peaks, but peak 3 was crucial for the study or analysis. And then SRC, this is the total SRC protein. SRC is one of the protein which has widely studied for the many different cancer, and it has been shown that like it is a crucial role in the stage 2 to stage 4. So that's why we were taking this stage, the SRC protein in the analysis. And when you see this one, SRC P6, like 6 number peak, and then third peak of the P70 as 6 kinase, and total per phospho ARC1, but not phospho ARC2 or any other protein. And if you combine these 3, and then you can completely separate this normal tissue to the stage 2 and stage 4. So that's very interesting. Like and this is the 2 of the, I think, normal biopsy where we have taken. Actually, it did not fall into this this category. So it needs a bit more, I think, more study and with the more number of study. But this is very interesting to see that and how the NanoPro technology help in detecting this normal versus the stage 2 or stage 4 biopsies. So now this is another technology I am going to speak, and which we have done, the proximity ligation assay. And this is the technique which was developed by the Professor Ulf Lenderg in Uppsala University, and we had a collaboration, and then we combined these 2 technology like NanoPro plus the proximity ligation assay, and how it is when the proximity ligation assay it runs inside the capillary, and what is the advantage and disadvantage of this combined technology. In the funnier term, we say this: we have done this marriage between this NanoPro and proximity ligation assays. And let's see, I'll go a little bit slow here to make you understand that what the proximity ligation assay is. So as the name suggests, like proximity ligation assay, that when there are two proteins are in the close proximity, let's say it's 30 nanometer or something, and then this technique works perfectly fine. So there are 4 steps. If you see here, this is a protein and this is another protein. And this is a primary antibody and there is a secondary antibody. This secondary antibody was in the both, it is the conjugated with the oligonucleotide. Here one oligonucleotide and here oligonucleotide is stretch of oligonucleotide is conjugated. So this secondary antibody is conjugated with the oligonucleotide plus or minus strain, which says one antibody it is with the one stretch of oligonucleotide. And then next step, when there are two proteins are in the close proximity and you it's the primary antibody bind, here is the another primary antibody bound, and then you have the secondary antibody with the one strand of oligonucleotide, and here another strand of the another pair of oligonucleotide. And then you put the like this is the stress of another set of oligonucleotides, which says if these two proteins are close enough, and then this oligonucleotide go and bind in such a way that one side, it is, if you can see here, one side of the oligonucleotide is hybridized with the plus strand and another side with the minus strand. And here is also, so it makes a circle. If these two proteins are close enough, and then these probes, oligonucleotide probes will go and bind in forming a circle. And then you put this in a ligase enzyme with the all the DNTPs, and then it form a complete circle with the ligase. So this is the now it forms a circle. And then you put all the DNTPs and tag polymerase to just like a normal PCR. And then what will happen? One strand will, depending on the which side of this three prime end, and then it will go and go and go and on and on and on. This is using as a circle. This behaves like itself as a template, and then also also you know that this is this is basically going on and on to make a long stretch of oligonucleotide. And this happens inside the cells or anywhere in the if these two proteins are there. And after that, you can put this oligonucleotide attached again the HRP probe, and then you can see the signal. So basically, this after this technique, it is generally its signal is hugely amplified, and then it is extremely easy to like very easy to find whatever you cannot see in the normal technology, you can see here. Okay, so what happens here when we combine this technology, and where do we combine this one? Okay, this is the step. We have created a space like in the program, in the machine. We have added these 3 different steps to accommodate this NanoPro plus PLA to work out. And then this is the step where after the secondary antibody, after the primary antibody, then in the secondary antibody place, you have the ligation, and then whatever steps it was required for this proximity ligation assay. And this is the then rolling circle amplification, HRP oligo hybridization, and then it goes to the normal chemiluminescence, and then you get the signal here. So let's see, this is the one. This is the normal scenario in the NanoPro, where you have a protein, then you have a primary antibody, then secondary antibody, and you have the HRP, and you get the signal. Here it is the NanoPro plus RCA. RCA basically it is the one primary antibody, but there are 2 secondary antibody, where one has the positive strand and then another one has the negative strand of the oligonucleotide stretch, and it goes on like rolling circle amplification. And this is the scenario in the RCA, NanoPro plus RCA. Here it is NanoPro plus PLA, where you have the two different antibody, two primary antibody and two secondary antibody, which is conjugated with this two different stage of oligonucleotides, or it is the one primary antibody which is conjugated with the plus or minus strand of the oligonucleotides. So in any way, it is the rolling circle amplification occurs in the both scenario. And then we test this whether it works in this when you combine this NanoPro and PLA. And you can see this result. This is the, we have analyzed the primary cell lines, and then you see the blue line is the normal standard NanoPro, whereas this green line is the NanoPro PLA. And this is the insert. You can see the quantification, and it increases several folds. And that is what we were expecting, and that we were very happy to see this increase of the so many fold. And then it can be potentially used for the other applications. So the next question we're asking, is this the one protein or one antibody? It works perfectly fine with this both setting. Is there any other proteins which we can use for this combined technology? And specifically, we were interested in the phosphoproteins, which is the one of the most difficult proteins to do the the experiments, because there are many disadvantages with the phosphoproteins, because you have to keep really what I say, that you have to keep the phosphoprotein intact, because it is very easily, it will detach the phospho part and then it misleads the result. So, and here we see this is the phospho-MEC1 and the phospho-SRC 418, and then this panel, it shows the NanoPro, and this panel which shows the NanoPro plus RCA, and here the quantification. So, and you see this is the all the other proteins also, it works perfectly fine, and it is of the phosphoproteins. So this is again the phospho-P38, and this is the housekeeping gene to see. So we have shown that this is the many protein, it works in the both the NanoPro and the PLA if you combine. So next we tested this one, whether it works for our cancer biopsies patient sample. And if you see here, this is the benign or normal biopsies, and this is the stage 2, and this is the stage 4. And this is the phospho-ARC1-2 panel. And it was not possible for us to see in this normal NanoPro. With the combination of the technology, then we can see the peaks. So that was very fantastic result. And then this is the phospho-MEC1, and then phospho-SRC1, phospho-SRC 418. And we all see this, and this is possible to see this, use the human cancer biopsy, which was earlier, it was not possible for us to see or visualize the signal. So then it comes to that one, what I talked earlier is basically the sensitivity of the technology. When you have both the technology combined, then this is sensitivity, it's greatly increased. But what about the specificity in the market? If you go or if you buy the antibody, the many antibodies are crappy, and also not validated properly. So what about that? Can you see some specificity of the protein? And then we took this normal NanoPro, you can see this one, and aldose reductase, and S100, S6, CDC34. And if you see this, always this, if you have the normal nano with the RCA or the PLA, then you can have the increased signal. Here, if you see there are many signals, many peaks that has been increased. But if you see this last panel, there are, you have a peak here, you have a peak here, but when you combine this both, there is no peaks when you have the NanoPro PLA. So that means these two peaks are coming from the normal NanoPro or NanoPro plus RCA was false. So in this way, you can detect the specific peaks or detect the, actually exclude out the non-specific peaks. So yeah, it is the conclusion is you can get the more specificity of the protein by using this both technology. And now just to conclude, the NanoPro technology and or in combination with the PLA, it could be potentially used for the diagnostic purpose. And signal intensity is greatly increased by RCA, this rolling circle amplification procedure, or the NanoPro plus PLA. When you combine this both technology, it has also the specificity, which is one of the important factor, has been increased. And this is to acknowledge, this is the Professor Lina Clason-Wells, where I was working mostly developing this both technology like NanoPro as well as in the combination of technology. And this is Professor Ulf Lenderking, who actually is the pioneer of the proximity ligation assay and RCA. And this is Dr. Jun Hong. And we were collaborating, like she was a PhD student of Professor Ulf Lenderking when we developed this combination of technology. And then Olink Bioscience is the company who have this PLA stops. And then Protein Simple is the NanoPro company. Both of them, they provided us samples. And this is all from my side. Thank you so much, and wish you a very happy and wonderful new year ahead. And I would be very happy to take questions if you have any.
Thank you, Dr. Pradhan. And wish you also a coming happy and wonderful new year.
Thank you.
New year. And there is one question from Dr. Roth. As there is no blocking step, how non-specific binding is evaded?
Yeah, that's a very good question, and also the relevant question, because this is the technique which works when the protein is in the natural form, so three-dimensional form or the folded form. So it is very difficult. It is quite difficult to get the signal from the specific protein actually in this technique. So you don't get generally non-specific protein, because most of the antibody, you know, that this is they are designed to bind in a denatured form of the protein. So, but here it is the sense it is so those antibody who works better for the immunoprecipitation, that works better here in this NanoPro technology. So you don't need actually, because you don't get any signal. So blocking step was not needed.
Right. I have a couple of questions. I will ask you later. First, I'm addressing his questions. Whether anybody in India is using this technique?
I don't know. Actually, this is. I know it is the people are using in Germany, France, and Spain. So not sure that probably they might have gone to India. Definitely, this is the company. The Protein Simple, now it is the Biotekin. I think that is the probably they are working in, but it is the in Germany, and then also the France, and many of the pharmaceutical company they have, I think, this technology they are using, because the drug screening with that is much more easier when you have the 96 different drugs or different samples at the same drug. You can screen quite efficiently.
Right. And how you modulate signal to noise ratio?
Here it is, you have a, like the software has what is called like this is the basic line. I think it
baseline, the baseline.
Yeah, the baseline it caught. I think after because it has the advantage of the software. I forgot that.
Actually, there are different baseline correction methods. Sometimes they use different techniques they use actually. Sometimes it may be a linear baseline, sometimes different techniques they use for algorithms they use.
Yeah.
And he's asking, is the technique translated in the industry?
This the both combine of the NanoPro and PLA that has not been translated to now because they're still working on that. I think there are bit complex. That we know that IP issues and these things are has to sort it out, and they're still working. The Professor Lendergren group. I don't know that how long it will take, and then what is their plan to like.
And he wants a cost comparison between NanoPro PLA system versus Western blot.
Yeah, this is a cost. Is definitely it will be always interesting, but it is relative term. When you see when we calculate the cost of the normal NanoPro, as I calculated, it is around 500 kroner for one Western blot. And if you repeat several times, and then you don't repeat here in the NanoPro, so cost will be actually similar, or you can have the less cost in NanoPro with more quality data. But again, it will, if you have not standardized this instrument, because it has an instrument cost and also the capillary cost and other consumables. So it's depend on the lab to lab actually how they look. But definitely the NanoPro and PLA, if you see this, because NanoPro has the good advantage with this taking the very little sample, 400 nanoliter. And then also the PLA reagents are also quite expensive. It is not the, yeah, you know that the scientific, the consumables are expensive, but that is, when you use the NanoPro in the capillary, this PLA, this reduces the amount of use. You know that you need the little sample from the PLA. So in another way, you reduce the cost for the PLA.
So even if you need to just do some additional experiments, you have enough sample left.
Sorry, you mean the...
Means you need very little amount of sample in this, so it compensates.
Yeah, exactly.
And he is asking whether the capillaries are reusable. I doubt.
No. It is not advisable to use the capillary because I think you cannot wash properly, fully. It cannot be washed. And this is the very sensitive. Once it is like you go through this UV immobilization, then probably you can have a lot of other false results. So it is not advisable to use at least at this point of time to reuse this one, because that is also the most crucial part of the experiment. The capillary, this is the light sensitive, and you have to preserve in a certain way.
So they used, I think, the UV ray to. They used UV ray. That's why probably there is some modification inside also.
Yes, and also that you cannot, probably there will be no stripping when you have the UV light put on and it fixed with the capillary wall, all the proteins. So you cannot, I think, strip after that.
Right.
Yeah, this is nobody probably not recommended to reuse the capillary.
And another question I have actually, it's about this sensor, because you are actually creating some kind of detector element for the protein. Okay. So my question is, say, if you have two epitopes and you have two antibodies there, now the distance between these two antibodies, there might be some influence in that. Because if the distance changes, then it might not be able to detect the right protein. Like
no, it is actually this is the interesting question, that this is how this separation is done by the isoelectric focusing point. So each protein, like different isoform, is separated because of this. They have different charge.
No,
And so
that
antibody is one.
I'm talking about the PLA part that you integrated in the NanoPro part. Because you actually created some kind of antibody system that is going to bind to some specific proteins.
Yeah, right.
Now the thing is, say you have two antibodies connected to the network. Now, if the network gets deformed a little bit, then the distance between these antibodies will also change.
Yeah, if it will not work if it is the more than 30 nanometer for sure. That is the PLA principle of the PLA.
Now,
So,
actually,
yeah.
why asking you this question? I used to work on biomimics at some point. The question is, usually in such cases, the choice of solvent is very important. Like they use usually acetonitrile for these kind of systems instead of using water or other solvents, so that the bond behind it, the molecules, they do not usually get deformed because of the dielectric. Is there any such limitation here also?
That interesting, but we have not checked with the other solvent or something, because it was part of, I think, recommended buffer we used from the protein sample, and also from the Olink Bioscience. I think we have combined these two, and then then it works, I think, perfectly.
Yeah,
perfectly
actually,
fine.
I used to draw molecules using ChemSketch software. I used to draw the target, and I used to identify the complementary functional groups, and I used to draw another molecule with the same distance. So that's how I used to work on biomimics, actually.
Okay, interesting.
And there is one question from Dr. Monish Paul. How much purified protein is needed to run this experiment?
This was what I have shown. It was from the total lysate. So if you have a purified protein, you really it is that the one of the figure it was shown in the human primary cell lysate, and it was from 4 nanogram of sample. So it was 4 nanogram of total lysate sample. So you need really, we have not done with the pure protein. Because pure protein you can do it like you don't need this much of sensitivity probably, unless you know that you do some kind of drug screening or something else. You need to spend a little bit extra money to do this one, because I would assume that if you have the pure protein, you need very little amount.
I have one question. If it is a pure protein, you can work with very small amount of protein, but in human samples, you might have lot of different proteins also.
Yeah.
So when your sample quantity is very less, due to non-homogeneity of the sample, we might miss some responses also.
Yeah, that is always there, because you cannot control with this is, you are talking about when you take the biopsies from the different patient, and which part of the biopsy you take, and then that is why it is good to have the like more analysis from the more sample or more proteins, more number of proteins, to take out or exclude the non-specificity of the analysis.
And Dr. Roth is again asking, however, we have to lyse the cell. We cannot detect in vivo. So the system does not work in vivo. That's what he's asking.
Yeah, this system works only if you do the PLA. that works in the confocal microscopic setting or the fluorescent setting. You can get within the cells or the tissues. But of course, this is in the capillary isoelectric focusing. You have to lyse the cells. Yes.
And just I'd like to know, what is usually the price of these equipment, NanoPro and PLA?
Oh, I don't know that actually. That is because it was we bought, I think, something, yeah, quite few years now, because we were developing with them, and also it's probably they have increased or decreased. I don't know that. This is
Because I can understand the working capital is really low, but I'm trying to think of the capital cost.
yeah. No. That is, of course, that's why I think it is the pharmaceutical industry where you have like, let's say we have the sample like for the MAP kinase analysis that is standardized, and then you don't need anything. Just put the sample and then switch on the machine, and it will go and then give you the result. And that is one of the protein which like many cancer patient, that because of this cell proliferation, and then almost I think every cancer that this MAP kinase increase, and then there is a correlation between this cancer stages and then with this protein. So if you have this, like, this protein has to be analyzed within the short time span, and then it is very advantageous, because it is not possible with this conventional Western blot. This is a lot of work, and also the lot of variation there.
Actually, we explained these complex procedures really, really easily, so we could really understand what are the underlying principles. So it was really nice, and the way you have just modified one step of NanoPro with this PLA, because that antibody part, there was not much specificity. So you introduce specificity by introducing PLA in that part.
Yes.
It was really innovative.
And that was very one of the advantage of this technology when you combine this one. Otherwise, you cannot know that which protein is specific or not. There are, of course, the method when you have suppose, let's say, the phosphoprotein, and then you put this the phosphatase, and then run with minus phosphatase sample with plus phosphatase, and if it goes out the phosphatase, then you can see the specificity. But that is another type of experiment.
Right. No, it was really a pleasure listening to you, and I thank Dr. Bhattacharya and Narendra Pradhan. I don't see any more question here, so I thank Dr. Pradhan and also Dr. Bhattacharya. It was really enlightening, and we learned a lot.
Thank you so much. Thank you so much.
Yeah, it was really a pleasure. So thank you very much for finding such a time. You just found a lot of time in your busy schedule, because I was there abroad, so it's really a tight schedule usually you have.
Yeah, only my kids are disturbing though.
Yeah.
That is the
At some point of time, I interacted with, I think, Dr. Anita Lloyd Spets of Sweden.
Okay.
So it's really a pleasure. And the way you explained, that was really commendable.
Thank you so much. Thank you so much. And then it was a pleasure for me to interact with all of you there. And then this was, and thank you, Dr. Roth, for inviting, and then this giving this opportunity to speak with all of you.
Thank you very much. So, Dr. Roth?
Yes, thank you, Chair, Dr. Professor Dipankar Bhattacharya, sir. And of course, I should thank Dr. Narendra Pradhan for waking up early. And of course, in this year end, though he was committed to many other things, but he took time and sacrificed his Christmas as well as the year end celebration, what his kids were demanding, and he committed to give the deliberation to the motherland in Western Odisha. And of course, I also thank Dr. Sukanta Shekhar Bhattacharya ji. So to take time, because he's heading a company, that's a big task. So taking time and delivering, I mean, taking then with 3 presentation he has delivered. The amount of work what he has done is really difficult to achieve in that very little span of time what he has spent abroad. We are very much thankful to the guest what we have today. Now I thank Professor Dipankar Bhattacharya sir and both the speaker. So we come to an end of the technical sessions. Now I hand over the session to my HOD, Dr. Santanu Bhattacharya.
Thank you. Thank you, Dr. Jyoti Prakash Datta sir. So, and good evening. School of
Dr. Bhattacharya, Dr. Santanu Bhattacharya, it is not audible at our end.
Sorry for technical issues. So, I must thankful our regional director, Mr. Pradeep Sarangi sir, and our principal, Somnath Sarangi sir, because within short of time they allow us to conduct this one-day national conference. And I must thank all the distinguished delegates from the India and the abroad, that namely Dr. Sandeep Danda sir, that he has excellently explained the scopes and opportunities of bioinformatics, and which was more helpful. And then Dr. Sonalisa Pandey. Then after that, in the second session, we have two excellent talk by Dr. Ranjan Nanda from ICGB New Delhi, and then from Dr. Niharika Shekhar Bhattacharya. On that, we have a excellent talk. We learned about the IPR and how to protect your invention. And in this session, we have a two excellent talk by Dr. Sukanta Shekhar Bhattacharya sir and Dr. Narendra Pradhan sir. So I thank all of the delegates and for their extensive speech, and who help us to make our all the participants to be advanced acquainted with the new techniques and new advancement in the biological research field. And I must thank to our Dr. Ranjan Nando and Ashwini Kumar Atto, and again Dr. Dipankar Bhattacharya sir to chair the sessions nicely, and they are for all the delegates. They take up the questions, and very smoothly they have organized the session. And I must thanks to the organizers, Dr. another convener, Dr. Jyoti Prakash Datta sir, our organizing secretary, Dr. Rukmini Misra, HOD from the Bhubaneswar campus, and Dr. Pudvarani Deep, and Mrs. Nomita Panda madam for helping us in organizing the seminar, this web conference in a short period of time. And it is not, but not the least, I must thanks to all our colleagues like Dr. Savarika Poeda, Dr. Kalpita Bhatta, Dr. Gyanaranjan Mahalik sir, Dr. Bhagashri Behera madam, Mr. Simay Pradhan, Mr. Aravinda Paricha, Mrs. Ms. Manjulata Palai madam, for their support to us to organize these events in a short period of time. And much thanks to for technical supports, Ms. Mrudula Satpati madam, Mr. Tamal Kumar Chattopadhyay and Mrs. Sudhanshu Kumar Chabdar for their technical supports to organize these events. And I must thanks to Mr. Bisal to supporting constantly, become the host and organizing from the morning to the day. And I must thank to all the participants joined here and have interactively participated and interact with the delegates in these occasions. So now I request Dr. Rukmini Mishra Madam to give the end of the concluding remarks on this session.
Thank you so much, sir. I must thank all the speakers. Really, my heartfelt thanks to all the speakers who took out time on a Sunday, and it was really very informative. All talks were from diverse areas, but I could connect so much. And if whatever it is like patents by Dr. Niharika or about the tuberculosis by Dr. Ramesh Nanda or by Narendra Pradhan, now he gave on this NanoPro. So all the talks were so nice, and I am very sure my students and all the students from different campuses, they might have gained a lot of knowledge, and will be conducting these seminars and conferences in coming times. And thank you to Jyoti Prakash sir for arranging such a nice conference. Also to Santanu sir, to Pradeep sir, Dipankar sir for his patience. Like whole day he is with us, and he is very such an active leader, I can say, and he always push us to do good research. So thank you so much, sir, and I thank you all. Thank you all the participants, all the students for having so much of patience and joining the conference. So thank you all, and wish you all a very happy new year ahead. Thank you.
Thank you.
Thank you, sir.
Shall I end the meeting, sir?
Yes.
Yeah, you can, please.
Okay, sir. Okay.
Thank you, Bisal.
Welcome, sir.
Thank you. A special thanks goes to Bisal Kadamji, sir, for arranging the everything seamlessly.
Thank you, sir. Thank you.
Thank you.
Thank you.